Bioinforange platformunun kuruluşunun 2. yıl dönümünde düzenlediğimiz Bioinfocongress IV etkinliğimize hepiniz hoş geldiniz.
Merhabalar, hoş geldiniz laboratuvarımıza.
Merhaba hocam, hoş geldiniz.
Merhaba, hoş bulduk. Sağ olun. Teşekkürler davetiniz için, organizasyon için.
Davet için tekrar çok teşekkürler. Organizasyon da çok başarılı gözüküyor. Çok teşekkür ederim.
Tekrar teşekkürler davetiniz için. Bugün bilgisayar teknolojisiyle biyomoleküler sistemlerin simülasyonu ve ilaç tasarımı konusunda konuşacağım.
Muhteşem. Çok teşekkür ederim. Evet, hepinize merhabalar. Organize komitesindeki arkadaşlara da çok teşekkür ederim beni davet ettiğiniz için.
Herkese merhabalar. İlk önce davetiniz için çok teşekkür ederim. Buradaki diğer konuşmacılarla beraber sizlerle beraber olmak gerçekten gurur verici.
Bioinfocongress'te sunum yapmaktan beni davet ettiğiniz için çok memnunum ve sanıyorum tamamen insan hücre fonksiyonlarına yönelik olmayan daha basit canlılarla ilgili olan tek konuşma belki. Ama böyle bir değişiklik getirmekten de çok memnunum.
Öncelikle hepinize teşekkür etmek istiyorum. Bioinfocongress oldukça güzel bir organizasyon yaptınız.
Tüm Bioinforange çalışanlarına ya da gönüllülerine teşekkür ediyorum. Beni davet ettiğiniz zamanınızı ayırdınız.
Merhaba, davetiniz için çok teşekkür ederim. Bu aktiviteyi organize ettiğiniz için de teşekkür ederim. Gerçekten çok büyük bir çalışma gerektiriyor.
Thank you, it's a pleasure. Thank you for the invitation.
İyi akşamlar. Başlamadan önce bu güzel organizasyonu düzenleyen arkadaşlara gerçekten teşekkür etmek istiyorum. Bu kadar büyük bir topluluğu bir araya getirmek, böyle bir organizasyon gerçekleştirmek gerçekten büyük bir başarı.
It's a really fantastic program that you've put together over these last few days. So it's been some brilliant science and some brilliant scientists speaking. So thank you very much again for the kind invitation.
Welcome everyone, I'm Neva Çalışkan. I'm a group leader at the Helmholtz Institute for RNA-based infection research. And today my talk will be about how genome, meaning of the genome can be altered by frame shift RNAs and how this response can be regulated by host and viral proteins.
Teşekkür ederim davetiniz için. Bugün de mRNA aşılarından bahsedeceğim sizlere.
Merhaba arkadaşlar, bugün sizlere katılmaktan çok hoşnutum. İlk önce Bioinforange öğrencilerine bu güzel organizasyonu bir araya geldikleri için teşekkür ediyorum. Gerçekten benden önceki videoları izlemekten çok hoşlandım.
Merhaba herkese, buraya geldiğiniz için teşekkürler. Bugün ökaryotik genomların organizasyonu hakkında bir konuşma yapacağım.
Merhaba herkese, katıldığınız için ve dinlediğiniz için teşekkürler.
Merhabalar, hoş bulduk. Başlamadan önce böyle güzel bir organizasyonda emeği geçen arkadaşlara teşekkür ediyorum. Ayrıca davetiniz için de teşekkürler.
It's a tremendous honor and pleasure for me to be here after starting my lab just over 2 months ago.
Gün boyunca 30 saat bir yayın süremiz oldu. 3 eğitim, 17 konuşma, toplamda 20 konuşmadan süren, görüyorsunuz yorgunluğumuzu da arka planda. Öncelikle bizlere konuşmalar, eğitimler veren 20 değerli hocamıza çok teşekkür ederiz. Açıkçası davet ettiğimiz hocalardan çok yüksek oranda kabuller aldığımız için bu kadar çok yoğun bir programımız oldu. O yüzden hocalarımıza çok teşekkür ederiz. Onların bize davetleri bu kadar çabuk bir şekilde, hızlı bir şekilde bizleri kabul etmesi, konuşmalar yapması bizler için çok güzel ve bizleri gerçekten çok motive ediyor. Hocalarımızın haricinde siz şu anda 277 kişi var bu saatte. Çok teşekkür ederiz. Değerli katılımcılarımıza çok teşekkür ediyoruz. Ancak büyük bir teşekkürü de ben buradaki ekip arkadaşlarıma yapmak istiyorum. Bioinforange'ın 2. yıl dönümü, doğum günü. Buradan Bioinforange'a emek veren, gönül veren, Bioinforange'ı seven tüm bilim severler, katkı sağlayanlar, öğrenciler, insanlar adına iyi ki doğdun Bioinforange diyorum. Bilim paylaştıkça güzel, bilim paylaştıkça ilerler. Bilimle kalın. Bioinforange platformunun bilim yolculuğundan 3.sünü düzenlediğimiz Bioinfocongress ilk etkinliğimize hepiniz hoş geldiniz.
Çok teşekkür ediyorum. Mehmet, senin şeyin nezdinde bütün organizasyonu yapan arkadaşları da tebrik ediyorum. Elinize, emeğinize sağlık. İyi bakın kendinize, görüşürüz.
Merhaba İvan Hocam, tekrardan hoş geldiniz.
Merhaba, teşekkürler davet için.
Report can you go very good time, transferring your knowledge to us and for this informative presentation. Kendim ve tüm katılımcılarımız adına çok teşekkür ediyorum hocam.
Ben teşekkür ederim.
Çok memnun oldum hocam, sağlıklı günler diliyorum.
Sizlere bir akşam.
Yep, thank you guys so much. I really enjoyed it. Great questions from everyone.
Merhaba Tuğba Hocam, tekrardan hoş geldiniz.
Hoş bulduk. Çok teşekkür ediyorum davetiniz için. Harika bir organizasyondu. Herkese selamlar.
Değerli vaktinizi ayırdığınız, bilgilerinizi bize aktardığınız ve bilgi dolu bu konuşma için kendim ve tüm katılımcılarımız adına çok teşekkür ediyorum.
Ben teşekkür ederim davetiniz için.
Merhaba Nilüfer Hocam, tekrardan hoş geldiniz.
Merhaba, hoş buldum. Beni iyi duyup görebiliyor musunuz?
Evet, görüşürüz.
Çok teşekkür ederim davetiniz için. Çok teşekkür ederim öğrencilerle böyle bu sanal ortamda da olsa bir araya gelmekten.
Merhabalar. Öncelikle davetiniz için çok teşekkür ederim.
Teşekkür ederim hocam.
Çok güzel bir event ortaya koymuşsunuz. Bu kadar değerli bilim insanı arasında yer almak gurur verici. Teşekkür ederim.
Çok teşekkür ederim. Nazik davetiniz için çok mutlu oldum. Bu arada bu hani YouTube'da olan sayın seyirciler, bu saatte cumartesi akşamı gidip başka bir şey yapmayıp bu konuşmayı dinlemeye geldiğiniz için size de hürmetlerimi sunuyorum.
Çok teşekkür ediyoruz. Hem kendi adıma hem de bütün katılımcılarımız adına.
Rica ederim. Ben çok memnun oldum. Beni onure ettiniz. Çok teşekkür ederim.
Biz onur olduk hocam. Çok sağ olun. Size çok teşekkür ediyorum. Katılımcılarımız ve kendim adına.
Ben de çok teşekkür ederim. Benim için de güzel bir deneyim oldu.
Bu harika sunum için katılımcılarımız ve bütün ekibimiz adına çok teşekkür ederim hocam.
Ben çok teşekkür ederim beni misafir ettiğiniz için.
Thank you. Thank you so much for this space. Thank you for having me here and for this opportunity.
Bilgi dolu konuşma için kendim ve tüm katılımcılarınız adına çok teşekkür ederim. Ben teşekkür ederim davetiniz için.
Tekrardan çok teşekkür ediyoruz. Katıldığınız için çok mutlu duydum.
Ben çok teşekkür ederim. Çok güzel bir organizasyon gerçekleştirmişsiniz. Beni davet ettiğiniz için tekrar teşekkürler. İyi günler diliyorum.
Biz teşekkür ederiz hocam. Bioinforange platformu olarak düzenlediğimiz Bioinfocongress 3 etkinliğimizde çalışmalarına yurt içinde ve yurt dışında devam eden değerli bilim insanları katılımıyla toplamda 15 seminer gerçekleştirmiştik. İzninizle burada organizasyonda emeği geçen arkadaşlarımı davet etmek istiyorum. Şimdi burada ekranda olan arkadaşlarımız haricinde kongremizin düzenlenmesinde organizasyonda emeği geçen tüm ekip arkadaşlarımıza teşekkür ediyoruz. Ve sizler gibi yani burada hepimiz öğrenciyiz biz ekip olarak. Özellikle öğrencilere bir katkımız oluyorsa akademik hayatı ve işte gelecek idealleri konusunda bir fikir verebiliyorsak buradaki konuşmalar, işte bizler bir katkı sunabiliyorsak açıkçası biz bundan çok mutluluk. Tekrardan etkinliklerimize devam edebilmeyi umuyorum. Bizi sosyal medya adreslerimizden takip edebilirsiniz. Sizlerin desteğiyle biz motivasyon buluyoruz. Bizler motive olunca da böyle güzel etkinlikleri sizlere sunmaya çalışıyoruz. O yüzden karşılıklı etkileşim ve destekleşmeyi umuyorum. Tekrardan hepinize çok teşekkür ediyorum. Katıldığınız için de çok teşekkürler tekrardan ve herkese bol başarılı akademik kariyerler, güzel bir hayat, sağlıklı bir hayat diliyorum ve sağlıklı günler diliyorum. Görüşmek dileğiyle tekrardan. Hoşçakalın. Merhaba, ben Mehmet Çalıseki. Bioinforange platformunun kurucusuyum. Bioinforange platformunun bilim yolculuğunda dördüncüsünü düzenlediğimiz Bioinfocongress 4 etkinliğimize hepiniz hoş geldiniz. 6 Şubat tarihinde ülkemizde gerçekleşen depremlerde hayatını kaybeden insanlarımızı anıyor, ailelerine ve ülkemize tekrardan başsağlığı diliyoruz. Depremde biz de ekip arkadaşımız Zeynep Nur Çıra'yı kaybettik. Kendisini sevgi ve özlemle anıyoruz. Tüm sevdiklerine başsağlığı diliyoruz. Kendisiyle birlikte ailesinin kurtarılması için sosyal medyadan çok uğraş verdik. Kendisini bugün aramızda görmek çok isterdik. Üzgünüz ve unutmayacağız. Depremde ve sonrasında bilimin önemi bir kez daha öne çıktı. Ülkemizde bilime ve bilim insanlarına gerekli değerin verilmesi ve onların dikkate alınmasını umuyor ve istiyoruz. Bilimin ışığında aydınlık günler görmeyi ve canlarımızı artık kaybetmemeyi diliyoruz. 2020 yılının Mart ayında bilim paylaşımı amacıyla faaliyetlerimize başladık ve bu kongre ile birlikte Bioinforange platformunun 3. yaşını kutluyoruz. Öğrencilerden oluşan ve kar amacı gütmeyen Bioinforange platformu olarak hem dergimiz Bioinfojournal'ın 6. sayısını yayınlamak hem de çok değerli bilim insanlarının katılımıyla 4. kongremizi gerçekleştirmenin heyecanı ile buradayız. 2 gün boyunca gerçekleştirdiğimiz 12 konuşmadan 6 konuşmaya katılan katılımcılarımıza İngilizce katılım belgesi gönderilecektir. Buna ek olarak katılımcılarımız vereceğimiz diğer değerli armağanları kazanma şansına sahip olacaktır. Yoklama formları konuşmanın bitiminde soru cevap bölümü esnasında açılacaktır. Yoklama linkleri yalnızca 10 dakika açık kalacaktır. Bu konuda ve diğer kongremizle ilgili birçok konuda aklınıza takılan herhangi bir durum olduğunda Bioinfocongress IV sitemizi ziyaret edebilir ve paylaşılan kongre kitapçığımızda gerekli bilgileri bulabilirsiniz. Hepinize tekrardan hoş geldiniz diyorum ve herkes için iyi bir organizasyon olmasını diliyorum. Görüşmek dileğiyle.
Bioinfocongress IV, Associate Professor Dr. Pınar Uysalonganer, MicroRNAs as Biomarkers in Cancer, Opportunities and Challenges. Associate Professor Dr. Pınar Uysalonganer is a reader in molecular biology and cancer mechanism research group leader at University of Westminster, London, UK. She has finished her PhD by completing her studies both at Center for Hepatology at UCL and the Medical School of Marmara University in Turkey. Her research experience involves a range of cell signaling mechanisms such as VNT signaling, microRNAs, and the functional and molecular aspects of cancer. She has also been actively involved in fundraising activities with charities such as the Cancer Research UK, the Pancreatic Cancer UK, and the Prostate Cancer UK. She is an Ethnoswan coordinator of University of Westminster and runs team activities for local schools, and elected as Fellow of American Chemical Society and an ambassador of EACR. Dr. Uysalonganer completed Prevention of Cancer and Control Summer Curriculum at National Cancer Institute, NIH, at 2017. She became a Fellow of HEA at 2014 and Senior Fellow at 2018.
Dear Associated Professor Dr. Pınar Uysal Ongener and participants, welcome all of you to our Bioinfocongress IV event. Associate Professor Dr. Pınar Uysal Ongener will give us a talk today on microRNAs as biomarkers in cancer, opportunities and challenges. At the end of the presentation, we will have a question-answer section prepared by our team and consisting of questions from our participants. Dear Associated Professor Dr. Pınar Uysal Ongener, welcome back.
Thank you.
Thank you so much. If you wish, you can start your presentation.
Trying. Yes, here we go. Thank you so much for this invitation, and good afternoon, everyone. Thank you so much for being here for this. I don't know how is the weather where you are, but I guess it's a very nice Sunday afternoon. So I am going to talk about microRNAs today, and I'm going to give a couple of examples of my lab, what we are doing, why we are doing those experiments, those projects. So let's start. So sorry to start with slightly, I guess, disheartening slide. According to the CR UK, let me just make my slide bigger. According to the CR UK, 1 in 2 people in the UK, this is not only about the UK, obviously, but like in general and worldwide, will get cancer in their lifetime. In an extremely basic explanation, as I'm sure you all know that the cancer is abnormal cell dividing, and some cancer may eventually spread other tissues. So the good news is we do have lots of very clever techniques over the years that we came across with lots of different challenges when it comes to cancer. However, we developed a lot brilliant opportunities to tackle cancer. Once again, very briefly, if there's a cancer, if there is a solid tumor, especially I'm talking about, we can easily cut the tumor, that is surgery. And then we can poison the tumor, that is chemotherapy. And then we can burn the tumor, that is radiotherapy. However, the cancer is still a huge global health problem, because most of the times it's not the cancer itself, but the metastasis kills people. Let's look at what metastasis is. In this, once again, extremely basic slide, you do see the primary tumor formation. In here, there are very abnormal cell growth we can see. However, they are still like very compact. And then when we move on to the local tissue invasion, obviously after a while, these cells, because they just keep dividing abnormally, there is not enough nutrients and there is not enough space. Therefore, they need to jump on this is a local tissue invasion. And then early into the vessels, they do need to jump on a vehicle, in other words. And this is most of the times a bloodstream. And then they do need to survive in the bloodstream, and then they need to move on to the different parts of the body. And this is what we call metastasis. Once they are escaping from the bloodstream, and then they start new colonies in the different parts of the body. So this is, in fact, is the problem when it comes to cancer treatment, because, let me just go back, because as I tried to very basically explain to you earlier on, if we know where the cancer is, once again, I'm talking about the solid tumors, we can easily tackle that. We can just remove the tumor, and then we can just use either chemotherapy or radiotherapy, and then we can just see whether the patient is going on healthily. However, most of the times, this like escaping cells, tumor cells, jumping on the vehicle, the vessel's bloodstream, and then like moving on to the different part of the body, it takes time. And most of the times when we diagnose people with metastasis, it is already too late. So it is already spread all the other parts of the body. So this is the actual issue with the cancer itself. So my lab focuses on cancer metastasis. We are basically trying to understand the mechanisms of the cancer metastasis. Moreover, we are trying to find out better biomarkers to understand which tumors are going to metastasize. And if we can come up with better biomarkers, of course, we're hoping that this will also open more doors when it comes to the therapeutic approaches. So one of those things that we have been focusing on, one of those potential candidates, is microRNAs. Well, microRNAs, hence the name, they are non-coding small RNA strands, single-strand RNA molecules. They are around like 18 to 20 nucleotide long, especially since the Human Genome Project, as again you know, it is completed around 2003, and we found that completion of the Human Genome Project, huge part of our body is not required for protein synthesis. So since then, we are trying to understand why do we have this, we used to call them junk, we don't call them junk anymore, this big part of our genome, what is happening? Why do we have this non-coding part of our genome? So one of those parts is microRNAs. The other one is, these are non-coding RNAs, the other ones are link RNAs, long non-coding RNAs. Today, I am going to talk mostly about the microRNAs. As I said, under the normal conditions, these microRNAs are responsible for controlling the cell growth, proliferation, differentiation, and the cell death. So they are like, it's been shown that one microRNA, as I told you, they are quite small single-stranded RNA molecules. They can control almost 100 different mRNAs expressions. You can see that they are quite small, but they are quite powerful. So if something goes wrong with the microRNAs, this time we do have problems such as cancer. So we know, again, because of the research, we know that some microRNAs, they behave like tumor suppressors. We call them tumor suppressor microRNAs. And some of them, they do behave like oncogenes. That's why we do call them oncomers. So as a summary, like a long introduction summary, one microRNA can moderate the gene expression at the post-transcriptional level. They can bind to the complementary sequences in their target mRNAs, especially from the 3' untranslated regions. Due to the interactions between the 3' end of the mRNA to microRNAs, they can control the expression of several mRNAs. So that means they can also involve in several signaling pathways. And also, it's been found that they do have significant role in several biological processes. Those processes, as I tried to show you in the previous slide, cell differentiation, cell survival, proliferation, invasion, apoptosis, angiogenesis, metastasis, and lipid glucose metabolic pathways. So only this part, if you see, we are already talking about cancer. If something goes wrong, so all of these things are going to affect it. So therefore, the cells, the cancer cells, are going to behave more aggressively. So let's start looking into the projects that we have been doing over the, I guess, like 5 to 6 years. So the first project is breast cancer and microRNAs. In this introductory slide again, I'm just trying to show you where the stroma, where the duct is. So you know, again, I'm sure the breast cancer is the second biggest killer in the world, and it is the biggest killer amongst women. We did have a lot of success in terms of the breast cancer treatment over the years. There are most of the countries, they do have screening programs. However, the breast cancer is quite, as all the cancers are, quite heterogeneous. So we do have hormone dependency and we do have hormone independency when it comes to the breast cancer. Obviously, these hormones are progesterone, estrogen, and EGF hormones. So what I'm trying to show you here, if you look at the normal breast, you can see the stroma, you can see the duct, and I hope you can appreciate there is a beautiful differentiation, and it is beautiful, in a way, order between the stroma and the duct. When we look at the benign lesion, still there is an abnormality happening. You can see that the hyperplasia region in here you can see that there are lots of cell division occurs, but you can still see this like beautiful ordering. So the differentiation, in other words, maintained in here. Whereas when we look at the malignant breast cancer picture on my right-hand side, you can see that it's just like in a way it's a mess. So you can see all those like green bits, they were like stroma, remember? And then the duct, so they sort of like divided and they sort of like in between each other. So this is the actual problem that we are trying to understand in my research. So in this particular project, we just wanted to see microRNA 21, which occurs or which behaves like an oncomer. So in other words, we do see the higher expression of microRNA 21 in pretty much all of the cancers. We wanted to see whether the microRNA 21 is overexpressed in the breast cancer. If so, does it really do anything in terms of the overall survival? For this purpose, we went to the TCGA breast cancer database and we wanted to see whether there is any correlation between the MER21 expression and the overall survival in almost like 1,000 patients in total, 995 to be exact. So in the left-hand side, you can see the Kaplan-Meier curve. And what you should look at, so in the red bits, you can see that if there is a higher microRNA expression in the patients, they don't survive as much as the patients that they do not have higher microRNA 21 expression. So this was our starting point. So then we wanted to go back to the lab. So we wanted to see whether we are going to see any hormone dependency. So by using the cell lines, MCF7, SKBR3, and MDA231 cells, these cell lines are very, I guess, like popularly used in all around the world. So we did see the triple negative breast cancer cell has the highest microRNA 21 expression. This pretty much fits our hypothesis because this microRNA 21 higher expression we did see in MDA-MB231 cells. This cell line is, as I said, triple negative, but most importantly, this cell line has the most metastatic ability when you compare to the other breast cancer cells. For instance, this MCF7 cells, they are also metastatic, however, they are very weakly metastatic because they also have the hormone dependency. So we did use MDA cell lines as a cell model to be able to understand the role of microRNA 21 in breast cancer. So by using the CRISPR gene editing technique, we targeted microRNA 21, and then you can see in this bar chart next to it. So when there is a GFP or the control MDA and B23 cell lines, and then we have like 4 different guide RNAs. We transfected the same cell line with 4 different guide RNAs, and then we found that 2 of our clones they show the lowest microRNA 21 expressions. So from now on, we use the clone 2 and clone 4. So another important area when it comes to all the cancer metastasis, and also the cell signaling, as again, I'm sure, none of the cells, they can live in isolation. They do need their neighboring cells to be able to communicate with the neighboring cells. They do need to express, they do need to secrete certain factors. So therefore, the secretion and then endocytosis are quite important in terms of the healthy communications between the cells. When it comes to the cancer, this becomes even more important because we know that the one cancer cells, they can secrete growth factors, the proteins that the other cell lines, the neighboring cell lines, can also take of these ones, and then they can behave almost like the cancer cells as well. Just to test this hypothesis, we wanted to look at the extracellular vesicle release and the cargo change, whether the cargo is changed when we CRISPRed, when we remove the microRNA from the MDA, MB231 cells. For this purpose, again, we looked at both of the clones 2 and 4, whereas in here you can only see the example of the clone 2 versus control. What do we see from this image or this slide? I hope you can see it in here in the D. Total EV released reduced when there is no microRNA 21. So with the control cells, they did have like more release, extracellular vesicle release, whereas if there is no microRNA 1, the cells release less extracellular vesicles. Moreover, the cargo, when we extracted these vesicles and then looked at their cargo, what they carry, when we found out the microRNA 21 CRISPRed cells had a lot less microRNA 21 than the controls. So so far it was good, but we wanted to see what does it mean. So is this going to affect the cell growth, for instance? So we looked at the cells again. So once again, you do see in here the wild type and the one of the examples of the knockout cell lines. We just started to grow the cells, but we sort of looked at them in 48 hours to 72 hours, in 2 different time points we use. If you look at the top panel in here, you can see that with the wild type MDA NB231 cells, you can see that the cells are growing like colonies, and you can see that they are sort of started to spread around. Whereas with the no microRNA clones, CRISPRed clones, you can see that they sort of stuck in compact. So they are still growing, but they cannot spread as much as the control. As I said, this experiment only done up to 72 hours. We didn't move more than 72 hours. But even in 48 to 72 hours, we started to see the difference between the wild type versus the knockout clones. Another marker that we use when we look at the metastasis, EMT phenomena. So EMT stands for epithelial mesenchymal transition. It is important because, again, mostly in solid tumors, in the epithelial tumors, we do see the epithelial cells are really nicely elongated, and they are, as I said, they do need their neighboring cells. They do have connections with the neighboring cells and also the surface. So if the cells are looking and behaving like epithelial cells, the marker for them is E-cadherin. If during the cancer we do see this, if these beautiful looking, well-behaved epithelial cells going through the dark side, this time they do have less E-cadherin expression, but they do have more mesenchymal markers expressions. Those mesenchymal markers are, in here we use snail, zeb, and vimentin. But mostly in the literature, people are using E-cadherin and vimentin as a marker of the EMT epithelial mesenchymal transition. In this slide, hopefully you do see with the control cells, there are almost no E-cadherin expression, meaning that these cells are already quite aggressive. They don't need any connections between the neighboring cells. Therefore, the E-cadherin expression went down. Whereas when we remove the microRNA from these highly metastatic triple negative breast cancer cells, we did see that the E-cadherin expression started to occur, hence the greenish signaling here. So when we move on to the mesenchymal markers, as I said, we use snail, Z1, and vimentin. Let's look at the vimentin. With the MDA NB23 cell, we did detect high level of mesenchymal marker vimentin. This was similar to the snail and Z1. However, when there is no microRNA 21 this mesenchymal markers started to disappear. So the expression level of the snail, ZEB1, and the mentin reduced. So this was quite an important result for us. So we moved on to the invasion markers. We showed that when there is no microRNA 21 invasion, matrigel invasion also reduced. So we published this data, I think it's almost 2 years now. And currently we are trying to generate another cell line and trying to understand whether this microRNA 21 is working in the similar line with another triple negative breast cancer cells. So this was the breast cancer. Let's look at the prostate cancer, whether we are going to see the similar approach on prostate cancer. Let's start with the stats again. According to the Prostate Cancer Research UK, 1 in 8 men over the age of 50 is diagnosed with prostate cancer. Prostate cancer is similar to the breast cancer in terms of the hormone dependency. This time our hormone is androgen, not estrogen, but androgen. But there is also something different from the prostate cancer. Most of the times, or at least until very recently, the approach was watchful waiting. So in other words, the clinicians were saying that, well, the prostate cancer is, you can see it in here, so mostly is diagnosed in the men over 50 years old, and it is a mostly slow growing cancer type. So instead of like interfering, instead of using our clever techniques that I mentioned to you earlier on, let's just like keep observing whether the cancer is metastasizing or just like being more aggressive. So this was the approach for a while. However, they found out very recently this is not working really well because more and more people are now dying from prostate cancer. The reason for that, the main reason for that, we still don't have a really good biomarker for prostate cancer. There are like 2 ways to diagnose prostate cancer. One is the digital rectal examination. Another one is the blood test called PSA, prostate specific antigen. Everyone knows that the PSA is not specific enough. It doesn't show us, it doesn't give us any more information than the high or low. However, this is still the case in, again, all around the world when it comes to the prostate cancer detection. So when we look at the molecular level, so you can see that the progression pathway for human prostate cancer. So this time, again, very similar, I believe, for the previous example. You can see that in the normal epithelium, these cells are like beautifully aligned in here. And then we move on to the metastasis. We do have like different stages. The first one is the PIN, prostatic intraepithelial neoplasia. You can see that between the normal to the PIN, there are like certain genes activated. So one of them is the very important one is the P10. P10 is a tumor suppressor. Almost all of the prostate cancers, we do see the P10 inactivation. Another important one is TMPRSS-ERK fusion. So 2 of the genes, TMPRSS and ERK, they do break and they become a fusion. And again, like almost 60% of the prostate cancer patients, we do see the TMPRSS-ERK fusion. And then we have the adenocarcinoma. I'm sure you already noticed that the cells are becoming like a bit more aggressive, and they started to go out from the normal epithelium, and that adenocarcinoma. And you can see the metastasis. And most of the time, when the metastasis occurs, the treatment doesn't work. So we start to see the castration resistance. We started to see the EMT markers again. E-cadherin goes down, mesenchymal markers such as vimentin goes up. So this is what happens normally. So we wanted to see if the P10 deletion in the prostate epithelium is correlated with the microRNAs, or whether the microRNAs are doing something with the P10. for this purpose, with the collaboration of the Institute of Cancer Research here in the UK, we use P10 knockout mouse model. This is not the human study, this is the mouse model study. So for this, for the mouse, if there is no P10, they don't completely grow for the metastasis or the prostate cancer, but they sort of like stuck at the pin, interplasia, which was a lot better for us because we wanted to see, especially with the P10 involvement, whether the microRNAs are also the early event of the prostate interplasia. So for this purpose, again, you know, when we have this mouse models, we just took their tissues and then we generated the sections of the prostate tissues. And you can see that this is a very long, big paper, but I just wanted to show you this. For this paper, we looked at the wild type versus the P10 knockout mouse models. We wanted to look at the microRNA 21 and microRNA 155, which is another oncomer, behaves like oncogenes. So we did see that with the wild type, almost like there is no change in terms of the microRNA 21 and 155. Whereas if there is no P10, both of the microRNAs are highly upregulated. So our aim was just to look at the inflammatory pathway as well as a response to the carcinogenesis, because in cancer, most of the times inflammation goes hand in hand with metastasis or cancer. However, we didn't see this, but the important part of this paper, we found that there is a oncomers such as 155 and 21 show a significant effect of the gene expression profile. When we inhibit the P10 signaling cascade, we did see the reduced expression of the microRNAs significantly. And we've also shown that the microRNA inhibitors to reduce the cell growth and to re-express several and the previously downregulated genes. So there is a clear association between these two microRNAs, microRNA 21 and 155, and the P10 expression in prostate cancer. While this study was going on, I was also trying to optimize a method in the lab, which was the microRNA in situ hybridization. So for this purpose, we already had human cells and human tissues, prostate cancer tissues. So if we first wanted to be able to optimize it, we first wanted to look at the Gleason 7s. I didn't mention about the Gleason. So the Gleason scoring is, again, another routine technique in the histopathology labs. So if mostly we do need to see if the Gleason 7, if the patient diagnosed with Gleason 7, you can see that there is a prostate cancer, but this is mostly localized. So the cancer is there, but it is localized in the prostate, and it is no metastasis so far. So if there is a Gleason 7, like lower than Gleason 7, this is in a way better, because this time it is a very early diagnosis of prostate cancer this patient has. If there is a bigger than Gleason 7, that means that cancer is already advanced and most probably the hormone independent at that stage, and most probably it has already metastasized. So this is the how the clinicians use the Gleason 7 Gleason scoring. We first wanted to start with the Gleason 7, that is almost like the mid-range localized tumors. For this reason, we collected 20 patient samples. These are tissue samples taken with the needle biopsies. They were like used with the sectioning. And you can see that we did see in here as well, this is sort of like the purplish color that we do see, I hope. So with this purplish part, which is a prostate, and you can see prostate cancer, you can see that there is a higher microRNA 21 expression in the human patient samples. When there is a lower Gleason, when the prostate cancer is detected in an earlier stage, this time almost no microRNA 21 expression we did see. Whereas if you move on to the higher Gleason score, we did see a higher microRNA-21 expression. However, if you look at this chart, you can see that although the sample sizes are slightly different, so you can see that there's almost like a bell curve going on. So it's not like a straightforward curve. So you can see that there is a lower microRNA-21 expression with the lower Gleason score, which you would expect. And then when there is a Gleason 7, almost like a localized prostate cancer, you do see a higher expression. Whereas when there is more metastatic patient samples, you can't see even like higher. It's just like sort of drops. That sort of tells us perhaps the microRNA-21 expression triggers the prostate cancer, even the metastasis, but once the tumor reaches to the aggressive stage, it is not needed anymore because the tumor is already metastasized. So we also know that microRNAs can be released into the bloodstream by the tumor cells. So the apoptotic cells and the active cells, I've given you the example in the breast cancer. I told you that the cells secrete extracellular vesicles we looked at for the breast cancer project. So this is pretty much like the everyday event. So we do have so many secretions going on. And I also told you that with the prostate cancer, we have a problem in terms of finding a better biomarker. So for this reason, we just wanted to look whether the microRNAs can be detected from the bloodstream. These are the circulating microRNAs this time. So we have already shown that we can detect and we can show microRNA expression in the tissue samples, both in the mouse models and the human samples, and also obviously the cells. But we wanted to see whether we can just come up with a potential biomarkers, better biomarkers by using the non-invasive techniques such as like taking a blood. Again, it's not a brilliant image for the Sunday afternoon, I believe, but you also know that obesity and the high-fat diet promote a prostate cancer progression. So that means we have two aims for now, right? So one, we are trying to find a better biomarker, non-invasive better biomarker for prostate cancer, but also we wanted to see whether there is any association that we can detect in terms of the different microRNAs. Is there any microRNA that we can find out which is the obesity related? And then we already have a couple of markers, couple of candidates for prostate cancer. Can we make these biomarkers, microRNAs, to detect the obesity and the high Gleason score, obesity and low Gleason score? Right. This is a very beautiful experiment, I like to say. So we found that 69 volunteer patients for this project. So we wanted to look at, and they were like grouped in different parts. So for instance, 41 patients, they do have the high BMI. BMI is an index that body mass index that we do use to, again, decide whether someone is obese or not. There are lots of controversy in the clinic, in the scientific world. Most of the people are saying that the BMI is not a good marker for the obesity, because many of us, we do look like slim, but it doesn't mean that means we have a low BMI, but it doesn't mean that we are healthy, because there's a new term called skinny fat. So perhaps, in the outside, we look like quite slim. However, our internal organs like liver, they do have the high fat. So therefore, the BMI is not like really good way to look at whether someone is healthy or not. But forget this. For this purpose, for this project, we just wanted to use the traditional method. So for this one, 41 patients with the BMI lower than 30, and then we have like 28 patients with the higher BMI. And then also they did have the different Gleason scoring. So we looked at 5 different biomarkers, 5 different microRNAs. These microRNAs were pre-selected by doing lots of reading and also doing lots of database searches. So you can see that the high BMI and the high Gleason score, they did have like different microRNA expressions. So we also wanted to check whether the ROC curves are going to show whether these microRNAs are sensitive and specific enough. And this paper was already published, I think, almost like a year ago again. So this was another way that show the microRNAs can be used non-invasively as a biomarker and distinguish between the not only the higher and the lower Gleason when it comes to the prostate cancer, but we can only distinguish by using microRNAs high BMI to the low BMI patients. So I think I have a bit more time, so I like to talk about the pancreatic ductal adenocarcinoma. This is the final project that I'm going to mention today. I'm going to sound horrible, but I told you that my lab is mostly interested in metastasis. So therefore, the most metastatic cancers are the better models for us. And pancreatic ductal adenocarcinoma is one of those. There are still like 4% overall survival rate for 5 to 10 years for pancreatic ductal adenocarcinoma. It is extremely aggressive cancer type. There is no biomarker, proper, better biomarker. Most of the times when the patient's diagnosed with the pancreatic cancer, pancreatic ductal adenocarcinoma, they are already, the cancer already metastasized, and most of them, they don't even go through the treatments because it's already too late. So we first wanted to look at whether the certain microRNAs are involved in pancreatic ductal adenocarcinoma progression. So for this purpose, this is a review that we published a couple of years ago. So you can see that we sort of mapped all the known microRNAs in terms of the cancer hallmarks. So you can see that there are microRNAs that are associated with the avoiding the immune destructions. There are certain microRNAs that involved in or associated with genome instability and mutations. Some of them are associated with the inducing the angiogenesis. What I hope you see in here, there are always the similar ones, the usual candidates and the usual ones like in 155, which we talked about it, 21, which again we talked about it. It just keeps occurring in everywhere. So it seems like these 2 microRNAs, or like a couple of them in 210, which I'm also going to mention in a minute. So these microRNAs are like pretty much coming out in every single hallmarks in here. So we wanted to see first whether we can just shortlist certain microRNAs. So for this purpose, we used this paper. We found that there were like small number of patients, but this small number of patients, they did have both the tumor tissue, pancreatic ductal adenocarcinoma tumor tissue, and also the normal part of their pancreas. So in other words, they are sort of like matched. They both are coming from the same patient. We wanted to see whether there is any signature comes out in terms of the microRNA expressions. So this is completely in silico study. We looked at these patients' RNA sequencing data, and we found out microRNA 21, 221, 375, and the other microRNAs, they sort of like highlighted in terms of the different patient samples. So we mapped out all the 20 tumors. So there were like 10 males and 10 females in this study, and also the downregulated microRNAs. And we created these charts as well. So you can see that there are certain microRNAs. Again, I'm not sure whether you can see it really clearly here, but the same microRNAs that we have been working already for other projects came out. So then we moved back to the lab. So we wanted to see what is happening with the in vitro cell line studies. So the HPDE here is the normal epithelial human cells, and there are like 3 different pancreatic ductal adenocarcinoma cell lines. This time, unlikely of the previous 2 studies, there is no hormone dependency we're talking about. When you look at the literature, all the pancreatic ductal adenocarcinoma cell lines are extremely metastatic. However, the BAX-PC3, we found out they are like more epithelial than the PANC1. In other words, BAX-PC3 is less metastatic than the PANC1. So we looked at 5 different microRNAs for this study. So what you can see, and then we normalize all of these expressions in terms of the normal human duct epithelial cells, HPDE. And you can see that microRNA 21, which we have been talking since the beginning, is beautifully correlated with the metastasis again. 155, again, we've talked. Again, you can see that the PANC1 has the highest expression. 221, it is another important microRNA, another oncomer. You can see that it is, again, the similar thing we are seeing. There's an upregulation in the PANC1 when you compare to the HDPE. But when it comes to the 2 of the tumor suppressor microRNAs, the scene is completely reversed. So you can see that the PANC1 or the MAIA-PACA2, they do have the least expressions of 2 of the tumor suppressor microRNAs when it's compared to the normal duct epithelial cells. Okay, so we first started with the in silico analysis by using the RNA sequencing of the patient samples, matched patient samples. Then we moved on to the cell lines, and we wanted to see what is happening with these cell lines. Using the similar approach that we used before, this time, again, we looked at the 2 different cell lines, and we CRISPRed 2 different cell lines. And then you can see that when there is no microRNA 21 in both of the cell lines, microRNA 21 reduced. More importantly, all the cancer stem cell markers are also reduced. So the cancer stem cells are important because we believe that according to the seed and soil model, we believe that according to the literature, there are some cells that they are already in a dormant phase, and these mostly are the cancer stem cells. All of our brilliant, beautiful approaches like cut, burn, and poison is not working because they don't care the hormone independency. They are not hormone independent anyway. So I think I'm sort of like running a bit out of time, so let me just like speed up a little bit. So you can see that the microRNA 21 knockout diminish expression of all the cancer stem cell markers in pancreatic ductal adenocarcinoma. So like 5 different cancer stem cell markers we looked at in both of the MAIA-PACA and the PANC1 cells, and this was similar in the flow cytometry analysis as well. And then the invasion also reduced when there is no microRNA 21. So as you can see, the microRNA 21 is highly important when it comes to the cancer metastasis, not only for breast cancer, but also another highly metastatic cancer type in pancreatic ductal adenocarcinoma. So of course, we looked at more of the different signaling pathways as well, and then we published this paper. So if you like to read about it, it's already available. So another side project that I wanted to mention to you. So my lab, we are already interested in the in vitro studies and trying to understand the mechanisms of the metastasis. But we know that this is not only about the tumor microenvironment. There are like more important things, like because we all like live in an environment. So more and more we are started to, I guess, like interested in the exposomes. What is the exposome? So the exposome by definition, refers to the totality of the exposures from the variety of the external and internal sources. So we have been already working on the internal resources, but we didn't look particularly the external resources as well, or the sources as well. So this is a word that I quite like. There is a quote in here: all illnesses have some sort of hereditary contribution. It's been said that genetics load the gun and the environment pulls the trigger. So our question was, what is happening with the environment? Or does the environment is affecting the microRNA expressions in our body? So for this reason, I'm not a toxicologist, but I have great collaborators. They are toxicologists. So we looked at whether we can use, the usual suspects, the microRNA 21, 155, both oncomers, and 126, that is a tumor suppressors, as a response to the environmental heavy metals, environmental toxins. So we published 2 studies on this. So we basically treated the cells in extremely low conditions, not toxic at all, cadmium and nickel. Both cadmium and nickel, we do see it, or we do expose a lot in our usual lifetime. Now, when we were writing this paper, for instance, I was quite scared because, for instance, the cadmium, we have a lot in as a health risk in rice. So we do have lots of rice most of the time. Well, there are lots of cadmium in it. All the pulses, all the nuts that we use, especially if you're a vegetarian, to be able to get the protein source, you are taking lots of lentils, you are taking lots of the nuts, lots of cadmium as well. So again, I found it quite interesting to see that when we treat the pancreatic cancer cells with extremely low amount of cadmium and nickel, up to 3 weeks, we did see their expression levels completely went up. So perhaps we can also use the microRNAs as biomarkers for the environmental toxins. Right, the conclusions. Alterations in the microRNAs are involved in pathogenesis in various of the human cancers. In here, I talked about pancreatic ductal adenocarcinoma, prostate, and the breast cancers. I believe they have a great potential in terms of using them as a biomarker because they are quite small. They're only like 18 to 22 nucleotide long single-strand RNA molecules. They are highly stable. And we can use them from the bloodstream. So we can just look at the circulating biomarkers. So they have a huge potential to be used as a non-invasive diagnostic tool. I haven't talked anything about the therapeutic approaches in here, but they also can be used as a therapy as well. Of course, we do need lots of specific studies to be able to understand whether they are specific and sensitive enough to be able to use as a biomarkers. But there is, these are the opportunities. The challenges, I must say, the problem with the microRNAs, I'm sure you've noticed. So microRNA 21, we've seen the upregulation in 3 different cancer types so far. So it is not cancer specific. That is one of the issues that we need to consider. Another one is when we use the circulating microRNAs, we do not know where the primary tumor is. There is no particular specificity of this microRNAs, whether it is coming from the prostate, whether it is coming from the breast cancer. So we cannot associate, or we cannot differentiate this certain microRNA in terms of the certain site, so because we are using this as a non-invasive tool. So we started with a bit of a depressing slide by saying that one of 2 people are going to get cancer in their lifetime. But the good news is there is a high survival rate of the cancer right now, and I'm hoping that like in 5 to 10 years, we are going to have even more survival rate. So we are going to have a lot more success than before. So as I said, I did not have time to talk about therapeutics, but already lots of labs are working microRNAs as a therapeutic targets as well. In this another review, we sort of mapped which microRNAs can be used as a therapeutic. In fact, we do know that there are certain clinical trials going on, and this is one of the examples. Miragen is one of the companies at the US. So they are looking at the microRNA 155 because similar to 21 story. So 155 expression is associated with the poor clinical outcomes. So therefore, they are trying to use this anti-MER 155 drug for the blood cancers and lymphoma. So this is a FDA-approved clinical trial going on. And if you look at the FDA's website or any recent review, you can see that there are lots of clinical trials going on to use microRNAs as a therapeutics as well. And mostly the Miragen is doing this. Right, I think I have only 10 minutes left. These are the wonderful people that I have been collaborating recently, and the data that I showed to you was due to the collaboration between different labs and the different groups. Thank you so much for the invitation, and thank you so much to all of you to listen to me to talk about microRNAs today. Right, I'm going to stop sharing and I'm ready to hear if you have any questions. Thank you.
Thank you so much, Dr. Onganer. We would like to thank you for this informative presentation. I'm directing the first question. In treating the malignant disorder and preventing tumor recurrence, diagnostic and therapeutic techniques targeting these cells and their regulators, in addition to differentiated tumor cells, are required. Could sample size limit the study when working with microRNAs at this stage? Would it be advantageous to take a larger sample size from different populations?
Okay, if I understood correctly, the sample size. So the sample size definitely is so important because the main issue is cancer is like quite heterogeneous. In other words, that everybody's cancer is unique to that person. So we are trying to find almost like the overlapping biomarkers that would fit to everyone. However, when it comes to the, we do, again, I didn't mention this to you in here, but we are like trying to validate all of those biomarkers in different sources. So for instance, one of the studies that we are doing, we do have fresh frozen tissue samples from the patients. You can see that those biomarkers or the microRNA expressions correlate beautifully when it comes to the disease status. From the same patients, if you get the blood, and if you look at the plasma and the serum from the blood, you can see that those same microRNAs are not as specific as the actual tissues. So that means we do need to expand the study. We do need to have more patients if we are talking about the circulating microRNAs, and then to be able to perhaps like validate more whether these microRNAs are specific and sensitive enough.
Thank you so much for your answer. I'm moving on with the question from the YouTube participants. In microRNA research, clinical and methodological heterogeneities are observed that can arise at many points during the study. Can a standard protocol be created to reduce this heterogeneity?
Okay, another great question. So yes, that is one of the challenges that I was trying to mention. So the opportunities are great, but we also need to tackle the challenges. So to be able to tackle that challenge, I believe we need to have a panel of microRNAs, not only microRNA 21 or 155, but we have to have like 3 to 5 microRNAs, and we need to look whether the patients are like expressing the same trend of this panel of microRNAs, rather than like focusing on a magic bullet in a way, like in one single thing.
Thank you so much. Another from the YouTube participants. Do the prognostic association of microRNA-21 differ when analyzed within breast cancer subtypes in TCGA?
Yes, wonderful, wonderful, Seçil. Thank you so much for picking that up. When we look at the TCGA, we looked at the all available breast cancer patients' data. So it was like almost, as I said, 995,000 patient samples we looked at, but we didn't group them in terms of the PG, progesterone positive, estrogen positive, or triple negative. It does differ. So we didn't do that in that paper, but then again, reviewer asked and then we looked at it. So you're absolutely right, Seçil. I didn't show it in here, but we do see there is a higher expression level in the triple negative breast cancer than the hormone dependent patient samples.
Thank you so much. I'm moving on to the next question. In your study, microRNA-21 expression levels were positively correlated with reduced overall survival status. There was no significant result for disease-free survival ratio. Have you any opinion on what means this result?
Okay, good question. No, I don't have any opinion because I am more interested in the overall survival rather than the disease-free. Remember, all of these studies are mostly focusing on the metastasis. So we are mostly focusing on already extremely aggressive cancer types. So I guess to be able to answer your question, we need to go back, perhaps the early diagnosed patients, and then look at what they are doing in the disease-free, like after their treatment. But particularly we, as a group, we haven't done that.
Thank you so much. I'm moving on to the question from the YouTube participants. When we think late diagnosis and resistance to chemotherapy in OC, how can we occur clear communication between tumor microenvironments, chemical compounds, and carrying cargo stability?
Very loaded question, Sevginas. Thank you so much. I guess we do need to look at the different parts of this, right? So with the tumor microenvironments, which we are currently doing, so we need to create the organoids to be able to tackle that problem. Because so far, almost all of the studies that I showed you, they were done either with the patient samples and or the 2D tissue or the cell culture studies. So we are already doing 3D cell culture, but to be able to answer your question, we do need organoids, not only the cells, pancreatic cancers or breast cancer cells, but we also need to have the macrophages and the fibroblasts. So we do need to create a similar model to be able to answer this question. And then the microenvironment chemical, I guess once we have the organoids, we can also treat them with the cadmium, nickel, or lead, or whatever environmental toxins that we are talking to. We can treat these organoids with them and then try to understand the mechanism better.
Thank you so much. I'm moving on to the next question. Is efficient modulation of downstream targets in the therapeutic scenario of microRNA-21 dependent on different regulators such as cell migration-related genes?
Okay, another good question. I tried to show you like 3 different studies, but I didn't show you the full papers. So if you are more interested, I definitely suggest you to go and read the paper. But with the migration, cell migration, we did look at the EMT markers in the epithelial mesenchymal transition and the cell migration. We didn't look at the gene expression. We just did the scratch assay for the breast cancer study. For the prostate and the pancreatic cancer studies, not only the scratch assay, but we also did the invasion assay. So these two are quite different than each other because the scratch assay shows you whether the cells are migrating, moving or not. Another one, the invasion assay. You know that with the invasion, the cells, they do need to secrete the enzymes, the lytic enzymes, to be able to invade the matrigel. So we've also done that, but we didn't look at any particular MMPs, for instance, matrix metalloproteases.
Thank you so much. I'm moving on to the next question. Is it known which genetic changes produce traits in cells that can metastasize? If known, what are they?
With genetic changes. So it is quite difficult to point out only one or two genetic changes. As I try to summarize, there are like so many things going on. So there is like the cell signaling pathways are activated, and most of the cancers, there's a genome instability. But more importantly, there are like new fusion genes occur as well. And I told you in the prostate cancer study, although we didn't look at specifically at the TAMPAS-ERG, but in one of my previous studies, we were looking at the TAMPAS-ERG fusion, which is an event almost like 60% of the prostate cancer patients. That very much involves in the gene expressions that those genes are identified to regulate metastasis. So it is a great question, Mehmet Emin. However, it is quite difficult to point out one or two genetic changes. So there are so many of them.
Thank you so much. I'm moving on to the next question. In your study, could you identify the possible mechanisms of GATA2 regulation by microRNA 21 in breast cancer?
Great question, Elif. Thank you. GATA2 is a transcription factor, and it is quite important. We are looking at the GATA2 as well, especially it is important for the cancer stem cells. So yes, we did find, but it is, egg or chicken problem again, right? So which one comes as a first event, whether the microRNA higher expressions are triggering all of those like abnormal cell signaling pathways, or these signaling pathways are already started to get mad, and therefore the microRNA 21 sort of pushes them to even faster to go to the dark side. That is a bit difficult to assess right now, but you're absolutely right. There is an association and regulation between the transcription factors and the microRNA expression in breast cancer.
Thank you so much. I'm moving on to the next question. Is early diagnosis possible with microRNA quantification, which will be widely adopted in primary healthcare? What are its limitations?
Wonderful question. That is my aim. So we are trying to use the microRNAs, or we are trying to propose the microRNAs, not on only one, as I said earlier on, as almost like a panel to be able to use in the clinic, perhaps like as a confirmation studies or the confirmation tests, because there are like lots of great things about the microRNAs, although there are challenges. The limitations, the major limitation in my opinion, is how to collect the samples. So if we are going to use the blood samples, obviously you need to collect the samples. You do need to use it in a certain tubes, because the hemolysis is a problem. You need to spin it down. You need to, if you are going to keep it in the plasma or serum, you need to keep it in the -80s or -20. So in my opinion, the biggest limitation or the challenge is the collection of the samples and also the storage of the samples. If we are going to use the tissue samples, this time again, I think I showed some examples of the immunocytochemistry in situ hybridization. This time, as soon as during the surgery, the tumors or the tumor samples tissue samples are collected, they do need to be in the formalin. So you cannot keep it in the room temperature, because this time the RNA quality is going to suffer. So the limitations are the storage.
Thank you so much. Could the EMT pathway being downregulated by inhibition of microRNA-21 lead us to say that microRNA-21 is a possible target to inhibit cell invasion?
Good point, Hilal. I think yes. The short answer is yes. In 3 different studies and 3 different cancer types, solid tumor cancer types, we did show that when there is no microRNA-21, invasion reduces. But remember, all of these studies are like in vitro. We haven't done any mouse models in terms of like checking whether the microRNA removing the microRNAs are going to reduce the metastasis. So in 2D cell models, the answer is yes.
Thank you so much. I'm moving on to the next question. Do microRNAs play functional roles in complement-mediated tumor immune escape? How can microRNAs be exploited to increase complement-mediated cell destruction? Thank you.
Thank you, Hilal. It's quite a good question. So I don't think the microRNA's functional role is well known enough in terms of the immune response. So there are like other targets for the immune response. So in the map that we sort of like put microRNAs in the cancer hallmark, we did identify or named a couple of microRNAs depending on the available studies. But first of all, we as a group never looked at the immune escape. And 2, there are like more imminent factors for the immune escape rather than the microRNAs.
Thank you so much. I'm moving on to the next question. Many microRNAs have already been reported to be promising biomarkers for certain types of cancer, but they have a specificity issue. Is there any solution in this problem?
Yes, Elena, you're perfectly correct. There is a specificity issue, but the answer is looking at more than one microRNAs and perhaps looking at the association between 2 microRNAs, as we do it with the EMT pathway, for instance. So just come up with like 3 or 5 microRNAs as a signature of that particular tumor and see whether those 5 microRNAs are already upregulated or downregulated.
Thank you so much. Can microRNAs be used as a therapeutic target to increase the efficacy of radiotherapy and reduce radiation-induced malignancy?
Wow, another great question. So you guys are brilliant. Thank you, Sedanur. It is a very good question. So the first part is yes. Can microRNAs can be used as a therapeutic target? Yes. So there are lots of companies are working on it. The radiotherapy, I'm not sure about that. I don't know. I've never seen any study that they can use the radiotherapy or reduce the radiation-induced malignancy with the certain treatment of the microRNAs. I'm not aware of any study, but hopefully in the future we'll see more.
Thank you. There is considerable research into the use of panels containing multiple microRNAs. However, how much do you think microRNA interactions are taken into account here?
Yes, Saida, you're also quite right. So there are lots of research going on. So some of the research now are not only looking at microRNA interactions, but like as the lots of you already mentioned, to increase the specificity and the sensitivity, we are also trying to identify link RNAs, long non-coding RNAs, and look at the link and microRNA associations, not only microRNA, but also the another layer, link RNA and microRNA association. And yes, we are looking at those mer-to-mer interactions as well.
I would like to thank you for your informative answers. Lastly, I would like to ask that what advice would you give to students studying in scientific fields?
Read more, ask lots of questions, keep asking questions and read more, and definitely be curious.
Thank you so much.
Thank you so much. Great to have all of you, and thank you so much for all those brilliant questions, and enjoy the rest of the day. Thank you.
Thank you so much. I wish you healthy days.
Thank you.
At 3:15, Assistant Professor Dr. Akdeserin Harmancı will be with us to give a speech on machine learning for single-cell data analysis. Thank you so much. Bioinfocongress IV. Assistant Professor Dr. Akdeserin Harmancı, Machine Learning for Single-Cell Data Analysis. Dr. Akdeserin Harmancı is an Assistant Professor at Baylor College of Medicine, Department of Neurosurgery. She was an Associate Research Scientist and a postdoctoral researcher at Yale University School of Medicine, Department of Neurosurgery, before working at Baylor College. During her postdoctoral studies, she and her colleagues discovered genetic epigenetic causes of brain tumors and identified new molecular targets for precision. She has studied the epigenetic and genetic mechanisms leading to the atypical and malignant transformation of brain tumors. Dr. Serin Harmancı completed her undergraduate studies at Sabancı University in Computer Science and Engineering. Then, she finished her PhD in computational biology at the Max Planck Institute of Molecular Genetics in Berlin. The primary focus of the Serin Harmancı Lab has been developing integrative approach to dissect the molecular landscape of brain tumors using single-cell RNA sequencing and spatial transcriptomics data. Currently, they are working on developing new methods for understanding tumor heterogeneity and the immune microenvironment in brain tumors using single-cell RNA sequencing for developing targeted therapies.
Dear Dr. Akteserin Harmancı and participants, welcome all of you to our Bioinfocongress IV event. Dr. Serin Harmancı will give us a talk today on machine learning for single-cell data analysis. At the end of the presentation, we will have a question-answer section prepared by our team and consisting of questions from our participants. Dear Dr. Serin Harmancı, welcome back. It's so great to have you here.
Thanks a lot, Efekan. Thank you for having me here. And thank you for the great organization. I know you have been working on this for some time, and it's a great organization. Thank you. So I think I can start with my presentation now. Sorry, yeah. Today I will be talking about machine learning for single-cell data analysis. So I will try to give an overview of single-cell technology, and hopefully I will go through some computational challenges in single-cell transcriptomics. I will also talk about past and current research on brain tumors that we have been working on for some time. And I will briefly go over trend topics in computational biology. Okay, so let's start with the overview of single-cell technology. You might be aware of this single-cell technology. It's been around since 2013. It was the method of the year in 2013. Since 2013, these technologies become routine when investigating cell-to-cell heterogeneity and transformed many areas of biology and medicine. Here is an overview of the scaling of single-cell RNA-seq experiments. In 2010, like the single-cell technologies were able to sequence 1 or 10 cells, but thanks to microfluidics, we now can sequence around million of cells, and we have a lower sequencing cost, and all these things, microfluidics, low sequencing cost, it empowered single-cell sequencing technology. And now, as I said, like we can sequence millions of cells in an experiment, and this is a huge data, as you can imagine. So it's a data science problem that we have. Yeah, so this is like a figure that shows the difference between the bulk sequencing and the single-cell sequencing. In bulk sequencing, we have a sample that contains like thousands or millions of cells, and DNA is extracted from all of these nuclei. And then we have the reads coming from all of these cells in the bulk sequencing, and we map all of these reads to the genome. However, in the single-cell sequencing, we try to extract DNA from one single cell, and we amplify the DNA, and then that amplified DNA coming from one single cell is sequenced and mapped to the genome. Here we have, as I said, like a data science problem. Here we have millions of cells coming from one tissue. But on the other hand, the data is noisier compared to the bulk data. It's much more sparse. So we have to treat the data in a way that we can remove all of these noises, and also we have to think about the sparsity of the data. This is a well-known analogy that shows the single-cell transcriptomics. It has been adapted from the Human Cell Atlas. So this really explains it well. In the bulk RNA-seq, it's like putting all the fruits in a blender, and we taste the average of all of the ingredients. In single-cell RNA-seq, we can taste all of these individual fruits. We can understand the detailed composition of the fruits within the fruit salad. So bulk RNA-seq is like a fruit smoothie, and the single-cell RNA-seq is the fruit salad. So single-cell data is much larger, and it's much more sparse. We have lots of zeros in the data, so we have to think about how to get rid of the zero values in the data. There are broad applications of single-cell sequencing in biological and biomedical research. For example, in neurobiology, we can use, it has been used, single-cell sequencing technology has been used broadly. There are lots of consortiums like Human Brain Atlas, Allen Brain Atlas, Human Brain Somaticism Network, and all the other consortiums. They sequence like millions of cells, and they try to understand like different neuron cell types. They would like to see the somaticism in the brain cells, and there are also other applications in immunology. There are like tons of different immune cell types, and it's not just like discrete cell types. We have states, so it's like a spectrum of cell types, and with these technologies we can build trajectories to understand all these different cell types and states. Here we will focus on cancer biology. My research is mostly focused on the cancer biology. And what are the main applications of single-cell sequencing in cancer research? We can resolve intratumor heterogeneity. As you know, in cancer, we have intratumor heterogeneity. We can investigate the clonal evolution in primary tumors. We can understand the invasion and metastasis. We can also profile, we can sequence and understand the circulating tumor cells. We can look at the mutation rates and the main driver mutations happening in the tumor cells. Where we can understand the resistant evolution to therapy. We can understand the cancer stem cells and cell hierarchies. And also we can study the EMTs, epithelial to mesenchymal transitions, by building trajectories. I said we can understand intratumor heterogeneity, metastasis, plasticity, and so on. But also we can have a better idea about the tumor microenvironment. We can characterize different cell types, different cell type compositions within the tumor microenvironment, and that is very critical in guiding the treatment of tumors. For example, the ratios of CD4 or CD8 T cells have been shown to predict overall survival and different immune responses. With the use of single-cell technologies, we can understand the different immune cell type compositions within the sample. And this is like a figure that shows what kind of information we can get from the single-cell data. Right now we can zoom in to one side of tumor that is happening in the body, and we can see that within that tissue we have a lot of different cell types. We have the tumor cells, we have the T cells, B cells, dendritic cells, fibroblasts, endothelial cells, T cells, and so on. When we do bulk RNA sequencing, here we see like 3 gene expressions in this plot, in this figure. When we do bulk RNA sequencing, we just take the average of that gene, and we don't look at each of these cell types, but we just take the average of all these cell types. So it just gives an overview. However, when we do the single-cell sequencing, we can understand what is that gene's expression within the neoplastic cells, within the T cells, B cells, and all these other vascular cells. And that can be used in personalized medicine, because we can understand like what is that single normal cell that transforms into a tumor cell. We can get a better idea about the clonal evolution. We can see how that single cells from the primary tumor form metastasis. And we can also understand the single tumor cells that survived the therapy, and we can potentially like develop targets like drugs for these surviving tumor clones. So understanding the clonal architecture of the tumor is very essential in clinical care, especially for determining treatment-resistant tumor subclones. Yeah, this is another figure that shows the same idea. In the bulk analysis, we get an idea about the average clone, and we develop a drug for the average clone, but we have other clones in the tumor. And if you just look at the average clone, we cannot really remove the tumor cells. If we have the single cell analysis, we can get an idea about all these individual clones, and we can have precision drugs targeting each of these individual clones. We said that like with the single cell technology, we have millions of cells. So it's a data science problem, and we need computational methods and techniques to remove the noise in the data, to interpret the data, and to get some meaning out of this data. This is like a basic overview of the single-cell RNA sequencing pre-processing pipeline. We have the patient tumor. It's dissected from the tissue. We have, as I said, like the malignant cells and all these non-tumor cells that is in the tumor microenvironment. And each cell within this tissue is sequenced. We have, after sequencing, we have a lot of reads. Like it's just like ACGT letters coming out from this sequencing machine. And what we do is like we map all these reads to the reference genome, to the human genome, and then we generate single cell count matrices. And these matrices have like genes in the rows and cells in the columns. We have, you can imagine that we have like 20,000 genes in the rows and 1 million of cells in the columns, let's say. And the other issue is that, as I said, like this matrix is full of zeros, so it's zero inflated, and our tools or methods have to take this into account. Because of the dropout, we might not be able to detect the expression of some of the genes. Some people do imputation before doing further downstream analysis. There are also some maybe dead cells that we have to remove. There might be some doublets. There are tons of different tools that are available to do all these analyses. I listed some of those here, like Seurat analysis pipeline is like one of the most popular ones to do all these pre-processing pipelines. There's Scampi, and if you're doing 10x single cell sequencing, 10x company has its own mapping tool, Cell Ranger. But we still lack some algorithms and methods that can properly analyze all these data sets. One of the other main issues is that batch correction, data integration, and dimensionality reduction. We might have data coming from different patients, and we would like to integrate all of this data, but there might be some patient-specific biases that we have to remove. So first of all, we would like to do a dimensionality reduction because we have, as I said, like 20,000 genes. And we cannot really visualize 20,000 genes and 1 million cells. One of the most popular ways of visualizing this data is doing dimensionality reduction. In bulk RNA-seq, we usually do PCA. It's a linear transformation, but in single cell data, PCA doesn't work well. Because of the sparsity of the data, we need a nonlinear transformation. So people started using TCNE or UMAP. Those are like nonlinear transformation techniques to visualize the data. So we had this 20,000 genes and millions of cells, let's say, and then we can reduce 20,000 gene dimension to 2 dimensions. We have like this is like a plot of UMAP dimensionality reduction plot. We can visualize the data with the UMAP nonlinear transformation tool. Here, each dot represents each single cell. And after we reduce the dimension, we cluster the data. And then hopefully each of those clusters are like homogeneous cell types that represents each cell type. Basically, as a summary, dimensionality reduction is needed for visualization, for clustering, for simplifying the complexity of the data, reducing the number of features in the data. And the other issue is the batch effect correction and the data integration. Let's say we have a study where we have drug treatments, like with drug treatment and control, 2 different data sets, and we would like to combine these data sets that is coming out from the single cell sequencing, the mapping platforms, the mapping tools. We would like to combine all these outputs of these gene cell count matrices. However, when we just combine it like in a very simple way, we would see that there's a study bias in the data, where we see that each of these like treatments are clustering within themselves mostly. So we need to have data integration methods that can align those data sets. So there's like different methods that can align those 2 data sets. One of them is the canonical correlation analysis that people use. So this is basically like aligning those 2 data sets in like another dimension. And after aligning the data sets, then we can say what cell types have different proportions in under which treatment. We can also get like cell type specific transcriptional responses to drug treatments after data integration. All of these steps are very crucial, as I said, dimensionality reduction, data integration, and I didn't mention it that much, but like the initial normalization is also quite important. Yeah, so let's move on. And now we will we have the clustering of the data. We reduce the dimension. We have the clusters, but we would like to annotate each of these clusters. Annotation of each of these clusters is a difficult task. People have been using different methods, but the main idea under all these methods is that like within each cluster, we expect to see 1 cell type. And we look at the markers of these cell types, and we annotate each cluster based on the marker genes. But the issue is that it's not always the case. It's sometimes we see heterogeneous profiles within each cluster. And the other issue is identifying tumor cells within this clustering. Again, tumor cells are not always clustered together. They might be heterogeneous. So what we do is we try to utilize the single-cell RNA-seq data and try to infer genomic profiles from the single-cell RNA-seq data. And as you know, like we see copy number variations in tumors, and if we can infer large-scale copy number variations from this data, then we can have a better idea which cells are tumor. And you know, we have an accumulation of somatic mutations within the tumor cells. So again, inferring mutations from single-cell RNA-seq data might facilitate to identify all these tumor cells. And again, as I mentioned before, so we have two components in cancer single-cell RNA-seq data. We have the tumor cell identification step, and we have non-tumor cell identification step. And people have been using different ways. We can either look at the marker genes and try to annotate each of these clusters based on some known marker genes. We can have correlation-based methods where we have a reference data and we have the labels of each of these cell types in the reference data, and we correlate our data with this reference data, or we train the data sets and we train known, well-labeled, well-annotated single-cell data sets, and then predict the cell type within our own data. So I will talk about our past and current research on brain tumors that utilizes single-cell RNA-seq. But first, I will go over my postdoc studies where we mainly used bulk RNA-seq or whole exome sequencing technologies. So I will briefly go over my past research on brain tumors. I've been working with the brain tumors for some time now, and we have been focusing on gliomas and meningiomas. Glioblastoma is a lethal disease. Average survival for patients with GBM is 15 months. And unfortunately, survival rates have not changed in over 30 years. And the brain is a therapeutically challenging organ system. We might not be able to take biopsies from the tumor, and operations are not very easy. During my postdoc studies, we were working with the whole exome sequencing data, and we were trying to identify the driver mutations in meningioma. NF2 mutation was known, but we were looking for some other driver mutations in meningioma. And for that, we did whole exome sequencing in around 300 patients. And here in this plot, each bar represents each patient sample, and we have identified driver mutations in TRAP7, AKT1, PIK3CA, KLF4, HACHOK pathway genes, and POLR2A. And we also saw that all of these mutations are mutually exclusive. And interestingly, this HACHOK pathway genes are targetable by drugs that they have been shown to work. HACHOK pathway targetable drugs have been shown to work in different cancer types. And we have also shown that all of these mutations have correlations with anatomical positions. NF2 mutations are mostly in the tumors that are in the cerebral, and all of these non-NF2 mutations were in the skull-based meningioma. We also looked at genetic and epigenetic causes of progression in meningioma and glioma. We had matched glioma samples that were primary initially and then recurred and progressed later in the later time point. We did integrated epigenetic methylation, RNA-seq mutation analysis, and we have identified some oncogenic pathways that might be playing role in the progression of glioma. And we have done the similar analysis for meningiomas and tried to identify the pathways that might be relevant for the progression of meningioma. So those were like some past studies, my previous studies, and those were, as I said, like mainly based on bulk RNA-seq, whole exome sequencing, and mutation analysis. And we have done, as I said, like integrated analysis to identify the molecular changes within meningioma and glioma, with the hope of finding targets that we can use for developing drugs. My current research interest is understanding the interactions between tumor cells, neurons, and immune microenvironment in glioma, and that we can do perfectly using single-cell RNA-seq data. And this interaction between the tumor cells, immune microenvironment, and neuron cells is very, understanding this interaction is very important, because that might answer what drives the progression of glioma. So we have in-house glioma single-cell RNA-seq data, and currently it's around 500K cells. It's like 500,000 cells that we have. Those cells have been dissected from around 10 different patients. They are like fresh taken out from the operating room and sent to single-cell sequencing right away within a couple of hours. So we are trying to understand this 500,000 cells. It is coming from different patients. We have this data integration issue. We don't want to have any effects of or any biases of different patients that are not biological effects. We have to remove all these non-biological effects from the data. We have to analyze all of these data sets and understand the intratumor heterogeneity. We have to understand the tumor microenvironment. And instead of annotating each cluster, we would like to annotate each single cell, because especially in brain, we have a high level of heterogeneity. So we need a single cell based annotation technique. So let's go over each of these points. And first, let's focus on differentiating tumor cells from non-tumor cells. So as I said before, we don't have simultaneous single-cell DNA and RNA sequencing. That's why we need to utilize the single-cell RNA-seq data and integrate the genomic landscape of the tumor cells with the transcriptomic landscape. So in order to infer all of these genomic changes, we use our CMB inference algorithm, Casper, and SNP mutation inference algorithm, Excavator. So, you know, like copy number variations happen in cancer cells, and we have large-scale copy number variations happening in cancer cells. We have large-scale chromosome 22 deletions in meningiomas. We have chromosome 7 and chromosome 10 CNVs, large-scale CNVs happening in IDH wild-type gliomas. And with the read coverage, we can have an idea about the CNVs that are happening in the single-cell RNA-seq data. Very local CNV events might not be very easy to detect using single-cell RNA sequencing, but we can have a good idea about the large-scale copy number variation events. So we can also use the variant allele frequencies to better characterize copy number variations, because if we have a deletion, then the variant allele frequency will go up to 100%. If we have amplification of one copy, the variant allele frequency will go around will be in the line of 0.3. But if we have amplification of 2 copies, then we won't be seeing any shift in the variant allele frequencies. So one of the main challenges here is that we have a heterogeneity, and in our tissues we have a mixture of cells that have CMVs and without CMVs, because in our samples, as I said, we have non-tumor cells too. And we need baseline control samples to correctly identify all these large-scale events. And we can use all these publicly available normal brain atlases like GTEx, Human Cell Atlas, or HubMap. Or we can detect the non-tumor cells within our single cell data and use those cells as a control for our CMV analysis. This is an overview of our CMV inference tool, Casper. So basically, we have the reads coming out from the single cell data. We map those reads to the genome. We generate the normalized gene expression matrix. And then we smooth this expression matrix by applying recursive iterative median filtering. And we smooth the signal. And after smoothing, we apply hidden Markov model to segment the data. It's a well-known technique to segment the signal. And we assign CMV states to each of these regions. And we use variant allele frequency shifts that I mentioned before to correct for our CMV calls. And this is an application of our algorithm in one of the public GBM single cell data sets. Initially, so this matrix that you are seeing on the top is single cell row gene expression matrix. In the columns, we have genes ordered by their chromosomal location. And in the rows, we see each of these single cells, and the color labels that you see on the rows are represents each patient sample. So we have 5 different patient samples here, and each of these patient samples have like hundreds of cells. So initially, that the row gene expression matrix doesn't give any information about the large-scale CMVs. But after we do smoothing and recursive iterative median filtering, we can see that some chromosomes are amplified within some patients. So as I said, like in the columns, we have genes ordered by their chromosomal location. You can see each of these chromosome labels here on the bottom, 1, 2, 3, 4, 5, and like up to 22, chromosome 22. And we see like a high expression signal in patient MGH30, for example. And we have the deletion of chromosome 10 in all of these samples. We see that like MGH29 has deletion of chromosome 15. So basically, using these techniques, we can have an idea about large-scale CMVs within each tumor samples. And hopefully we can also see some intra-tumor heterogeneities within one patient sample. And so from the smoothed signal, we binarize this data to like either amplified or deleted. And we see like from the signal, we can have another matrix where we have the large scale events marked. If it's deleted, it's blue. If it's amplified, it's red. And we can also visualize the mutually exclusive events within one sample using our tool. So we have an edge in between the CMV events if that occurs like mutually, if it's co-occurrent, and we have a dashed line if it's mutually exclusive. And this is the application of our tool on our in-house GBM single cell data. In GBM, we have chromosome 7 amplifications and chromosome 10 deletions. It's widely known. And by applying our data, we can see that we can detect the tumor cells using CME calling, using the output of the CME calling algorithm. We can see where the tumor cells in our clusters. It is nice to see that they are not really homogeneous. There might be different tumor cells, and they might not cluster together. And we can identify which cells are tumor using the CME calling. But sometimes CME calling might not be enough because, especially in IDH mutant tumors, we don't have CMEs. So we need some other features that might support the evidence of tumor cells. So as I said, in IDH mutant gliomas, we sometimes don't have any large-scale CMVs, so we will utilize the mutation calls that we can have from the single cell data. So we applied our algorithm excavator for characterizing the variant impact from the single cell RNA-seq data. Here, the red points are the cells with the mutation, and the blue points are cells without the mutation, and the gray points are the cells without any coverage. So in single cell data, we have a lot of cells, but they usually don't have coverage for all the genes. We have 1 or 2 reads per gene per cell. So here we can see that these clusters that we show here, the red ones, they are tumor cells because they have an accumulation of IDH mutation. And that is an evidence for us for labeling those clusters as tumor. And we have been talking about, until now, we have been talking about the tumor cells, identifying the tumor cells and understanding the intratumor heterogeneity. But we also need to understand the tumor microenvironment, especially in gliomas. Brain infiltration is very high. Brain tumor cell infiltration is very high, and tumor microenvironment changes the tumor cell behaviors, and also vice versa. Tumor cell infiltration changes the tumor microenvironment, and we would need to understand the changes that are happening in the tumor cells. And for understanding the tumor microenvironment, what we did was we have like public data sets, healthy brain data sets that are available like from all these consortium data sets, human Allen brain atlases, developing brain atlases, and like human cell atlas, immune cell atlas. We have also publicly available glioma immune cell data sets. So what we did was we trained all of these like data sets with a neural network. We have around 1 million of healthy brain or GBM brain data sets that are well annotated. Like we know which cell is which cell type from these data sets, from these publicly available data sets. So we can use all of these publicly available data sets as a training data set. And what we did was, as I said, like we used all these publicly available data sets as a training data set. And we then predicted the non-tumor cells within our in-house single cell data using all these trained data sets. So after putting together all these like methods and steps, we ended up with a pipeline that we call Scrum. And in Scrum, for non-tumor cell annotation, we use the training models of the healthy brain data sets or publicly available well-annotated GBM data sets. And we also make use of our CMV calling algorithm and SNP calling algorithms. And we combine all these annotations in single cell resolution, and we try to see what kind of co-occurrences we see in single cell resolution. So we might see some tumor cells that show like neuron properties or immune properties. So with our SCRAM pipeline, we were able to identify neuron-like tumor cells in IDH mutant glioma samples. So within the complex tumor microenvironment, we have detected that there are tumor cells that can adopt partial properties of non-tumor cells to favor tumor cell survival, proliferation, invasion, and metastasis. And like these glioma stem cells that are in the tumor cells that might lead to transdifferentiation into functional non-tumor cells such as neurons. So using our pipeline, as I said, like we have detected neuron-like tumor cells, so these are some of the figures. So our paper is in bioRxiv right now. We are still making some changes into our pipeline, but basically what we show here is that like in the panel of A, B, C, and D, we show that there is a cluster in our single cell data that have copy number variations and IDH mutations, and also express markers of GABAergic neurons and also glioma stem cells. So in order to experimentally validate all these findings, we did PAT-seq. PAT-seq is a very recently developed technology. We can do whole cell recordings and whole cell patch clamp recordings, followed by single cell RNA sequencing. We can get the electrophysiological properties of each single cell using this technique. From the PAT-seq, we have seen that some of the tumor cells have electrophysiological properties of neuron cells. And these findings were specific for the IDH mutant gliomas, and we have not detected this finding in the wild type mutant, in the IDH wild type gliomas. And we have also found some immune-like tumor cells, and those might be also relevant for the immunotherapies. Yeah, so if you want to look into the details of our findings, maybe it will be better to go into bioRxiv paper. Next, I would like to just briefly go over the future trend topics in computational biology. So, you know, deep learning is everywhere. We have like ChatGPT and all these like complex deep learning models that we have been talking about recently. And deep learning is also in bioinformatics, in machine learning field. There are tons of different applications of deep learning models in single cell data, especially because, as I said, like single cell data has like millions of cells, and that is a perfect data for deep learning. We have a lot of data points that we try to understand and learn. So deep learning methods have been used widely for single cell RNA-seq data analysis for different purposes, for removing the noise in the data, for imputation of these 0 points, or for cell annotation. It has been widely used. Or transfer learning, it has been used for this concept called transfer learning has been used to map different type of data sets and learn from one, maybe like the from mouse model, we can learn the human model and so on. And another application of deep learning was about predicting the effect of mutation. We have like billions of A's and C's and G's and T's in our genome, and we would like to understand if a mutation has a deleterious effect or not. And people have been doing deep learning to understand if a change in the mutation, if a change in the genome would lead to a deleterious mutation or not. Yeah, and also I was talking mostly about the single cell RNA sequencing. So I said like single cell RNA sequencing is currently in everywhere in all the studies, the number of publications related to single cell sequencing has been going up exponentially. But recently, within the last couple of 2 years, now we have also specially resolved transcriptomics technology. So in this specially resolved transcriptomics technology, we can have an idea of the gene activity, the expression of the gene, close to single cell resolution, and also we know where that gene is located within the tissue. So if you go back to this fruit smoothie analogy, we have the fruit salad. We said bulk RNA sequencing is like a fruit smoothie. We just get an average taste of the ingredients. In single cell RNA-seq, we said we can taste each of these individual pieces of the fruit. And in special technologies, we can rebuild this fruit salad. We know the physical location of each of these fruits in our fruit salad bowl. We know where is like bananas are located in the bowl, where the strawberries are in the bowl, and where the blueberries are in the bowl, and we can reconstruct this this physical location using all these special technologies. And if you are interested in single cell genomics, I wanted to include this. This is like a single cell genomics day. I think that's a very nice workshop that is free. And if you want, you can just register for this workshop. And it's on Friday, April 7th. I would like to acknowledge all of those, all of our collaborators, like all these computational methods we developed together with Arif Harmansi and also Su Wang, Xia Chan, and Xiaobo Zhao. And for all these experimental validations in the mouse models and providing all these experimental support, I would like to thank Rachel, Benjamin Denin Lab, Akash Patel, Timo Klish, and and Malcolm McDonald, and I would like to thank Dr. Rao and Dr. Patel for providing human patient samples that we did single cell sequencing, and all the previous work that has been done at Yale about meningiomas. I would like to thank Dr. Murat Günal Lab. And I think that's it. And if you have a computational background, you can email me for any possible postdoc or intern positions. Yeah, hopefully it was useful for you all. And I will be happy to get any questions. I see that there are some questions coming up. Yeah.
Thank you very much for this precious presentation, Dr. Serin Harmancı.
Thanks a lot, Efekan. Thank you for inviting me again, and thank you for the great organization. I think you did a great job, like putting everyone together in a very nice way.
You're welcome. Then I'm starting with the first question. What solution can machine learning offer for dealing with errors and missing data in the identification of variation from single-cell DNA sequencing data?
So if we are only talking about the DNA sequencing, not RNA sequencing, so right now, most of the data that I was talking about was about RNA sequencing. DNA sequencing has been developed also for like for 10 years or 15 years now. But it's not as wide as in single-cell RNA sequencing because the amplification is not that easy. So we have a lot of errors. So that's a great question. Like we have a lot of errors and missing data in the single-cell DNA sequencing data and also in the single-cell RNA-seq data. So what we do specifically for the DNA sequencing data or calling mutations or identification variations, we pool all the cells coming from one patient together so that we have a higher power. It's like I don't know if you are familiar, but like there's this famous GATK pipeline from the Broad Institute that people use for variant calling in whole exome data. So it is kind of a similar idea that we do. We have to pool all the cells together. In this GATK pipeline, they also pool all the samples together to increase the power. We pool all the cells together in the DNA sequencing data, and we call variants from all these pooled data, and then we go back to each cell and genotype each cell based on the mutation calls of the pooled data. I don't know if it makes sense, but to you. But so the other thing is, of course, we might have, if we are focusing on cancer and if we are studying cancer, we will have germline variants, and we are not interested. I mean, some people might be interested in, but mostly we are interested in the somatic mutations, the mutations that are happening specifically in the tumor cells. So what we do is, like, after doing the variant calling, we look at the databases like COSMIC. It's like well annotated tumor mutation databases. So we look if our mutations are also happening in the tumor databases, if they are also there in the tumor databases. And if we are looking for an exact mutation, like an exact change in the DNA at an exact protein location, then that is much more easier. We can be more confident. Yeah. So hopefully that answers.
Thank you very much for your answer. Can you hear me?
Yes.
Thank you very much for your answer.
Thank you.
Our next question is, how can we select cell population for tumor and immune cell crosstalk before and during malignant progression in single-cell transcriptomic?
So that is a great question. So this question is mostly about the cell-cell communication, I think. So first we need to annotate each of the cells in the single-cell data. We have to have this annotation of the tumor cells, immune cells, and so on. And then the next question that we can ask is, what is the cell type communication that is happening here? And I didn't really go into that question in this talk, but people have been looking at the receptor ligand expressions. Like there has been databases that show like pairs of receptors and ligands, and after we do the annotations, we can go over all these receptor ligand pairs and expression of these receptors and ligands within different cell types and try to infer the crosstalk between those different cell type populations.
Thank you for your answer. Our next question is, can we use ML in single-cell omics data for general tasks such as cell type recognition, methylation, and histone modification?
There has been like tons of different methods for cell type annotation that use machine learning, especially Fabian Theis group from Germany. They have been developing different deep learning methods for single cell data sets. And there has been some methods that combine histone modification data sets to predict expression, or like different combinations of omics data to predict the opening of the chromosome, and some other features that we can predict using these different machine learning methods. And I also would like to say that there is a conference coming up in Istanbul, and you might already know, but I just wanted to remind. It's called Recomp, and it's a computational biology conference. It's a very, very popular and well-known, well-recognized conference, and this year it's happening in Istanbul. And there is gonna be like great talks from all over the world. It's gonna be an in-person meeting. So if you want, you can attend. Yeah. And
Thanks
there
for
will
the
be a lot
information.
of, there will be like all these like PIs, like professors working on single cell mixed data and machine learning methods over there.
Thanks for the information and your answer. Our next question is, how can we use machine learning to quantitatively describe the tumor evolution process and evaluate different possible modes against each other?
So there has been some methods to create this phylogeny tree. They call it the phylogeny tree, the mutation phylogeny tree from the data, from the mutation data. There has been different theories how the mutations evolve, but one of the main theories is like a tree kind of like evolution. So there has been some methods that try to reconstruct this tree from the exome data or from the single cell DNA sequencing data.
Thank you for your answer. Our next question is, sparsity in sRNA-seq data can hinder downstream analysis and is still challenging to model or handle appropriately. So how can be improved?
Yes, that's a great question. Sparsity is a very it's a big challenge in this data analysis. So some people do imputation, so there are lots of 0 points in the data, and it's 0 not because it's not expressed, but it's because it cannot be detected. It's like a technological artifact that we have. So we can do imputation. There are like different tools that does imputation. We can predict the expression of that gene at that cell by taking information from all the other cells and genes and impute. And most of the models, the underlying models in all these models, like in these tools, is that it's a 0 inflated data. So the underlying statistics is based on this 0 inflated distribution of the data. Yeah.
Thank you for your answer. Our next question is coming from YouTube. What are the challenges to integrated analysis of multimodal single-cell omics data using machine learning methods?
Yeah, thanks a lot for the great questions. So that's another topic. It has been studied recently a lot. This integrated analysis of multimodal single-cell data, because we have proteomics data and we have single-cell attack-seq. That is like the next common data that we have. We have single-cell attack-seq and we have the single-cell RNA-seq data. And what we want to do is we want to integrate single-cell attack-seq and single-cell RNA-seq. And we can integrate these data sets in gene level. We can predict the gene level activity of cells for the single-cell attack-seq, and then we can use some integration methods to integrate the single-cell attack-seq with the single-cell RNA-seq data. One of the ways can be like this transfer learning based methods, and also all these the correlation, canonical correlation analysis that I was mentioning, that we try to align those two data sets into the same space. That is another thing that we can do, and there are different tools available, like Seurat pipeline is also a nice tool. Yeah.
Thank you for your answer.
Yeah.
Moving on, machine learning has some challenges, such as single-cell omics data quality control, noise processing, and batch effect. How can we solve these issues?
Yeah, again, we have we know all of these issues exist in the data, so we have to be careful when we are doing the analysis. We should be aware of what kind of problems that we can have in the data, so that we don't make any non-biological conclusions out of this data. So we have to remove the batch effect. in order to properly align the data sets coming from different studies. And we have to remove the noise. As I said, like we can either do imputation, and quality control is also important because we might have a lot of barcodes in the single cell data that don't contain any cells. So we have to remove all these barcodes within the data. Yeah. But there are like lots of tools for all of these things. People have been working on this for a long time. Yeah.
Thank you for your answer. Our next question is coming from YouTube. Do you think the learning difficulty is a disadvantage for researchers because of the exponential number of approaches for dealing with single-cell genomic data? With over 800 published tools for scRNA-seq analysis, most of which are based on ML approaches.
Yeah, that's a great question. That is an issue that I agree. There has been a publication like last year from, again, Taste Group, that they said like there are more than 1,000 tools available for different single cell analysis purposes. I agree that there is like a lot of tools for different problems in single cell RNA-seq, but the good thing is like it is still improving, so it's building on top of each other. So the reason why we had to develop our own pipeline to analyze our in-house data is because the existing methods were not effective in analyzing our own data. Because it was not brain cancer specific. The tools that are available were not brain cancer specific. They were not really asking the questions that we were interested in. So we had to come up with another tool, even though there exists like tons of different other tools. So there is still a need for new methods and tools because everything is like, every problem has its own questions, and like answering those questions requires sometimes developing new tools. Yeah.
Thank you for your answer. Our next question is coming from YouTube again. How can to analyze the single cell omics data with the increasingly larger gen expression matrix contained many zero item and data with multiple types of noise generated by different sources?
Yeah. So again, we have to be careful about all these issues, and so we can either follow some standard analysis pipelines that are available to like remove the batch effect and noise and things like that, and also look at the data by eye a lot, like going over all these like plots and trying to make sense out of the data by really being together with the data. Yeah.
Thank you for your answer. Our next question is, while differential expression analysis, gene expression may largely vary within a group or be expressed by a percentage of cells at the single cell level. How can we determine and evaluate whether these effects were representative of a phenotypic shift or a subset of cells which change during cancer progression?
Yeah, so most of the standard tools, so that's a great question. So usually, like, as I said, like the typical way of analyzing the data is reducing the dimension, clustering, and then looking at the changes in expression within each cluster among different conditions. So, but these pipelines have the assumption that all these clusters are like really homogeneous. But in our case, in the brain data, we have seen that it was not the case. So we, instead of doing cluster-based analysis, we did single cell-based annotation with the hope of having hopefully more homogeneous like cell groups. So that is an issue. If you start from the cluster-based analysis, if your data is really heterogeneous, like brain cancer, then you might end up with a lot of variability within one cluster. So it might not be very easy to evaluate the expression changes. and you might not really know if it's because of any artifact or if it's real. Yeah, so that's an issue.
Thank you for your answer. Our next question is coming from YouTube. Which features of HCVATR can be distinguished from other available methods?
Yeah, so we developed this tool because in our in-house data, we would like to identify the tumor cells. So the input is the single cell RNA-seq reads. So we pull all the reads coming from all the cells, and then we call mutations, and then we go back to each cell and genotype each cell by each mutation. And so at the end, we what we do is like we map the mutations to the UMAP or the reduced dimension, and then we do shuffling, coverage aware shuffling to assess the density of the variant within each cluster, so that we can have an idea whether that cluster is tumor or not. And we also look at the cosmic annotations, and we look at rare mutations. We filter out all these like mutations. We don't look into common SNPs. We focus on the mutations that are reported in cosmic database. And yeah, that's it basically.
Thank you for your answer. Our next question is, today we have a lot of approaches in data science, but why could not we develop common, powerful, and reliable analytical methods and computational tools?
So that's a great question. So like in like there are like deep learning tools applied on different data sets, like images currently. Like we can try to distinguish if an image is a cat or a dog and other like another object. So those questions are, I think, easier compared to the biology, because biology is like really complex. We don't really know what it is. We don't even even the training data sets that we have with the labels, they may not be correct because there's no correct labeling, because we don't maybe have discrete cell types. We might have a cell type spectrum. It can be like not a discrete cell type, but like a cell state. So even having we don't even have well annotated training data sets. So I think biology is much more complex than the other data science problems that we have for images, for other maybe language models that we try to have in the ChatGPT or things like that. So we don't have the the training data, well labeled training data. The biology is so complex, and everything is all these genes are interacting with each other, and yeah, so it's not very easy to get that information out of the models.
Thank you for your answer. And our last question is, which advices Dr. Serin Harmancı may give for students studying in scientific fields?
So it's like currently it's great. Like I'm also learning. We are continuing on learning. If we are not learning, then we will be out of date. So that question applies to me also. So I'm trying to learn, for example, all these deep learning methods and techniques because they did not exist when I was studying. So the good thing is there's a lot of resources available online, and all these courses are available online. And for coding, all these AI tools are a great start. If you ask how you can write this in Python or how you can write this in R into all these AI tools, that gives you a good starting point. But from my experience, I think it's great that we have a lot of resources available. We have all these Coursera's and YouTube videos of all these professors. It's great to have all these things, but I think it gets more useful if you have a problem in hand. let's say if you want to solve, you have to start with a problem. You have a data, and you want to solve. You have a problem that you want to solve using this data, and then you start learning, because otherwise you just listen something and then you forget about it. But if you are trying to solve a problem, then you actively try to understand how you are going to resolve it. So that's how I do it, basically. That's how I try to learn myself. Yeah.
Thanks for the great advices.
Thank you.
On behalf of myself and all our participants, I would like to thank you for spending your valuable time, transferring your knowledge to us, and for this informative presentation.
Thank you. Thanks a lot. And also, I would like to say, like, if there are from the earthquakes and all these disasters, like for the victims, I would be happy to be in contact, also give advices about any computational problems that they might have, and also anybody who is interested in this topic, you can write me, and maybe we might have some positions available. Thank you. Thanks a lot.
Thank you so much. I wish you good and healthy days.
Thank you.
At 4:30 p.m., Dr. Serdar Bozdağ will be with us to give a speech on integrating multi-omics datasets using graph representation learning. Thank you.
Bioinfocongress IV. Associate Professor Dr. Serdar Bozdağ. Integrating multi-omics datasets using graph representation learning. Associate Professor Dr. Serdar Bozdağ received his bachelor degree in computer engineering at Marmara University and Doctor of Philosophy degree in computer science at the University of California, Riverside. After a postdoctoral fellowship in National Cancer Institute at the National Institutes of Health. In 2012, Dr. Bozdağ joined Marquette University as an assistant professor in the Department of Mathematics, Statistics, and Computer Science. In 2015, he received the Way Klinkner-Young Scholar Award. In 2019, he received the NIH prestigious Maximizing Investigators Research Award, a 5-year single PI research grant. In 2020, he joined University of North Texas as an associate professor in the Department of Computer Science and Engineering and the Department of Mathematics with an affiliation with the Biodiscovery Institute. Dr. Bozdağ has served as a program committee member in several bioinformatics and machine learning conferences, including ISMB, KDD, BIOKDD, BIBM, and BCD. He is an editorial board member of PLOS ONE and Cancer Informatics journals. He has been co-organizing the annual international workshop on high-performance computing, big data analytics, and integration for multi-omics biomedical data since 2020. Dr. Bozdağ's research goal is to develop open-source integrative computational tools to analyze high-dimensional biological, clinical, and environmental exposure datasets to infer context-specific gene regulatory interactions and modules, and to predict disease-associated genes and patient-specific drug response.
Dear Associate Professor Dr. Serdar Bozdağ and participants, welcome all of you to our Bioinfocongress IV event. Dr. Bozdağ will give us a talk today on integrating multi-omics datasets using graph representation learning. At the end of the presentation, we will have a question-answer section prepared by our team and consisting of questions from our participants. Dear Dr. Bozdağ, welcome back. Thank you for being here.
Thank you. Thank you for the invitation.
If you wish, you can start your presentation.
Absolutely. Thank you for the organizers for inviting me to this wonderful program. I checked yesterday's videos, really wonderful speakers, wonderful talks. So I will be talking about some of our recent research on integrating multi-omics data sets, particularly using graph representation learning today. So let's get started. So as you all would know, that human genome sequencing project was one of the important, very crucial milestones in the recent history for biomedical science. The project itself started in 1990. It took 13 years to complete, a little less than what they anticipated in the beginning. The cost was about $3 billion. Nowadays, we can do the same thing in a couple of days, and it would cost less than $1,000. So it's a great improvement, achievement for the biomedical field. With that technology development, lots of data sets have become available. Many international consortiums have been established to generate many different biological big data sets, from gene expression to protein expression to transcription factor binding sites. Lots of different data sets are publicly available. Anyone could download and utilize those data sets, those huge data sets. Most of those data sets are based on the central dogma of the molecular biology, where we try to understand the protein activity from the DNA data. So the transcription, at the transcription level, we look at the gene expression, and from the gene expression, we predict the activity of the proteins, because proteins are the building blocks in our bodies. They are involved in many functions. So to understand the gene activity is really crucial. So most of those data sets, you'll see that gene expression data is generated as a proxy of the protein activity. Gene activity is crucial because it's been already established that the gene activity is tissue specific and also disease specific. If you look at if you collect different RNA from different tissue, look at the RNA abundance, gene expression, you could be able to differentiate what tissue it came from. You could also look at the data from two types of cancer, or cancer versus normal, or one disease versus another disease, and look at the gene expression data. You might be able to distinguish the diseases as well. So our group and others have utilized such data sets to build predictive models to better stratify patients into subgroups, like glioblastoma patients, brain cancer patients, into different subtypes based on their gene expression value, as well as their DNA methylation activity. So all of those are great, but when you look at only one data set at a time, you might be able to mischaracterize the disease or the current underlying biology. To be able to really understand the whole biology, or at least move towards to that direction, it's crucial to look at different data sets simultaneously and utilize the complementary effect of those different data sets, what they tell us. Otherwise, we may not really fully characterize the elephant in the room. Like one maybe striking example for that is I'm going to give about the glioblastoma example, GBM. So back in 2009, the Cancer Genome Atlas group published their landmark study on GBM, gene expression data. So they've done unsupervised analysis and came up with those 4 subtypes of GBM. So those 4 subtypes are proneural, neural, classical, and mesenchymal, and this is the gene expression heat map for some number of genes. And we just see that there is a distinct separation between them based on their gene activity. A year later, so particularly let's focus on the proneural group, because a year later, there was another study based on the DNA methylation of the same cohort, GBM patients. Now we do unsupervised analysis. We cluster the GBM patients based on their DNA methylation. And this time, 3 groups emerge, particularly one group, this is called GSIMP positive group, and the GSIMP negative group. That this group is being shown to divide the proneural group into 2 subgroups. So the proneural group is the group over here. Based on the gene expression, they look pretty homogeneous, but based on their DNA methylation signal, there's a striking difference between them. So some of the proneural groups are in this blue group, some of them are in this red group. And when we look at the survival data for those patients, so there are 20 GSIMP positive group and then 49 GSIMP negative group, and this is the blue one is the non-proneural, the black one is the non-proneural group. Now, when we look at the survival difference, so this is the survival curve. For those of you who are not familiar, x-axis is the time after diagnosis, and y-axis is the probability of survival. In the beginning, everyone in the cohort is alive, so we are at the 100%, and as time passes, the survival rate decreases. And in the blue group, within 240 weeks, almost the entire population passed away. Whereas in the red group, they are still alive. So the red group is doing much better survival compared to the blue group. And again, based on the gene expression data, we cannot really understand this difference. So utilizing more data modalities is the crucial thing. So I'm gonna use multi-omics data sets. So I want to just briefly tell you a little bit about those data sets. So what we talk about DNA methylation, which is an epigenetic signal, basically, in our genome, meaning that there is no change in the DNA, but there is a chemical change, especially in the cytosine. There's a methyl group added to the cytosine nucleotide, and because of that, that nucleotide is methylated, and this methylation has a huge impact on the activity of the genes. When we look at the gene expression versus DNA methylation correlation, there is usually a negative correlation between methylation and gene expression, meaning that when methylation is up, gene activity around that area will be down. Another omic data is structural changes in the DNA. These are not unlike DNA methylation. It's not epigenetic. It's a genetic change. It's either amplification. A portion of the DNA is amplified, so there are multiple copies of that, or it's deleted, so that copy is lost. Usually we have 2 copies of a gene, but due to copy number changes, we might have more or fewer copies. So this is also another indicator of gene activity. there are lots of biomarkers associated with different diseases based on the copy number data. Another one is microRNAs. These are short non-coding RNAs, and previously they were thought, oh, these are just junk information, not useful. But then it's realized that microRNAs actually bind to the mRNA, especially the 3' UTR of the mRNAs, and they inhibit their translation. So mRNA is not translated to protein through translation due to microRNAs. So microRNAs also have been shown really involved in several normal physiology as well as diseases like cancer. There are several onco microRNAs and also tumor suppressor microRNAs available. So utilizing microRNA data is also a crucial thing because it tells us something additional, some additional information about the underlying biology. So maybe in 1990s, the biggest bottleneck was to generate the data. It was taking years, billions of dollars to generate the data. But nowadays, that bottleneck is no longer exists. Data generation is relatively easier, faster, and cheaper. The new bottleneck, in my opinion, is on the computational side. All those data sets, how we're going to analyze, integrate all those data sets to generate some interpretable results, not only like some predictions where nobody knows why this prediction is made, but also interpretable so that it can be also applied in the clinical setting. Epigenetic data, RNA expression data, structural changes data, as well as the collective knowledge base that is growing every day, gene ontology, protein-protein interaction, pathways, lots of annotations about drugs, diseases, phenotypes. All of those could be added together as multi-layered approach, genome, epigenome, transcriptome, proteome, metabolome, and could be able to utilize in various aspects of biology, particularly precision medicine, where the goal is to come up with individualized treatment options based on their based on people's individual data, not only coming from the multi-omics, but also environmental exposure, like the diet, lifestyle. This kind of information could be also added to the ingredients of lots of other data sets to be able to better build predictive models, better clinical decisions. So to this end, I want to tell you that in our lab, we are dedicated to this problem, how we could come up with computational tools that will combine different data modalities. I would say that at the core of our lab, we do machine learning, deep learning for various problems. Like for several of the cases, we do multi-omics data integration, and we do work all the way from at the cellular level, trying to understand gene regulatory networks for different contexts, for different conditions, as well as at the population level, some work on precision medicine. So today I'm going to talk about at the population level type of work, where we've come up with a method to do cancer subtype prediction, integrating multi-omics data sets. And for machine learning, we are actually utilizing a graph representation learning method called graph convolutional neural network. So this work is available on bioRxiv as a preprint at the moment, and it's under review at a journal. The code is available on our GitHub page, and this work was mostly done with by my PhD student, Zinet Nesibe Kesimoğlu, who is graduating hopefully this summer. So I'm gonna give you some background on this work. I'm gonna share our results, and then I'll conclude and take any questions you might have. So as you all know, cancer is one of the leading causes of death, as mentioned in Sudraka Mukherjee's book, Cancer is the Emperor of All Maladies. The number of cases increase and is expected to increase partly because there are more diagnostic methods nowadays, but also the economic burden of cancer is also huge. Among those, breast cancer is the most prevalent cancer type in the world at this time. There are different groups of breast cancer, and depending on those groups, the survival rate differs, the treatment options differs. So one grouping is based on the biomarkers, estrogen receptor, progesterone receptor, HER2 protein. So if those receptors or protein abundance exist in the patient, then there are certain treatment options that would target those receptors. But there are also cases where none of those are positive, so it's a triple negative breast cancer, which is more aggressive type of breast cancer. But still, when breast cancer is diagnosed early, the survival rate is way better compared to 15-20 years ago. So this is based on the immunohistochemical data, but also there are molecular subtypes of breast cancer. So based on the gene expression data, again, we're coming back to the gene expression data. As I said in the beginning, gene expression data can be a good data set to characterize the diseases, and it was easier to generate as well. So for many diseases, for many tissues, gene expression data was generated. That was one of the most commonly used, like omic data, transcriptomic data. So in 2009, Parker et al. Identified 50 genes out of 20,000 plus genes in human. Those 50 genes were identified to cluster breast cancer subtypes into 5 groups. So in the heat map, we see those 50 genes and those 5 subtypes: luminal A, luminal B, normal-like, HER2-enriched, and basal-like. So this is totally based on the gene expression data, and based on those 5 subtypes, there are different characteristics that exist. In fact, the TCGA group, when they published their landmark study on breast cancer, they show for different data modalities what are the character signatures for each of those 5 subtypes. So you see the 5 subtypes here, and then we look at the mutation data, clinical data, copy number data, and look at the differences between those subtypes. And this is also their clustering and how that clustering concords with the PAM50 clusters. Okay. So based on all of those, like we have not only gene expression data, but different data modalities for breast cancer. We ask, can we utilize those other data modalities to predict cancer subtypes? And the other question is, okay, if we could do that, because more data might give us more information, how are we going to integrate those multi-omics data sets was the next obvious question that we had to tackle. So multi-omics data sets and clustering based on multi-omics data set is a really popular area. There are several methods on this, like iCluster. iCluster Plus is one of the methods where the goal is, by applying dimensionality reduction on different data modalities, trying to make the data sets align at the latent space. Similarity network fusion is doing a different approach, building patient networks, patient similarity networks for each of the different data types, and integrating, merging those networks by applying a nonlinear fusion step. On the other hand, pins plus is also another clustering method, and the intuition behind this method is that if two patients are in the same group, so they are in the same subtype, then if I perturb their data slightly, still they should end up in the same cluster. So they do perturbation on the data and check how stable the clusters are. If certain samples end up being in the same cluster through those perturbations, then they end up being in this same cluster in the final results. So like in CNF, graph is a natural data structure to represent biological data sets. So we see lots of examples of that in biology. We look at the gene regulatory networks, we look at the protein-protein interaction networks, we look at co-expression networks, we look at gene ontology. All of those different data modalities are represented as graphs. Once we have the data as a graph, then we can basically identify communities. We can identify nodes with too many connections, so which could be maybe the important nodes, important genes, important terms, and we could also identify different groups that work together potentially. So what we want to do is a little different than that. We would like to build those graphs where each node is a patient. But our goal is, what is the subtype, breast cancer subtype in this particular example of that patient? We would like to make a prediction. So it's not unsupervised analysis, it's a supervised analysis. We want to do a prediction, but how am I going to do a prediction on a graph data? Graphs are not Euclidean space. For a graph, there is no ordering. For a graph, there is no fixed features. So I need to do something, and this is called graph representation learning, node embedding, network embedding, graph embedding. There are different names for this, but the idea is each of the nodes, can we come up with a vector representation? Just a bunch of features, maybe 100 or 200, maybe 500 features, where the similarity of those vectors in this embedding space will reflect the similarity of the nodes, like they are close to each other in the network. So they have similar topological structure, or they have similar labels. So from the graph data, come up with an embedding where the similarity in the embedding will reflect the similarity in the graph. And once we have that embedding for each of the nodes or for the entire graph, then using machine learning methods will be straightforward, because it's just going to be like a tabular data now. So we could do node classification, we could do link prediction, we could even do community detection again, like applying clustering methods on that embedding data. All of those are possible. So in the recent years, these methods have become extremely popular because graphs are great to basically load lots of different data sets. And then from graph to machine learning tasks, we need this graph representation learning step to move forward. So there are two streams of work in this domain. One of them is the the so-called the shallow network embeddings, where this is mostly based on a random walk approach, but there are also some matrix decomposition based methods as well. And there is deep learning based methods, which are collectively called graph neural networks. And that's what I'm going to talk in more detail in the next couple of slides. So this part of the talk is a little bit technical. For those of you who are interested in this concept, I wanted to add these details. I also put some links to some tutorials. So Leskovich group generated really nice courses and tutorials on this. So if for those of you who are interested, I would highly recommend. So to be able to do graph embedding using deep learning, so graph neural networks, what we start with, this is like a very high level picture. Basically, what we see is the graph is an input, and then we do some type of graph convolution to gather information for the neighborhood of each of the nodes. That information gives us some initial embedding. And this embedding is passed through an activation function, goes to another layer, and in that layer, maybe we'll do some regularization like dropout. Maybe we'll have another convolution layer, and then at the end, we have this output layer where we have the final embeddings. So if you are familiar with neural networks, this is the same idea. It's just how we define the graph convolution is the part that we need to figure out. But the rest is very similar to the neural network approach. So the graph convolution thing is based on two things. One step is we're gonna define the neighborhood of a node, and we will do the aggregation from those neighborhoods. And how we're gonna do this aggregation, are we gonna do the sum or the average? Those are some of the questions, and that's how the different methods differ. So here in this toy example, we have a node A. I want to learn the embedding of this node. And to be able to learn the embedding of this node, I'm going to look at the neighbors of this node. The immediate neighbors are B, C, and D. So I'm going to get the embedding of B, C, and D, aggregate them, will feed that information to a neural network, which will be trained. And that through that process, I'm going to get the embedding of A. But this is a two-layer model in this example. So two-layer meaning a B also gets the embedding from its neighbors, which is A and C. So the information comes from A to C to B, and from B back to A. So in a two-layer approach, we get the neighborhood. So for instance, A that way gets the information from the two-hop distance. It gets the information from the immediate neighbors, B, C, D, but also the neighbors of the neighbors, for instance, F and E, and collect all this data and come up with the final embedding. If you go a little deeper to the math, this is the idea. And again, if you are familiar with the neural networks, this formula is also not too complicated. So we have a multi-layers in this example, 2 layers. Here, K is the total number of layers. Based on the previous layer, we look at the information from the neighbors in the current step, the neighbors. We look at the embedding of the neighbors. We average that embedding, and we do a linear transformation on that embedding, and we add it with the current embedding, and everything is passed through a nonlinear activation function, and that will give us the embedding of the K+1 layer. And we continue like that. At the end, we get the final embedding. So initially, we have some node features. Those node features will be our initial embedding. And with that initial embedding, we do embedding at layer 1 and embedding layer 2, layer 3. Mostly we do 2 layers to avoid some over smoothing issues. One of the popular methods is graph convolution networks, and this whole process is summarized into this formula, where H is what we are learning, the embedding. So this is the embedding at layer L. A is the adjacency matrix to represent the graph, and D is the degree matrix. Again, this is also coming from the graph. So H, in the beginning, we have some initial node features that we use as embedding. So all of those are coming from the graph. And the W is what we are learning, the learnable parameter. And sigmoid here is just an activation function like ReLU or sigmoid or softmax, whichever is used, but mostly ReLU is used. Okay, so I'm just gonna, so this is kind of concludes the technical part. So there are some graph convolutional network-based methods on omics data. GCNC, MogaNet are some examples, but there are some limitations. So they use single graph or limited omic data, and they don't analyze multiplex networks simultaneously. Like they analyze one network at a time, not like in the network, there's only information about one data modality. So to address these limitations, we come up with Supreme subtype prediction methodology. Supreme is a node classification framework. We encode multi-omics features, and then we do convolutions on multiple graph structured data. Again, convolutions on multiple networks. We utilize features and associations from multiple data types at the same time. And there's no limit on number of data modalities that you can utilize. So here's the framework. And I also want to say that this is a generic method, not cancer or even not related to biological data necessarily. As long as you have data from different data modalities for some entities, then you can have graphs, and then you can learn the embeddings of those graphs using Supreme. So here's the idea. So let me check the time. Okay. So we basically built patient similarity networks, multiple patient similarity networks for each data modality, let's say gene expression or clinical data or co-expression data or DNA methylation data. For each of those data modalities, we built a patient similarity network. And for each network, here the nodes are patients, and the nodes have some features. So those features are also features from all those different data modalities, all combined. So the topology of the network is based on a single data modality, let's say gene expression. But the features are from all the data modalities. So for all data modalities, we have those networks generated. Then we apply graph convolutional network on each of those graphs separately, and we get node embedding for each of the graphs. And the nodes are the same. They are the same patients. So all networks have the same nodes, but the topology is different. Features are also same because they are coming from all data modalities. So we learned the embeddings. After we learn the embeddings, then we say, okay, we have, in this example, 7 types of data modalities. So we don't know which of those will be most useful. So what we're doing all combinations of them. For all combinations, 2 to the 7 minus 1 cases, we integrate those node embeddings. And optionally, we also integrate the features that we started with over there as well. I mean, they were using the graph, but maybe that information got diluted. So adding this feature is also another option. And then after that, we do the prediction. For prediction, we use any machine learning task. In our example, we use multi-layer perceptron. And the final outcome is the subtype of this patient. Basal-like, HER2-enriched, luminal A, luminal B, or normal-like, which is the PAM50 subtypes that I mentioned 10 minutes ago. Okay. So we use data from TCGA, the Cancer Genome Atlas, and Metabrick databases. We use several data modalities from those data sets. Metabrick didn't have all those data modalities, so we use fewer data modalities for that one. And we also combined these two data sets and did another analysis. So we extracted features, and selecting the top features based on the Baroda R package. We z-normalize all the features and combine them and use them as node features. To generate the networks, we use different similarity metrics like Pearson, Jaccard, Gower, based on the data modality. If it's a binary, then we use the Jaccard index. If it's gene expression data, then we used Pearson correlation. We use the top ads as the patient network. So I'm not going to go into too much detail, but there are these are some of the things that we had to play with. So we tried different values, how many gene ads to keep. And so we then finally we fixed that number through hyperparameter optimization. And again, features from all data modalities are added as node features. So here are some results. So first of all, some information about our data. As I said, we use the data from TCGA and Metabrick, and we also combine them. These are the number of raw features that we use. 29 features from clinical data set, 500 from copy number, 16 co-expression, 1,000 genes from expression data, 1,000 probes from methylation, 343 microRNAs, and 200 mutations. And for Metabrick, the numbers were more or less same. And these are the number of samples. So we have slightly over 1,000 samples in TCGA and almost 1,700 samples in Metabrick. And these are the groups of samples in each of the subtypes. So as you can see, most of them are luminal A. So the subtypes are not balanced. So that's why in our metrics, I'm going to show F1 score, which is more reliable for unbalanced cases. And the proportions are more or less same for the Metabrick. Again, luminal A is the most prevalent group. Then we have normal is the fewest case. And then we combine all those data sets together to generate this combined data set. Okay, so remember, we try all combinations of all data modalities. So we have a bunch of results, and this violin plot illustrate those results. And based on these results, we were able to show that Supreme outperformed all the baseline methods and state-of-the-art methods like DeepCC and MogaNet and GCNC. And the baseline methods like random forest, support vector machine, XGBoost, and multilayer perceptron. For those baseline methods, we basically use the features and then do the prediction. Here, we're using the features as well as the node embeddings. And here, Supreme minus, we are only using the node embeddings. So the features are not added at the end. If you remember, I mentioned that we might add the feature as an option at the very end. So this Supreme minus is the one without those features added. And on TCGA data, we were able to show that there's a statistical difference between Supreme and other approaches. In Metabreak data, we were able to see the same trend. Supreme outperformed all the other methods. And when we combine, we were able to also show that there's like Supreme is doing better than the other methods. In this case, the number of data modalities is only 3, so there are only 7 runs. Because of that, it's hard to show statistical significance. So those values are not statistically significant, but that's because of the number of samples. The other thing that we've done is, okay, so we're doing this prediction. So we have 5 groups based on our predictions. Can we look at the survival differences between those 5 groups? And is there a significant survival difference between them? If there is, that's a good thing, because then our differences, our subtypes are meaningful subtypes. So here we look at the survival difference, statistical significance of the survival difference. So we get the p-values. Here, the lower the better, because it's more significant. And what we see here is Supreme and also Supreme minus, for most of the runs, got quite well separation. The other methods, there was significant difference with all the other methods, as can be shown here. Some of the methods actually got really high p-value for most of the cases. Some of them were still like giving low p-value, but still there was a statistically significant difference. Here, the solid red line is the 0.05 cutoff, and the dashed line is the TCGA like the ground truth PAM50 subtype survival difference p-value. So this is the p-value that we get with the real data. So if you could zoom in to this plot, 0.05, 0.03 is what PAM50 is doing, and most of our runs are doing way better than even the ground truth. So again, I want to say what this implies. Like, we are making subtype prediction. If we do exactly same as PAM50, then our accuracy will be 100%, our F1 will be 100%, because we use PAM50 as the ground truth. But we are making some mistakes. Because of that, our subtypes are different, but those differences end up give us a better survival difference. So perhaps by using other data modalities, we are saying that a luminal A group is luminal B, for instance, and we also show that based on the survival difference, that our grouping is doing better. Like, this is the Supreme subtypes versus PAM50 subtypes. We look at the Kaplan-Meier plot, the plot that I showed in the very beginning when I gave the TCGA GBM example. X-axis is the time, survival probability is the y-axis, and what we see here is we compute the statistical significance between the differences between those curves. So it's 0.01 for Supreme, and it's 0.03, which is the 0.03 that you see here for PAM50 subtypes. It's hard to distinguish by eye, but it just says that the difference is higher for Supreme case. We also look at the single data modality. So what if we only focus on one data modality, not all 7 of them? So it's just one run in this case, but we repeat that run multiple times, so we have some standard deviation shown here. So for most of the data modalities, Supreme did better than other methods, except for a couple of cases. Sometimes Supreme minus did better, but most of the time adding the features at the very end showed us some useful feature. And we got this more or less the same trend for the Metabrick data as well. So in conclusion, I today talk about different data modalities. I talk about graph representation learning, especially deep learning on graphs, and discuss you about a tool that we recently developed, Supreme, which integrates different data modalities, and we show the results on breast cancer subtyping. Our results show that we outperformed baseline and state-of-the-art methods, also got significant survival methods. And this tool is publicly available. It can be applied to any other data type, disease type. It's very generic and user-friendly. With that, I like to give acknowledgement to my students, particularly Zinet, who did most of this work, and our funding agency for their funds. I'm really grateful to be working with a wonderful group of students. So we do lots of research, but from time to time we also have fun together. Next week, for instance, we're gonna have another lab lunch to celebrate Zinet's graduation. So she defended her dissertation successfully 2 weeks ago. So we'll celebrate that. So she will be joining NIH as a postdoc soon, hopefully. With that, I also would like to tell that we are hiring. My group is hiring postdocs and PhD students to work on those projects. We are in Dallas, Fort Worth Metroplex area. It's a wonderful place. It's an extremely growing area. The weather is beautiful. Schools are great. Like if you have kids, there is direct flight to Turkey and many other places in the world. And UNT is a R1 university, a research intensive university. So if you're interested, feel free to email me. So I put my contact information over here. Thank you so much. So I'm happy to take some questions.
Thank you so much, Dr. Bozda. I am directing our first question. In future, can Supreme be utilized attention mechanisms which allows getting weighted contribution from different data types and also weighted neighborhood from networks?
Wonderful question. So I'm really happy to hear this question. Actually, this is what we're working right now. So probably in a couple of weeks, hopefully, we're gonna release another preprint where we do attention mechanism between different data modalities, as well as between different nodes. So at the node level and at the network layer level, we compute attentions and we do the integration based on that. So we have some preliminary results. Hopefully, we're gonna make it available really soon.
If you'd like to, you can close your presentation and see the questions yourself.
Sure.
Okay, thank you. Thank you for the response. I'm moving on to the next question. GNNs learn a node's features by iteratively propagating information from the neighboring nodes until convergence. They have high computing costs and the learning filters lack of the localization attribute. Is there any solution?
So, right. I'm not sure I fully understood the question, but yeah, the cost of analyzing GNN is out there. So there are some scalable GNN methods that could be use there. So there are some papers, like published from different groups, like Leskovich group recently published a work on that as well. So there are some approaches. So to be able, so first of all, so when we run, our most of the time we spend is on hyperparameter tuning. There are some parameters that we need to optimize, and for that we have to run many times. Each run is not taking lots of time for us, but again, our graphs is relatively tiny. But if there's like, let's say graph like a social network where there are millions of nodes, then this becomes there's a scalability issue. But yeah, there are some work on that domain to address this issue, but still it's an ongoing research problem.
Thank you for your response. I'm moving on to our next question. Omics are heterogeneous entities with different data distributions or data types. How is it possible that these inconsistencies between omics do not hinder their integration?
Oh, it does. It does. Like if you simply say that, okay, let's aggregate all the features all together and then do the prediction, more the merrier. It doesn't work that way. If we force that these omics data sets had to like align with each other, then that's also another problem, because as illustrated in this question, there could be limited data, or there could be some bias or noise in the data. And if we really force that all those data sets need to agree with each other, then we might be able to lose some of the signal. So I think the key thing is, and this is a research problem that is not fully resolved yet, but I believe that there needs to be a trade-off between, like, at some point we need to say that this particular data piece coming from this particular data modality is not contributing, it's not agreeing with the others, so there is a higher chance that this is not complementary but noisy, so we should eliminate. But how to do that automatically in a customized manner, that's difficult. And again, that's why this is an active research area. Hopefully down the road there will be better solutions.
Thank you for your answer. We have a question from YouTube. Can self-supervised graph representation learning integrates multiple molecular networks and decodes gene-disease relationships?
Yes, it does. So so graph representation learning is a pretty custom method. So there are different downstream things that can be done. So this is also another work we're interested in. So we publish a work which is not using deep graph representation learning, but shallow learning method for this problem, like gene-disease relationship in a sense. So once you have multiple molecular networks, let's say a gene network and a disease network, and even the gene network could be multiple layers, the disease network could be multiple layers between different disease ontologies, then you can define some tasks that tries to be able to do the self-supervised learning. You need to come up with a task for the program to learn by itself. And that task could be just based on the topological similarities of the nodes. For instance, sometimes this gene network graph is perturbed, so you have a different graph, and you try to say that the node embeddings that you learned will be similar in the real graph compared to the perturbed graph. Or there are some other tasks that you can come up with. After that, once you learn the embeddings, then to be able to do the link prediction, what people usually do is the embeddings of these two nodes are passed through a multi-layer perceptron, for instance, and then you do a machine learning, or you do dot product between these two embeddings, and based on that score, you say, okay, there's a link between this gene and disease or not.
Thank you for your answer. We have another question from YouTube. More data brings with it more information, more noise, redundancy, and increased computation time. How do you overcome this problem in multi-omic integration?
Yeah, so it's, again, there are different paradigms for multi-omics integration. So it's a really hot area. So we've been organizing workshops for the last few years on this topic, and many other groups are also working in this area. So there is not a single solution, but there are different approaches. Like there is early integration approach where all data sets are integrated in the beginning. There is late integration approach where data is handled separately and you make some final outcome or generate some type of process data from each data modality and then fuse them later, like our Supreme is a late integration example. There are also hierarchical integration approaches where some data modalities are known to be related to each other, like utilizing this biological knowledge into the integration process. And completely different from like graph representation learning paradigm. Several years ago, like we've done this, and many other groups also have done that. Like some omic data sets could be utilized to generate some prior knowledge, prior network, and then using that prior information on a Bayesian approach could be another method for the multi-omics integration. But but I don't have a final answer to this problem. Again, today we're working on this. In the maybe 10, 15 years, we'll have a better understanding on this, because the data noise will hopefully reduce, and we'll get more data modalities, and we'll have a better methods to handle these issues.
Thank you for your response. Another question from YouTube. Since graphs are formed based on samples rather than features, the complexity of the whole pipeline does not significantly increase by adding new omics modalities. Is this an issue on data?
So for this particular work, our grant truth came from the gene expression data. Remember, the PAM50 subtype labels are based on the gene expression data. So we've done great when we included gene expression features to the results. But again, that makes sense because the data, the grant truth, is based on gene expression. When we didn't have that, even we have other data modalities, we were not be able to achieve extremely high performance. I think this case is not extremely clear whether adding more data modalities are improving the results or not, because there are some ablation studies that we've done. I didn't mention in the presentation. For some omic layers, if we remove them, we didn't see much difference in the performance because the other data modalities were able to handle it. But then some omic layer, especially I remember copy number, for instance, when we don't have the copy number, then we had a significant drop in the performance. So yeah, there are some mysteries as well, but I think there are more tests that need to be done to be able to say whether certain data modalities is always useful or not useful at all.
Thank you again for your response. I'm moving on to our next question. Why do we not pursue an easier strategy than unsupervised learning? That is to train an end-to-end GNN in semi-supervised fashion.
Yeah, so we wanted to make this method as a generic method where the goal is to learn the node embedding. What you do with those node embeddings could be differ. But if you have a specific problem, yeah, end-to-end model could have been done. So our approach is in two phases. We learn the node embedding, and then the next problem becomes the machine learning problem of subtype prediction. But this was intentional. If we had the goal of just subtype prediction, we could have made the model end-to-end, and like the back propagation goes all the back to the beginning of the pipeline. Yeah, this could be done, definitely.
Thank you again for your response. Another question from YouTube. Could tasks such as the creation of multigraph representation learning-based ensemble models and prognostic models and molecular subtypes for specific diseases become easier in the future?
Yeah, I think it will be easier in the future. Like the history taught us that as time passes, we learn more and more things, that we end up with more difficult problems to tackle on, but the problems that we work on today will be, yeah, relatively easier down the road, because there are, like when we started this project, back then we didn't, like this project started many years ago when there was not even graph representation learning. We've tried different machine learning methods in the beginning. Then we switched to graph representation learning. But then when we started, we used GCN. And after that, attention-based methods emerged, and many other advanced methods emerged. So yeah, the field is moving really fast. And if you look at some machine learning conferences, this is one of the most popular areas. Every day there are really high quality publications. So yeah, I hope that the problems will be easier and easier, but we'll come up with new problems to work on, which will be the next level.
Thank you for the response. Another question from YouTube. There are some regulatory relations, such as competing endogenous RNA regulation, which has been recently discovered with important insights into cancer. To adopt this kind of regulatory relations, can Supreme be used?
Yes, actually, Zinet worked on this problem, cRNA interaction, as her first project, and that's one of her future goals as well. Like, can we utilize this information based on the cRNA network? Can we get another omic layer to this? Maybe whoever asked this question, Sedanur, maybe you read the paper, but yeah, thank you for this question. Absolutely, this is what we are thinking at the moment.
Thank you for your answer. Another question from YouTube. When conducting multiple omics experiments, collecting large amounts of data can often be done on a small sample of patients. Wouldn't this cause data sets with a number of variables exceeding the number of samples and machine learning algorithms to overfit high-dimensional data sets?
Yeah, this is a common problem that we face. So here we start with about 1,000 samples, which is relatively high compared to many different examples. But then if you look at all of our features, they are in tens of thousands of features. So to address this problem, we did feature selection. We reduced the features to a more manageable number. So there were about 4,000, 5,000 features at the end for 1,000 samples. And we check for overfitting. So we look at the performance on the training data and the validation data and the test data. And there was not much overfitting in our case. Thanks to the feature reduction, I mean, if you use all the features, definitely overfitting is a inevitable problem.
Thank you for the answer. Another question from YouTube. One of the toughest problems with neural networks is their black box structure, i.e. their non-interpretability. Is having a good predictive model enough for them to be interpretable?
No. So definitely there are some additional work need to be done to be able to better understand what part, what data modality, or even within the data modality, what subset of features are contributing most to the overall prediction. So there are several methods for this. Like for those of you who are interested in this domain, there are explainability methods for deep learning methods in general. Like deep lift is one of them. For GNN explainer is another method. So there are knowledge distillation methods where you try to build a simpler model to mimic your complex model. And this simpler model, like maybe based on decision tree, will be more interpretable. And so you could do this. So this is doable. We've done ablation studies to show the impact of different data modalities, but yeah, more things can be done to better interpret this complex model.
Thank you for your response. Our next question is from YouTube. In your presentation, data sets show different numbers of samples and results. How we can decide which data set is more accurate information?
So the TCGA data is more comprehensive, so they have more data types, but the number of samples was fewer. I mean, it's hard to say which one is more accurate. All of them do rigorous pre-processing to make sure that the data sets are accurate, and they eliminate all the samples that they don't pass the quality control protocols. So we've tried those data sets separately, and we also combine them to look at the results, just to see if what we are getting is data specific. That was one concern we had. Like, we're getting those results because our model fit to this particular database or not. So we were happy to see that we were able to show performance similar like trends in two different data sets. So, I mean, the answer to that question, we don't know which data is more accurate or which data set have the most accurate information. But yeah, we use all the available data that we could use.
Thank you for your response. We have another question. A multi-graphical representation learning-based ensemble models are being developed to predict potential gene disease by extracting gene disease associations such as microRNA diseases, protein diseases, and gene pathways. Do you think that a similar path can be followed to improve the prediction of gene disease association?
Absolutely, and there are right now I don't have any tool or paper to give on top of my head, but there are several studies that utilize this information, like represent different intractome and different layers of information, and to make those associations for sure. As well, gene-drug interaction is also another domain. The similar idea could be applied to find associations between drugs and their target genes.
Thank you for your response again. I'm directing our next question. Mixed integration transforms each omics dataset independently. Much of the large matrix resulting from early and intermediate integration is not utilized by the machine learning model. So how do we determine which approach to use for multi-omics integration?
Yeah, very nice question. I don't know a definite answer to that. In my opinion, early integration is too simplistic, and late integration could be also potentially too late, meaning that if you just rely on 3, let's say, data modalities, and then you just do the prediction of each of them separately, and if you look at the consensus, then you might have just by chance, we might have some mistakes, especially if some of the data modalities have some issues for some cases. So I would recommend, like, don't fix to a particular method, but try different approaches in the beginning, trying baseline methods. If you're able to get what you plan to get using baseline simpler methods, I would go with those. If you are in the computational science domain where your goal is to come up with new methods, then looking at the state-of-the-art methods, different approaches, and try to see under what conditions those methods might fail, might give you some hint about what would be the new novelty, new innovation in developing a new model. But if you're a simpler user, like you have data and you want to know how we're gonna try it, simpler the better. I would say start with simpler models, and then if you are not happy with the results, go to more complex methods.
Thank you for the response. And moving on with the next question. Although there have been advances in causal modeling, existing approaches have difficulties in applying them to high-dimensional datasets due to the numerical complexity in the application of the approaches and the multicollinearity of the data. How do you think these difficulties can be overcome?
Wow, that's a really difficult question, really important question. Yeah, none of those things that we do have anything to do with causality, because I mean we're computing correlation. We do associations basically when we build those patient similarity networks. When we talk about causal interactions, it reminds me more like at the cellular level. For instance, gene regulatory network. Even for gene regulatory network, it's a difficult problem, because when you know that two genes have similar expression profile, those two genes might regulate each other, or they might be co-regulated by a third gene, a transcription factor, for instance. So it's really difficult. Based on the data, it's ambiguous to be able to say anything about causality. But if you use additional data, for instance, if there is ChIP-seq data, and if the ChIP-seq data gives us some idea about what transcription factor is binding to what gene, that could be one way to be able to define some directionality. Other knowledge bases, existing regulator target interactions could be another approach. And also in different areas of machine learning, there is like causal interaction inference methods exist. They apply to social networks, for instance, to be able to look at if certain tweet posts, for instance, influence certain actions. But for that, they have lots of data sets that they could use. For the biological data is a little limited. Like another thing that comes to my mind is if you have time series data, then you look at maybe the cause and effect over time. But again, time series data is not available as much as the static data that we have. Yeah, it's good research area for sure, but the most of the issue comes from the data limitation.
Thank you for your response. As of our last question, what advice would you give to students studying in scientific fields?
Okay, wonderful. First of all, the questions that were asked, I'm quite amazed with the level of these questions. Wonderful. I'm really happy to again give this talk. I'm humbled. For for scientific fields, like if we're talking about bioinformatics, I would say that let me step back actually. So when people tell me like what major to choose for their undergrad degrees, I usually tell them do interdisciplinary, like have maybe another major or minor, and like learning a foreign language, make sure to learn a programming language, whatever area you are in, biology, chemistry, journalism. Still, like learning computer language is gonna make it really useful, because data is becoming available everywhere nowadays. So to be able to handle this data, analyze this data, to be familiar with those methods would be useful. The other thing is just like attending these events are really useful. If you are a PhD student at that level, one recommendation that I would have is just make sure that you write every day, even if it's if even you don't have much to write, still sit in front of the computer and like 15 minutes, not more than that, just write about your project, about your experiment, whatever you have. And that's just helps the brain to digest the data more, and that helps. Nowadays, blog pages and all the YouTube tutorials, they are really useful. So I learn a lot, and myself, and I would also recommend to utilize those resources and learning, to make learning as part of your routine. So writing, learning, and of course doing the research. I said I'm hiring students, so if you're thinking abroad, I would really recommend you to plan for that. Reach out to professors, read their papers, and ask them questions. And yeah, nowadays we're really, I personally have difficulty to hire good students to my group. It's very competitive. So from outside, it might look like that it's difficult to get into those positions, but actually it's much easier if you make the first step. Thanks again for the kind invitation. It was wonderful to be with you all today.
Thank you so much, Dr. Bozda. On behalf of myself and all of our participants, we were so grateful to you. We are so grateful to you for spending your valuable time, transferring your knowledge to us, and for this informative presentation. Thank you.
Thank you. Bye-bye.
I wish you very healthy days.
Thanks.
Thank you. At 6, Associate Professor Dr. Tae-Sok Moon will be with us to give us a speech on systems and synthetic biology, constructing smart and programmable microbes to address global problems. Thank you all.
Bioinforange platformunun kuruluşunun 2. yıl dönümünde düzenlediğimiz Bioinfocongress IV etkinliğimize hepiniz hoş geldiniz.
Merhabalar, hoş geldiniz laboratuvarımıza. Merhaba hocam, hoş geldiniz.
Merhaba, hoş bulduk. Sağ olun. Teşekkürler davetiniz için, organizasyon için.
Davet için tekrar çok teşekkürler. Organizasyon da çok başarılı gözüküyor. Çok teşekkür ederim.
Tekrar teşekkürler davetiniz için. Bugün bilgisayar teknolojisiyle biyomoleküler sistemlerin simülasyonu ve ilaç tasarımı konusunda konuşacağım.
Muhteşem. Çok teşekkür ederim. Evet, hepinize merhabalar. Organize komitesindeki arkadaşlara da çok teşekkür ederim beni davet ettiğiniz için.
Herkese merhabalar. İlk önce davetiniz için çok teşekkür ederim. Buradaki diğer konuşmacılarla beraber sizlerle beraber olmak gerçekten gurur verici.
Bioinfocongress'te sunum yapmaktan beni davet ettiğiniz için çok memnunum ve sanıyorum tamamen insan hücre fonksiyonlarına yönelik olmayan daha basit canlılarla ilgili olan tek konuşma belki. Ama böyle bir değişiklik getirmekten de çok memnunum.
Öncelikle hepinize teşekkür etmek istiyorum. Bioinfocongress oldukça güzel bir organizasyon yaptınız.
Tüm Bioinforange çalışanlarına ya da gönüllülerine teşekkür ediyorum. Beni davet ettiğiniz, zamanınızı ayırdınız.
Merhaba, davetiniz için çok teşekkür ederim. Bu aktiviteyi organize ettiğiniz için de teşekkür ederim. Gerçekten çok büyük bir çalışma gerektiriyor.
Thank you, it's a pleasure. Thank you for the invitation.
İyi akşamlar. Başlamadan önce bu güzel organizasyonu düzenleyen arkadaşlara gerçekten teşekkür etmek istiyorum. Bu kadar büyük bir topluluğu bir araya getirmek, böyle bir organizasyon gerçekleştirmek gerçekten büyük bir başarı.
It's a really fantastic program that you've put together over these last few days. So it's been some brilliant science and some brilliant scientists speaking. So thank you very much again for the kind invitation.
Welcome everyone, I'm Neva Çalışkan. I'm a group leader at the Helmholtz Institute for RNA-based infection research. And today my talk will be about how genome meaning of the genome can be altered by frame-shift RNAs and how this response can be regulated by host and viral proteins.
Teşekkür ederim davetiniz için. Bugün mRNA aşılarından bahsedeceğim sizlere.
Merhaba arkadaşlar, bugün sizlere katılmaktan çok hoşnutum. İlk önce Bioinforange öğrencilerine bu güzel organizasyonu bir araya geldikleri için teşekkür ediyorum. Gerçekten benden önceki videoları izlemekten çok hoşlandım.
Merhaba herkese, buraya geldiğiniz için teşekkürler. Bugün ökaryotik genomların organizasyonu hakkında bir konuşma yapacağım.
Merhaba herkese. Katıldığınız için ve dinlediğiniz için teşekkürler.
Merhabalar, hoş bulduk. Başlamadan önce böyle güzel bir organizasyonda emeği geçen arkadaşlara teşekkür ediyorum. Ayrıca davetiniz için de teşekkürler.
It's a tremendous honor and pleasure for me to be here after starting my lab just over 2 months ago.
Gün boyunca 30 saat bir yayın süremiz oldu. 3 eğitim, 17 konuşma, toplamda 20 konuşmadan görüyorsunuz yorgunluğumuzu da arka planda. Öncelikle bizlere konuşmalar, eğitimler veren 20 değerli hocamıza çok teşekkür ederiz. Açıkçası davet ettiğimiz hocalardan çok yüksek oranda kabuller aldığımız için bu kadar çok yoğun bir programımız oldu. O yüzden hocalarımıza çok teşekkür ederiz. Onların bize davetleri bu kadar çabuk bir şekilde, hızlı bir şekilde bizleri kabul etmesi, konuşmalar yapması bizler için çok güzel ve bizleri gerçekten çok motive ediyor. Hocalarımızın haricinde siz şu anda 277 kişi var bu saatte. Çok teşekkür ederiz. Değerli katılımcılarımıza çok teşekkür ediyoruz. Ancak büyük bir teşekkürü de ben buradaki ekip arkadaşlarıma yapmak istiyorum. Bioinforange'ın 2. yıl dönümü, doğum günü. Buradan Bioinforange'a emek veren, gönül veren, Bioinforange'ı seven tüm bilimseverler, katkı sağlayanlar, öğrenciler, insanlar adına iyi ki doğdun Bioinforange diyorum. Bilim paylaştıkça güzel, bilim paylaştıkça ilerler. Bilimle kalın.
Bioinfocongress IV. Associated Professor Dr. Moon, Systems in Synthetic Biology. Constructing smart and programmable microbes to address global problems. Associated Professor Moon is an Engineering Biology Research Consortium Council member and a Synthetic Biology Young Speaker Series Chair. Associated Professor Moon has expertise in systems and synthetic biology. He aims to solve global agricultural, environmental, manufacturing, and health problems through engineering biology. Associated Professor Moon's projects and prior research efforts have resulted in 72 publications, 125 invited talks, and 10 patents. His achievements have also been recognized with many awards, including a Langer Prize for Innovation and Entrepreneurial Excellence. a BNB Daniel I.C. Wong Award, an NSF Career Award, an ONR Young Investigator Award, a John C. Slaughter Fellowship, an ISU Foundation Award, an LG Chemical Fellowship, and the SNU President Prize. He has been an active member of the global community of researchers who work in the fields of chemical engineering and bioengineering. Also, has actively participated in organizing many international conferences as a conference co-chair or organizer. Nowadays, his global leadership efforts include that one is his activity and role at EBRC as a council member to provide the vision to address national and global needs through synthetic biology, and the other is his service to SymBISS as the founding chair to provide a weekly, virtual, and multi-year forum where a global thought leader gives an opening 5-minute talk, followed by a 45-minute Rising Stars talk for more than 1,000 global audiences.
Dear Associate Professor Dr. Sayaç Moon and participants, welcome all of you to our Bioinfocongress IV event. Associate Professor Dr. Sayaç Moon will give us a talk today on systems and synthetic biology, constructing smart and programmable microbes to address global problems. At the end of the presentation, we will have a question-answer section prepared by our team and consisting of questions from our participants. Dear Dr. Moon, welcome back. If you wish, you can start your presentation.
So, can you hear me?
Yes, I can hear you.
Okay, can you see my slide?
Yes, we can see.
Okay, wonderful. So I will start. So thank you so much for the nice introduction and also invitation. It's my great honor to be here. And hello, everybody. My name is again Taesuk Moon. I'm representing today Engineering Biology Research Consortium as a council member and Synthetic Biology Young Speaker Series as a founding chair. EBRC is a non-profit organization that provide vision to global synthetic biology community and also many government, including US White House and many funding agency. At the same time, I'm the chair of SYMBIS, the seminar series. I'll probably talk about that one at the end of my talk. So thank you everybody to join today, and let's start. Let's start with my dream. My dream is engineering biology. Our ancestors dream of flying in the sky, but instead of creating birds or transforming themselves into birds, they analyze birds, design wings and propellers, and assemble them together to make airplanes. I want to take the exactly same approach. Instead of creating completely new cells, I analyze existing cells, design synthetic DNAs, and assemble them together to make engineered cells that can be used for biochemical generation, biomaterial production, and even for some medical and environmental application, including plastic, removal and some toxic chemical bioremediation. We call it synthetic biology. To solve these issues, I recently started multiple ambitious projects that have high risk and high return, and these are the summary of our current 12 projects. And if you're interested in working on this project as a postdoc or PhD student, please contact me at tsmoon7@gmail.com. So let me start with one question I asked more than 10 years ago. What if we can combine bacterial sensors, circuits, and actuators in a human-defined way? If that is possible, we can use bacteria for our goal, from creation of tumor-killing microbes all the way to toxic chemical degrading bacteria, including plastic-eating bacteria. My first topic is probiotic engineering to achieve one of these goals. I envision our future as follows. We engineer microbes that sense medically relevant signals and respond to them for diagnostic or treatment applications. One of the main challenges 10 years ago when I started my lab was lack of well-characterized application-relevant sensors. For example, we had used lead chemical inducible sensors for so-called toy system demonstrations, but we cannot use these in the human body because these sensor, including IPTG inducible sensor, this chemical might be toxic to human. Also, this chemical, including IPTG, not available in human body, or sometimes this chemical is expensive. Considering the short pH pulse in the stomach and aerobic to anaerobic transition that happening in the gut. By the way, gut is aerobic, anaerobic condition without oxygen. And also temperature shift when the bacteria are released into the environment from the body. My lab has developed pH, oxygen, and temperature sensor, etc. since 2012. These are some examples, including temperature sensor data that kind of showing very interesting temperature response. I'll talk about later briefly. Oxygen sensor and pH sensor. At the same time, my lab also developed biogeographical sensor that would activate only in the large intestine but not small intestine, by focusing on one chemical called propionate that is actually only found largely in large intestine, but not small intestine. Additionally, my lab developed various pollutant sensors for bioremediation, including phenol sensor. An important thing is this sensor only responds to phenol, but not other aromatic compound structurally similar. However, developing medically relevant chemical sensors is still lagging. And actually, other labs have recently developed or used sensors and circuits for various medical applications, as we recently reviewed, and some selected examples are shown here. In fact, we did extensive literature search many years ago. And then during COVID, because we cannot do any experiment, we decided to basically write review papers. And then we surprised by the number of papers or sensor we at that time we had, and that is not that many. And then in fact, this table actually just summarized quite well the total number of sensors we could use at that time. So with that, limited number of sensors, we realized we need to develop new sensors. So another problem is, despite these recent advances, ligand-specific sensing is very critical and still challenging, especially when sensing and responding to structurally similar chemicals with distinct biological outcomes. For example, phenylalanine and tyrosine are structurally very similar. The only difference is one compound does not have oxygen, but the other one has oxygen in the aromatic ring. The problem is these two compounds are structurally very similar, but associated with distinct disorders. Similarly, phenylethylamine and tyramine are neurochemicals found in the gut with distinct biological outcomes. If we use those kind of sensor, that will lead to misdiagnosis, and then doctor could give you the wrong drug rather than the right one. So we need to have specific sensor that only detecting one chemical but not the other. Let me introduce our neurochemical sensor that actually developed for the first time. Notably, we employ a dual knob system consisting of amine neurochemical converting enzyme and corresponding aldehyde binding transcription factor. We confirm the dual knob sensing mechanism by showing that the TIN-A enzyme is required. Interestingly, TIN-A and FIR is actually the transcription factor in this case that bind to aldehyde, that is conversion product of TIN-A enzyme. And then that FIR transcription factor bind to this aldehyde, and that binding trigger interaction between this molecule complex and the DNA, and then that interaction basically lead to GFP production, and then we can see the signal. Interestingly, this TIN-A and FIR homologs are found in many human intestinal bacteria, especially E. coli, but not in our host Lister probiotic strain, enabling orthogonal sensing. Based on this finding, we performed protein ligand docking simulation, noting that the bigger threonine residue compared to the original alanine may play a role in limiting bigger ligand access. Additionally, considering that the ligand is a non-charged aldehyde instead of charged ammonium, not too much hydrophilic but not too much hydrophobic residue would be ideal for PEA specific sensing. Therefore, we hypothesize that size and polarity of the 81 residue collaboratively dictate ligand selectivity. The important part of this slide is not the technical detail I just mentioned, but the most important thing is I was amazed by my student Matt, who approached me one day actually during the COVID. At that time, we are struggling to develop specific sensor because our sensor I just introduced earlier, that kind of using enzyme and then transcription factor, this dual together. But unfortunately, this sensor respond to all 4 chemical we tested. In this case, DA, PEA, TIRA, and tryptar. All of that basically giving some signal. The key problem was how to make this sensor specific enough to only one chemical, but that didn't work. Even when we screen randomly mutated library and total size was more than 10 to the 7 different mutant, and we screen using some gross-based screening method, but multiple try for more than 2 years, and that didn't work. And then one day, Matt approached me saying, "Professor, I wanted to learn some computational algorithm, including AlphaFold or Rosetta. And at the time, I just need to do anything available." And then I just said, "Please go ahead." And then several weeks later, he said to me, "I know how to use those Rosetta program." And then he said, "Let me try what kind of amino acid residue we need to target." And then to me, that's fantastic. So I just said, "Please go ahead. Looking forward to your result." And then, several weeks later again, he identified total, I believe, 5 residue. And then we basically changed all 5 residue to original amino acid to all 20 amino acid. And then we basically tested total 100 different mutant. And then we got some hit, including the one I just showed here, that is a A81L mutant of VRR that only respond to one molecule, in this case, PEA. So that one is actually, published, I think last year in Cell System, and we are very excited to see this paper. And then I also appreciate Matt's effort. So in summary for this part, without showing more detailed data due to the time limit, I'm saying we developed for the first time specific sensor for structurally very similar amino acid and neurochemicals, laying the groundwork for developing medically relevant probiotic sensors. I right now envision our future something like this. we have many patients in the world that kind of suffer from many different diseases, and one of the disease is actually some neurochemical related disease. And another thing is I wanted to develop some probiotic, and probiotic is basically some bacteria, but good bacteria that help our health. And typically that bacteria basically contained in yogurt. I believe many people in Turkey also take yogurt. But what I want to do is this probiotic bacteria in the yogurt, and then everyone just take the yogurt I develop and then drink or take, and then I'm sorry I'm talking about poop, but when you look at your poop, depending on the chemical level in the gut, you see different color in your poop. I'm not sure you heard about neurochemical found in your gut, but several years ago, many scientists basically discover neurochemical. People typically think neurochemical found in your brain, and brain basically produce neurochemical, but some neurochemical actually found in your gut. And also scientists discover some bacteria in your gut actually utilize neurochemical, metabolize, or even synthesize some neurochemicals. I'll give one example. Serotonin is the neurochemical, very important, because that control your mood, happiness, and so on. If you develop serotonin sensor, and importantly, serotonin is produced by not the brain cell, but the gut cell. And actually gut cell produce more than 90% of neurochemical serotonin in the gut. And if you have the neurochemical sensor that's specifically sensing serotonin, the probiotic bacteria come out of your body, and then in response to serotonin level in your gut, you can make serotonin level detector by just drinking yogurt that contain your probiotic. So my goal is something like this. Probably 5 years later or 10 years later, you drink my yogurt, and then you see your poop, and then today your serotonin level is, let's say, 100, and then bacteria basically produce some green color, and let's say 100 is good level of serotonin, and then today is a happy day. And unfortunately for tomorrow, your bacteria now sense less than 20 serotonin in your gut. That means you are not happy day. Today is not happy. Tomorrow is not happy day, because your bacteria will generate red color instead of green color. And why but we stop there? And then my additional goal is I wanted to manipulate that probiotic bacteria that basically sense neurochemical. Let's say serotonin level between 20 to 100 is the right level, and that is good, happy level. And if the bacteria sense too much neurochemical serotonin, and then bacteria start to produce, in response to that high level, enzyme that degrade serotonin. In that way, the range is right for the happiness. If the level is too low, now bacteria start to produce serotonin, making the serotonin level in between that the right level. So maybe 10 years later, if I'm successful, the commercial may say, do you want to be happy all the time and drink yogurt, and then you will be always happy? And that's one my dream. Okay, so next project. You may say, Dr. Moon, your idea is fantastic about probiotic engineering, but the probiotic you just mentioned is so-called GMO, genetically modified organism. And then how you gonna make sure this GMO, once they do the job in the gut, curing or detecting disease or making you happy and release into the environment, how you make sure this bacteria not doing something harmful to the environment? And that's fair question. And my idea is to develop so-called kill switches that allow this engineered bacteria, I call it genetically engineered bacteria or microbe, GEM, genetically engineered microbes, to self-destruct once they accomplish their mission. In this case, the mission in the gut, curing disease or make you happy. The idea is very simple. The bacteria do whatever mission I just described earlier, and once the bacteria come out of your body, bacteria start to produce some toxic molecule or cleave the DNA. In that way, outside the environment, outside the gut, the bacteria start to die. And this is so-called kill switch. So in one sentence, once mission is accomplished, then they self-destruct for biocontainment. So rather than kind of talk about entire technical detail, we basically use CRISPR system, and I believe you heard about CRISPR system from many years of research. And basically CRISPR is molecular seizure that cleave DNA. And in our case, in contrast to other people basically using this seizure to counter-select after genome engineering, but we actually use this seizure to cleave genome and then kill bacteria. How that works? In many bacteria, CRISPR system actually working, but this CRISPR system, once they cleave the genome of DNA, bacteria typically do not have non-homologous end joining mechanism. That means there's no way to stitching that together if they don't have any homologous DNA you add. But we do not add any DNA homologous to that cleaved region, and cut means bacteria basically die. But as you can imagine, no one wants to die and no bacteria want to die. So I basically showing you the data set in this slide, and I will explain one by one by focusing on the middle figure. The middle figure, the first two bar is basically from control, that basically you can say there is no kill switch. And gray is the no induction, and red is the induction. In this no kill switch control, y-axis is the number of cells, you can say that way, and the level is basically stay the same, 10 to the 8 cell per milliliter. That means control basically do not kill themselves. the rest of the columns basically show uninduced level is around 10 to the 8 cell per ml. But once that kill switch is induced, the level drop all the way to 10 to the 4 cell per ml. And that's a lot of killing. However, US NIH guideline is basically you should have efficiency such that if you start with the 10 to the 8 cell at the beginning, the final number of cells should be only 1 or lower number. And then that means in our case, we didn't meet the criteria NIH actually imposed. As I said, no bacteria want to die, and then bacteria, if they somehow mutate our kill switch, in this case, mostly by deletion of our kill switch machinery, for example, Cas9 gene, bacteria basically survive. And then even if you kill almost all bacteria, if only one bacteria survive, this one bacteria become 2, 2 become 4, and then they basically dominate entire population. So this is a very interesting but very challenging problem because bacteria wanted to survive, but I want them to die. And basically, I also appreciate another student in my group, Austin, who now PhD, after defending beautifully his defense, he basically suffered from this mutation problem for more than 5 years, but he never gave up. And one day in hospital, I just thinking about this problem to help my student Austin, and I realized, what if this hospital lose electricity? Operation could be affected? That was my question. The short answer to that question is that is not the case because hospital always have backup generator. And once they lose the power, the backup generator basically kick in, allow doctor continue to do any operation. So my answer to my problem, why don't we have the more than one kill switch? In that way, if one kill switch not functioning, but the other backup kill switch still working, and at the end of the day, we add total 4 Cas9 gene, and then 3 guide RNA, and we call it redundant kill switch, basically allow us to achieve the guideline NIH imposed. And this is basically the data we tested using real world scenario. But in this case, we use the mice. So basically we feed our probiotic, and then mice basically take this probiotic, and then green and gray is basically showing control total number of probiotic. The number doesn't decrease, but once we have the kill switch, that's a red case, red dot, we now see complete elimination after 48 hours, and then there's no survivor in this case. For this experiment, we use so-called ATC, that is the N-hydrotetracycline, that actually not found in the gut, so we can specifically kill our bacteria. If our bacteria do something wrong in the gut or become pathogen, we want to only target this bacteria, somehow become bad guy. and then we just need to add the ATC, and then we can kill this bad bacteria, or become bad. The bacteria become bad bacteria, and we can kill those things. But you may say this is not real biocontainment, because I talk about once bacteria come out of your body, bacteria should start to die. So to do so, we basically use so-called temperature sensor we developed. And in this case, in your body, the body temperature is typically 37 Celsius plus or minus 2, depending on you have fever or some cold condition. But outside condition is typically lower than 30 degree, even hot summer, but at least at night in Africa or some hot weather country, at night the temperature sometimes go down below 30 or around 30. And then my kill switch now depend on temperature. So bacteria inside your gut, bacteria still do not activate the kill switch. But once bacteria come out of your body, bacteria start to generate the killing agent molecule, Cas9 system, and then start to kill. And then we demonstrate using the same mouse model. And then we basically demonstrate there is no survivor after we treated this bacteria outside condition, that is the temperature at 30 Celsius. So where we are going with this one? So this paper actually published in Nature Communication, and now we wanted to test the same kill switch concept in other bacteria, including some photosynthetic bacteria. And you can think, you may hear about algae bloom that destroy the environment. What we can do is we can use some specific chemical, and if my photosynthetic bacteria which designed to generate biofuel in the open pond, but we can basically control if they become too many cells in the outside open pond that destroy the ecosystem. But in that case, we can add this specific chemical, only kill this increased number of cells in the open pond by using this kill switch concept. We can use the same concept to engineer bacteria that eat plastic in the environment. That is my last topic of today. But once this bacteria eating all the plastic, they also need to die, because that is engineered GEM, genetically engineered microbes. And then to kill those bacteria after they completely consume all the microplastic in the environment, kill switch will be triggered, and then they will die. And then we are finishing up that project, and then we are, hope to, publish some paper in the future. The same thing, if the bacteria clean up toxic chemical in the environment, and then bacteria start to die, then that is kind of our future goal I wanted to implement. So where we are going with this kind of kill switch concept, and also other concept we currently working on. And after 10 years of idea fermentation, I finally published one paper in Trend in Biotechnology. And then now I see soil or entire planet as a huge bioreactor to capture carbon dioxide, of course, by photosynthetic organism. At the same time, provide some nutrient, including fixed nitrogen. In that way, in developing country, they don't have much nitrogen fertilizer that kind of decrease crop yield, but you could rely on this nitrogen fixing bacteria that provide nitrogen source, in that way increase crop yield. At the same time, my bacteria, I will talk about in the minute, that bacteria basically clean up, including plastic waste, and then kill themselves after they clean up. And that is basically, I call it bioremediation application. At the same time, if this bacteria, after eating plastic, if they generate some chemical or materials with higher value, you can also make some chemical that originally coming from petroleum, and then we could say sustainable production. So this is my big vision, or crazy ambitious vision, and this is basically my effort next 20 years. To make this ambitious dream come true, we demonstrated in recent paper in PNAS the potential of our machine learning based computational program to design guide RNA, that's a CRISPR system guide RNA, and to use it for in situ microbiota engineering. The essence is basically something like that. I introduced the concept of kill switch, but now we expand the concept entire microbiota, that's the community of bacteria in the real environment. For example, in the soil, there are some pathogen. I wanted to target that pathogen, but if you add some chemical or antibiotics, all the bacteria will die, almost all bacteria will die. But if we use our kill switch concept and then specifically targeting only pathogen, now we can kill only one bacterium in the environment. And actually we demonstrate using our new targeting weapon, and so using 4 different member community, and then we specifically kill one bacterium. At the same time, we could also target other bacteria except one. In that way, we could easily isolate some useful beneficial bacterium out of multiple community of bacteria. So this is probably very useful in the future, and I'm excited to see more application out of this program. And one another thing I want to mention before I briefly mention about my plastic project, each molecular biology lab or biology lab generate lots of clone or plasmid that contain antibiotic resistant gene. And current practice is basically use bleach or autoclave to kill bacteria, and then we basically allow to release that killed bacteria into the environment through the sink or garbage can. But we do not consider another important neglected fact. That means even autoclaving or adding chemical do not destroy the DNA. The problem is plasmid contain this antibiotic resistant gene, and if that is released into the environment, this gene could be transferred to another bacteria, generating antibiotic resistant super bacteria that cause a lot of problem. To solve this problem, this is the century-old problem nobody tried to solve. We recently developed new way to clone, new way to make the plasmid without any antibiotic resistant gene in the system, and that paper hopefully will be approved and accepted in the spring, and then I hope to see or talk about this one in the near future. Okay, probably next 8 minutes-ish, I will talk about my last topic. That's basically, I call it metabolic engineering project. I envision our future as follows. We engineer microbes that take waste as an input and generate value-added product as an output. One of the critical waste is plastic, and our goal is not such beautiful plastic recycling, but PT upcycling. In fact, I need such an on-site or on-demand manufacturing during my 10 weeks of non-stop seminar trip to give 25 invited talks worldwide last year. But today, I'm talking about my shoe with many holes after 70 days of just walking around the world. And I'm thinking about that plastic shoes. But today I'm talking about this upcycling. Our process is a hybrid process consisting of a hydrolysis step to generate TPA, the monomer, and ethylene glycol, the monomer, another monomer, and then microbial conversion of this into value-added product. We are interested in waste PT upcycling because waste PT represent 12% of global solid waste, and current PT chemical recycling suffers from the relatively high cost for hydrolysis, monopurification, and its repolymerization compared to the virgin PT price that is only $1 per kilogram. In contrast, our process uses a novel microbe that is flexible to potentially produce any desirable products, allowing us to cope with market price fluctuations. Especially, I want to end the current practice of extracting carotenoids from food, such as lycopene from tomatoes and beta-carotene from carrot. Food is something to eat, not something to play, my dad always says. When lycopene is being extracted now, especially some country like China, many people in Africa die every day due to lack of food. So my dream is basically to end this practice by providing cheap processes to solve food inequality, at the same time to solve global plastic waste problem. Let me introduce our novel high salt tolerant rhodococcus strain named Arpad for the first time. Because of time limit, I will just skip, but basically we identify bacteria that eat this 2 monomer as a sole carbon source without adding any other carbon source, and basically bacteria grow very well. And by using some metabolic engineering technique, we basically finally successful using this plastic pellet. That is basically something you just grind the plastic and then make the pellet, and then our develop process basically achieve 1 milligram per liter lycopene production using PT as a sole carbon source. So in summary, for this topic, without showing the more data because that is already published this year in Cell Report, we discovered a novel salt-tolerant microbe that grew very well in this plastic degradation product, and we demonstrate lycopene production up to milligram per liter scale. And of course, we need to increase its titer to make this one commercially viable. At the same time, we use the same computational algorithm-based directed evolution to make mesophilic enzyme that degrade plastic, including PET. In that way, you could even deploy this bacteria that degrade plastic into the smaller molecule and then eating that smaller molecule to generate some high-value chemical, as I introduced earlier. With that, I want to thank all my lab members, more than 20 people at this moment, and my 2 advisors right now at MIT, Chris and Chris, and also 12 funding agency. And if I have some more time, a couple of minutes, I wanted to introduce you the SYMBIS seminar series I started with Professor Francis Arnold, Nobel Prize winner, 2021. And you could watch my YouTube video by typing my name. At the same time, we released Climate Roadmap last year, and then that you could also freely download from ebrc.org. And that is we suggest solution to climate problem using synthetic biology. More detail is kind of here. This seminar series is basically weekly seminar series, Thursday, 10 a.m. US time, Central Time. You can just click Zoom link. I post every week through my LinkedIn account or my Twitter account. And then everyone could just join, or you could watch also YouTube video. And one last thing I want to mention is basically this, slide. This is the Jeff Gordon microbiome father, and I believe he probably will get Nobel Prize next 5 years. And this is the beautiful quote, but I learned this by my experience, but it took me almost 50 years to realize this by heart. This quote touched me so much. And one example is to go fast, go alone. But to go far, go together. That is actually an African proverb that talking about collaboration. In the last 3 years, because of COVID or something else, we have many, many problems, but we do not share. You may break up with your girlfriend or boyfriend, or deaths of family members due to COVID or others. Also, recent earthquake in many different regions of the globe, including Turkey. Also, you may become a victim of harassment or discrimination. This is not the world we wanted to live. This is not the world I wanted to hand over to our next generation. However, I strongly believe we can change this world together by giving our time, resources, attention, support, compassion, kind word, empathy, forgiveness, and of course, sharing our love. This one, I also heard from Dustin, a magician and brilliant storyteller at America Got Talent, a TV show. He basically said, giving is the foundation of change. I want to say, despite of all the difficulty we currently facing, I saw the hope last year when I traveling more than 160 days to give seminars. I saw hope from the people, especially young people, our next generation and next leaders. So I want to say the future is made by young people. That's why I'm willing to and happy to present today in this platform. I'm not a big name, but big name give the vision. However, the real implementation is done by my student, Austin, Chenggang, Jinjin, and so on, Matt. They basically implement. My job is just encourage them, inspire them. That means you young people are true leader in the future, true driver to make this world better. Because of you, I am optimistic and I believe we can change this world together. So if you have some trouble, I just mentioned earlier, never give up and dream high. Nothing is impossible. Even myself, suffering from discrimination and harassment, and that make me suffer, and I literally dying for 4 years, but I miraculously recover, and then now I'm happily sharing my struggle. And if I inspire you in somehow, my life will be fulfilled. And that's one thing I want to mention. Because of time, I just want also probably the one last thing I want to mention. Professor Bob Langer at MIT is the most famous or productive engineer in history. However, he was not accepted by the community when he started his professor position. But he never gave up, and now his technology licensed to more than 300 pharmaceutical companies. He filed more than 1,200 patents, and his paper cited more than almost 400,000 times because he never gave up and he tried to achieve his goal, and he became my role model when I start my PhD program at MIT. And you will be the next Bob Langer. So I want to say that one more time. And with that, I want to thank you for your attention. And I'm just showing this on because I'm forming a company, Moonshot Bio, and I'm happy to answer any question you may have. And thank you so much. And I'm ready to take any question.
Thank you very much, Dr. Moon, for very informative speech and your researches, also opinions for the nature and humanity. I would like to start for the first question is, a multidisciplinary approach combining synthetic and systems biology to study microbial community dynamics open up new possibilities. How can methods be improved to collect higher resolution temporal microbial data sets?
That is very, very great question. So, the short answer is we need to collaborate, because one person cannot solve any problem right now. The global problem is too complicated. So me is only bioengineer. I need to team up with soil expert, for example. If I wanted to solve the problem I lay out, because I do not understand how soil working. Also, I want to collaborate with some people like sequencing of entire soil microbiota, and then from that data I learned. Also, I sometimes need some mathematic genius because sometimes we need to do some modeling. So that's my short answer. I mean, I can give more, but I guess you may have more questions, so I need to just say it that way.
Okay, thank you. One question from YouTube. Is there any limitation of microbial biosensors to health-related applications?
There are many limitations and challenges you should solve the problem. So one limitation would be you have the biosensor, and then the way I kind of see is the probiotic kind of as the biosensor. But as you can imagine, probiotic should respond to that signal quickly enough to generate right signal, because your gut environment is very complex, dynamic, food coming in, go away, and then you should have the right response within reasonable time. And also you need to consider biocontainment. I already demonstrated, because people will be freaked out because that is GMO, and then you don't want GMO released into the environment. So there are multiple different, that's an excellent question.
Thank you very much for your response. Other question is, how feasible is it currently to assess transcriptional and metabolic changes at species resolution in a microbial community?
So that is another great question. Yeah, so that is a very good question. So currently, many people try to understand or assess those kind of change and specific resolution, and I believe the current, the future technology and current technology will solve that problem by using some, spatial resolution technique, like sectioning each gut region, also temporarily kind of doing some sequencing or some metabolomics kind of the technique, and by doing so and combining those technology, I believe they could be solved in the future. I'm very optimistic.
Thank you for your answer. Next question is from YouTube. Can we turn into any type of material to microbial biosensors?
There's no limit. It's just limit is your imagination. So yeah, but you also need to be realistic. So answer is yes.
Thank you. Next question again from YouTube. What can be done for the discovery of microorganisms that use waste as the sole carbon source?
So that's a great question. So what we did for plastic case, we basically have more than 2,000 bacteria variant we collected by using some adaptive evolution and so on for 10 years. And then we basically screen those bacteria to make them eat plastic waste, and then we identify only one. So this is not an easy problem, but you could have some way to discover those things. Actually, there are multiple bacteria actually isolated from environment that eating plastic.
Thank you for your answer. Next question is today's technology enough to change our capacity to tune microbiomes with unprecedented resolution in space and time? What are the shortcomings?
That is another great question. So I already answered that question. I believe we have technology, we're developing technology right now. The current shortcoming is better resolution in terms of location, also in terms of fast response, because that is kind of one challenge we currently have. So, transcription factor-based, response takes many minutes to hour. So you should kind of think about some, other sensor like host population related one that takes seconds, something like that. In that way, you could solve that problem. Spatial resolution is another problem, but there are multiple different ways, including imaging technology and some other technology that basically resolve that kind of problem.
Thank you for your answer. Next question is from YouTube. Given its high processing costs relative to purchase of virgin pets, which recycling strategy can be prominent?
That is another great question. This is $1 million question. If you know the answer, let me know, because I want to make the technology commercialized. As I said, probably I didn't mention that one. So current virgin PT price is around $1 per kilogram, and many companies try to make plastic out of the plastic. So one example is the French company. They did fantastic job making PT out of the PT waste. But the problem is when I calculate the cost of processing, that roughly costs $2 per kilogram, and that doesn't make any sense economically. But of course, environmentally, that's a very important task, and they did great job. That's why I chose lycopene or carotenoid, because the price is up to $7,500 per kilogram. That is the more than 1,000-fold increase in price. So that's my reason intentionally choose that one. But our titer is currently milligram per liter, but we need to improve. But lycopene, many decades of research improving 1,000-fold can be done. So I'm very optimistic, and my student and postdoc are working every day to improve that titer.
Thank you for your answer. Next question is, how far has it come in transforming commercial PET into high-value chemicals?
So as I said, I want that happen tomorrow, but not tomorrow. That's not possible. I would say if we achieve gram per liter scale production, in terms of my lycopene, I believe we could consider commercialization, and I believe that will happen within 5 years. And then after that, I may retire and buy mansion in Hawaii, and then I enjoy my life by drinking cocktail, seeing sunset in Hawaii Island.
We hope it.
Sure. We all hope. Yes.
Next question is from YouTube again. In your study, how did you ensure the stability of engineered and wild-type probiotics population prior to kill switch induction?
That's a great question. So, interestingly, our kill switch is tightly controlled by using a very tightly controlled promoter, and also we use the so-called degradation tag, the protein, and then we actually screen more than several hundred of mutants, and then among that we just chose very tightly controlled kill switch. And then but still we need to think about mutation. So after we achieve that goal in terms of stability, we typically have the mutation. So we decide to do long-term stability study. And in fact, my student Austin come to office and lab every day for 4 weeks. And then we tested stability, and then the stability working beautifully work. And then after 4 weeks, he tired, and he just contact me saying, "Oh, professor, I want to just take off." And then I didn't realize at that time he already done 4 weeks of experiment, because typically in the probiotic, they only stay in the gut roughly several days, but he already done 4 weeks of experiment. So I said, "Oh, you should take off." I mean, you need to take a break. And I didn't know, and then we have enough. But if you're thinking about stability in the real environment after bacteria eating all the plastic, you should have probably more than several weeks, because we don't know how many days it takes to consume all the plastic in the environment. But right now, we tested 4 weeks, but we could test more.
Thank you very much for your answer. Next question is again from YouTube. How can we use C. testosterone and other non-model species for biological upcycling or remediation of recalcitrant aromatic compounds? Does their cellular physiology know?
So the specific organism you mentioned, I believe there is one paper published, and then you could just read that paper. So that organism basically consumes some aromatic compound. And then Rhodococcus, I worked on more than 10 years. They also very, very tolerant to that toxic chemical, but still, you need to kind of engineer. Actually, we use the adaptive evolution to make those bacteria tolerate and utilize more. So there are multiple different ways, but probably the most feasible way is using the adaptive evolution. And then to understand cellular physiology, you need to do sequencing, you need to do some phenotype analysis. So to do so, we basically do RNA sequencing, DNA sequencing, at the same time, lipid, analysis and so on. And that takes 10 years. But I believe with the current technology, the total time will be shorter, like 5 years or even 3 years, something like that.
Thank you very much for your answer. Next question is again from YouTube. We already heard that T lymphocytes were modified as a part of CAR-T immunotherapy. Unlike cells, are there any way to construct a microbe for cancer treatments?
That's a fantastic question. So, I'm not expert in that area, but there are many studies for more than 20 years, basically using bacteria that kill cancer. So one of the problem in cancer therapy is, the chemical drug cannot, or very difficult to specifically only kill the cancer cell. But another problem is basically those chemicals attack the normal cell, healthy cell. So important thing is the specificity. But cancer cell has some specific marker, including oxygen level is very low, also lactate level is high. So people actually use some sensor that detect low oxygen or high lactate, and then use a bacteria, and bacteria basically gather around only cancer cell, and then inject some toxin, and then kill the cancer cell, and that is actually demonstrated multiple times in the literature. So that's possible. Yes.
Next question is there any synthetic biology approaches for antibiotic resistance microbes or bacteria?
So there are also multiple, example. So, multiple antibiotic resistant bacteria is huge problem in society. And that's why, many pharmaceutical companies, stop developing antibiotics. So one of the example I show you today is basically we use those CRISPR system that completely just cleave the genome. In that way, you could kill those very difficult to kill bacteria, the pathogen. But still, the problem is how to deliver those CRISPR system into this pathogen. But we develop liposome system that basically fuse with any bacterium, because bacterium has some lipid system, and that lipid kind of friendly each other. and then they kind of fuse into, and then we could kind of deliver. But the key problem is the efficiency of this delivery system, because we will be limited by the efficiency. So that means we cannot kill all the bacteria in the system. But there are multiple different ways you could think of. Other people also use phage, that's a bacteria virus that kind of target those kind of pathogen and then kill only that bacteria specifically. So there are multiple different ways, but many people working on. So let me put it this way. I'm just one of the bioengineer. I cannot solve all the problem. That's why I need your help. You need to be scientist and engineer to solve all the problem together. I'm just one of them. I'm not the only one to solve that problem.
I agree. Thank you. Thank you for your answer. It was our last question. On behalf of myself and all our participants, I would like to thank you for spending your valuable time transferring your knowledge to us and for this very informative presentation.
So what is question again? I didn't
It was our last question. I said that on behalf of myself and all our participants, I would like to thank you for spending your valuable time transferring your knowledge to us for this informative presentation.
Okay, it's my honor.
We all thank you.
Honor. It's my great honor to be here. And again, I have the one daughter, and I want to make this world, better place for my daughter. At the same time, all of us. And because, as I said earlier, we suffer a lot, especially last 3 years. And also, I believe many people in Turkey suffer a lot. But I see hope because we have younger generation. And younger generation are always passionate to solve all the global problem. And then you will make our future better. And then I live my old age period thanks to you. So please make all the problem go away and then make the world better place to live, regardless of your problem you currently have.
Yes. Hope which makes we are alive. So we need to hope always.
Sure.
Thank you again.
Sure.
I wish you healthy days. Bye-bye.
Thank you so much.
It's 1915. Dr. Gülçin Pekurnaz will be with us to give a speech on metabolic and mitochondrial plasticity in neurons. Thank you.
Bioinfocongress IV. Assistant Professor Dr. Gülçin Pekurnaz, Metabolic and Mitochondrial Plasticity in Neurons. Dr. Gülçin Pekurnaz is an Assistant Professor of Neurobiology at the University of California, San Diego. Dr. Pekurnaz received her undergraduate degree from Ankara University. She completed her PhD training at the National Institute of Health. She pursued her postdoctoral studies in the Department of Neurobiology at Harvard Medical School. Her research focuses on the molecular principles of metabolic homeostasis in the nervous system. By studying the interplay between neuronal metabolism and mitochondrial functions across cell classes, she reveals fundamental insights into the mechanisms that regulate cellular bioenergetics and pinpoint the underlying causes of energy impairments that lead to neurological diseases.
Dear Assistant Professor Dr. Gülçin Pekkurnaz and participants, welcome all of you to our Bioinfocongress IV event. Dr. Pekkurnaz will give us a talk today on metabolic and mitochondrial plasticity in neurons. At the end of the presentation, we will have a question-answer section prepared by our team and consisting of questions from our participants. Dear Dr. Pekkurnaz, welcome back. We're so glad to have you here.
Thank you. Thank you for, I'm trying to figure out all my windows here, but thank you for having me here. And it's a great honor. I wanted to say that this is your weekend. It's already Sunday evening there. Thank you all for organizing this and spending your time on the weekend to bring all this amazing science together. We definitely needed this, especially in this stressful day. So you're giving all of us hope, and I'm really excited to be here.
Could you go to full screen?
Yes.
You can?
Good
Thank
now?
you so much. Yes.
Okay.
Thank you so much.
I will just get started. I'm going to talk about metabolic and mitochondrial plasticity in neurons, and as you've all heard, I'm an assistant professor at University of California, San Diego. And the main focus of my lab is to really understand the molecular mechanisms that provide energy in neurons and then sustain neuronal health by continuously providing this energy. And this is important because most of the cells, their energy demand does not fluctuate as much as neurons. And neurons, especially here in this picture showing the synapse, it's really high and energy demanding as neurons go through the activity cycle. And when we say energy homeostasis, we also have to think about many different concepts. Not only when we say energy, we're talking about ATP, which is the currency of energy in cells, but we're also talking about this local regulation of energy delivery through the mitochondria. And that requires mitochondrial localization coordination, and that requires mitochondrial function coordination, and then even the recycling and then constantly providing the healthy mitochondrial pool for the synapse. And we're trying to really understand the plasticity mechanisms of mitochondria and energy homeostasis pathways. So why this is important, I will give a really extreme example from these dopamine neurons. These are the neuron subclass that degenerate in Parkinson's disease. And so in the human brain, this is a single neuron. In the human brain, the size of this neuron has been estimated to be about 4.5 meters. And right as this brain region that's called striatum, there are about 1 to 2 million synapses. And if you think about each synapse consumes approximately 1 million ATP, and this ATP demand is within milliseconds, you can imagine how demanding process this for this one single neuron. And then to provide ATP exactly where it's needed in this complex neuronal site architecture, all these metabolic pathways has to be coordinated. And at the center, you see the cycle. This is the TCA cycle that actually occurs inside the mitochondrion. But all these other metabolic pathways has to coordinate and then play a role in converting nutrients to ATP and other metabolites that is needed for the function of neuron. And we know all these reactions, all these metabolic reactions from textbooks, and you all learn in the biochemistry, but we don't understand in neurons where are these reactions are actually happening in neuron. How do these enzymes find each other within 4.5 meter distance in a single cell? And we're trying to really figure out the spatiotemporal regulation of these metabolic pathways in neurons. So just to add more complication, now if we could zoom in these neuronal axons and then look at the mitochondria, so this image is acquired from cultured neurons, and we culture them in microfluidic devices, so you can imagine all these axons are clustered together, and every red dot you see here actually is mitochondrion. So now I'm going to play this movie, and you can see some mitochondrion, they are stationary. They stay in one spot as if they reach their final destination. Whereas if you look at the others, they move as if they know where they're going, and we're trying to figure out how these reactions are coordinated with the positioning of mitochondria and also the motility of mitochondria. And of course, these are very complex cellular processes and reactions, and there are many diseases associated with the defects in these processes that ranges from Parkinson's disease, the stroke, and epilepsy, and even psychiatric symptoms are now associated with mitochondrial and metabolic defects. Another thing that I want to highlight here, even though most of the mitochondrial proteins are encoded for nuclear genome, a single point mutation of one of these proteins can cause mitochondrial dysfunction, but cell type specific phenotype is very different from neuron type to neuron type, and that's another topic in my lab. We're trying to understand how different neuron subtype utilize different pathways in neurons, and I'll try to talk about that a little bit. So now I'm going to dive into one of the projects in our lab, and I'd like to highlight the name of my lab members who primarily involved in this work, and we're going to talk about spatiotemporal coupling of neuronal metabolism and mitochondria. And this most of the work is done by really talented graduate students Howard Wang and Blind Yu in the lab with the amazing support of the all lab members. So what do I mean by spatiotemporal coupling? So now this is more accurate demonstration of a neuron. So here you see neuronal soma, and then long axon. And then if we could look at the soma, mitochondrial distribution in soma is very continuous, and they form this network and that expands in dendrites. But then if you look at the axon, as you've seen in the previous movie, you see individual mitochondria, and then this distribution is pretty much accurate throughout the axon, even though density changes a little bit. So now this is for mitochondria. But for mitochondria to provide ATP, mitochondria utilizes nutrients. And major fuel for mitochondria is glucose in the brain. And we know this because if you deprive brain from glucose, it causes confusion, seizure, coma. And on the extreme side, if glucose level is really high, it can go all up to neuropathy or pain and seizure. So how do glucose and mitochondria actually find each other and talk and communicate? How this glucose metabolism can happen in neurons? Because just to remind you, for mitochondria to start ATP synthesis, the metabolite is pyruvate. So pyruvates get transferred into the mitochondria after glucose is metabolized through glycolysis. So we decided to investigate this coupling reaction in neurons. And the typical textbook knowledge for this, glycolysis happens in the cytosol, and it provides small amount of ATP, and then the pyruvate, which is the end product of glycolysis, gets generated in the cytosol, gets transported in the mitochondrion, and then that initiates the basically oxidative phosphorylation and citric acid cycle, and then that's how mitochondria generates ATP. But now, if we start thinking about the initial reactions of glycolysis, the first rate limiting step enzyme is called hexokinase. And hexokinase consumes one ATP and converts glucose to glucose 6-phosphate. And that's how glucose can be trapped in the cytoplasm. If it's not phosphorylated, glucose actually leaves the cytoplasm. So one of the first questions that we ask, where is this first rate limiting step enzyme of glycolysis in neurons? And it's quite fascinating, and this is not our finding actually. If you look at even like early literature from 80s, there is already description that hexokinase 1, which is the neuronal isoform of hexokinase, is mainly localized on mitochondria. Here you can see the endogenous staining of hexokinase 1 and mitochondria. This is neuronal soma and dendrites, and you can see the zoomed in image, and also in axons. So under baseline conditions, it's been reported previously that about 70% of this first rate limiting step enzyme of glycolysis is mainly localized on mitochondria. And another important fact that if this hexokinase localization and activity is been known, and activity is another measure that is being mainly used in clinic, that if we inject labeled glucose in patients, and you can basically monitor brain activity, which basically hexokinase activity. And we know that in cognitive impairment and Alzheimer's disease, the brain glucose metabolism declines, which is imaged by hexokinase activity. And then also, we know that as we go through our regular daily cycle, blood glucose level changes, and also brain extracellular glucose level changes. Let's say if you haven't had anything for a long time, and if you eat really sugar-heavy diet, we can see up to 4-fold change in brain and blood glucose level. So we decided to ask question. So as we vary glucose level, what happens to hexokinase? Is there any change in hexokinase positioning? Is it always mitochondrial, or do we see any alterations? And then we decided to investigate this in primary cultured neurons. We take these neurons from embryonic rat brains and dissociate them. These are active firing neuronal networks. And then we culture them in physiological glucose levels, which is ranging from 1 to 5. So if you look here, this is the now hexokinase staining pattern. When we culture these neurons in lower glucose level, 1 millimolar, we see more some mitochondrial but more cytoplasmic hexokinase distribution. But then if we give a glucose pulse which mimics the postprandial glucose rise, and as if you just ate a really glucose-heavy meal, well, we see hexokinase positioning changes from cytoplasm to mitochondria, which is also quantified here. This is an indication that first rate limiting step glycolysis enzyme hexokinase positioning actually is sensitive to glucose level in neurons. And now here's with live cell imaging, we can also capture this behavior. You can see now this is mitochondrial hexokinase 1, and this is cytoplasmic. I didn't include here to the mitochondrial marker to simplify, but this area is a mitochondrial area. So I hope you can appreciate that we give the glucose pulse. You can see the cytoplasmic proportion is going down, but the mitochondrial proportion is going up. So this is the live cell capture of that behavior. And we can see the same thing even in vivo in the mouse brain. We go through these feeding fasting cycle with mice. So we either fast them for 24 hours or keep them on their normal diet. And then after fasting, we refeed these animals. And what you can see, this is a brain region called hippocampus. In the hippocampal area, when we see mitochondria, when we stain mitochondria, it's really dense, a lot of punctate staining, because brain actually is really many neurons have mitochondria and really high density. So you can see this staining pattern. And then I hope you can appreciate here that in the fasted brain, hexokinase staining is more diffuse. But then when we refeed the animals, glucose level go up, and then that the hexokinase becomes more enriched on mitochondria again, even in vivo. So overall, what I've shown you so far, this pathway that we think it is cytoplasmic, it's actually not that simple in neurons. In fact, this first rate limiting step enzyme for glycolysis, hexokinase 1, the neuronal isoform, it shuffles back and forth from cytoplasm to mitochondria based on the glucose uptake and glucose fluctuation in neurons. But we asked two questions after this. One, how do hexokinase can actually sense glucose availability and make this decision whether to be on mitochondria or in cytoplasm? And two, what is this good for? Why do neurons utilize such a pathway? What kind of advantage this is going to provide for neuronal metabolism and neuronal health? So to really go into the details of the answers, I want to introduce you a new pathway that's called hexosamine biosynthetic pathway. So whenever cells take up glucose, most of this glucose is being used for ATP synthesis through this pathway called glycolysis that I've been talking about. But then some portion of glucose goes through another pathway. It's called hexosamine biosynthetic pathway. And then the end product of that pathway is UDP glucanag. And UDP glucanag is being used as a substrate for this metabolic sensor enzyme called O-glucanag transferase. And what O-glucanag transferase does, it uses this glucanag moiety and post-translationally modifies serine and threonine residues of the proteins. And this modification, you can think about that through these pathways, glucose can be used as a signaling molecule, and through this pathway, glucose level can be transferred as an information to all these proteins that are being modified through O-glucanag transferase. Therefore, this pathway and O-glucanag transferase is considered as a key glucose sensor pathway, and OGT is the key glucose sensor enzyme. So another important fact, many of you may be more familiar about kinases and phosphatases, and this is the phosphorylation on serine and threonine residues of proteins. There are over 500 kinases and 100 phosphatases. So OGT is encoded by single gene, and the enzyme that removes this O-glucanag-related moieties, OG, is also encoded by single gene. And this enzyme pair post-translationally modify over 8,000 substrate proteins. So it's really, really heavily utilized in cells. And this is how, again, the nutrient flux is being informed at many different levels. And OGT is highly expressed in the nervous system, and many synaptic proteins are being O-glucanag modified, and OGT mutations actually cause intellectual disability. And if you delete OGT neurons, it causes early postnatal mortality. And even the acute deletion of OGT for a couple of weeks actually leads neurons to lose its ability to sense nutrient flux, and these animals become obese from overeating. So previously, as a postdoctoral fellow in Tom Schwarz's lab, I've shown that this enzyme also guides mitochondria to glucose-rich areas by post-translationally modifying this motor adapter protein called Milton. And then this basically creates the stop signal for mitochondria. And if we create O-glucanag resistant Milton, and mitochondria loses its ability to sense glucose level, I'm going to show here when there's glucose activity and OGT activity goes up and mitochondria becomes stationary. But then when we create O-glucanag resistant Milton, so mitochondria basically loses its guidance about nutrient availability and it just starts becoming motile. So knowing this, we hypothesized that what if this master metabolic sensing mechanism couples mitochondrial localization and glycolytic enzyme localization on mitochondria for metabolic efficiency in neurons. And we decided to test that hypothesis by investigating hexokinase 1 localization. And we've done the experiment that basically stops mitochondrial motility and also upregulates O-glucanylation. And what we've seen, even though glucose level was the same, hexokinase became more enriched on mitochondria when O-glucanylation was upregulated. And on the opposite side, when we downregulate this O-glucanylation, what we see is more cytoplasmic hexokinase 1. This suggests that this metabolic sensor enzyme, OGT activity, indeed can compartmentalize hexokinase 1 on mitochondria. We can reconstitute this phenotype in other cell types, such as HEG cells. In HEG cells, if you look at hexokinase, it's mostly cytoplasmic. There's some mitochondrial pool, but then the moment you upregulate O-glucanylation, you see this hexokinase almost gets sucked into mitochondrial outer membrane. So this suggests that hexokinase actually, localization is regulated by O-glucanylation, but how? So we hypothesized that perhaps O-glucanag moieties are being added to hexokinase through serine and threonine modification. And in immunoprecipitated hexokinase 1, you can see the immunoprecipitation. And then when we upregulate oligonucleation, either genetically or pharmacologically, and you can see the increase in total oligonucleation of hexokinase 1. This is the condition that we see more mitochondrial hexokinase in cells, also quantified here. So these immunoprecipitation experiments indicate that indeed hexokinase 1 can be directly post-translationally modified via OGT. And we went ahead, we did mass spectrometry, post-translation modification mapping, and I'm just going to show some final conclusion here that we identified one threonine residue that is being post-translation modified via oligonucleation on hexokinase 1. And I have to say, we looked at other hexokinases. This regulation and this modification is very unique to neuronal isoform hexokinase 1. And now when we mutate this threonine residue to alanine, we can generate oligonucleation-resistant hexokinase 1, meaning the recycling of this oligonucleation is abolished. Here you can see the increase with enhanced oligonucleation, but then in the mutant version, this increase was not observed, which is also quantified here. And now if we go and now look at the positioning of this hexokinase, and I hope you remember that under this condition, wild-type hexokinase was actually mostly mitochondrial, but now when we generate a version that is resistant to oligonucleation, it becomes mostly cytoplasmic. That was in hex cells. We see the same thing in neurons. So basically, hexokinase loses its ability to change its localization based on the oligonucleotide flux. So, which is also quantified here. What was also interesting, I'm not going to go through all the biochemistry here, but when we see hexokinase being compartmentalized on mitochondria, we see other glycolytic enzymes are also being recruited to mitochondria. And here is a subsample of them, aldolase and PGK and PKM. And you can see this is not due to change in overall level. We measure that. So it's really the transfer from cytoplasm to mitochondria. So it's almost this signal kind of brings all the glycolytic machinery on the mitochondria surface. And then when we use the mutant hexokinase, this glycolytic enrichment was also abolished. So how about the function? So far, I've shown you that hexokinase can be localized on mitochondria, and this is through this oligonucleation pathway. And then this brings other glycolytic enzymes to the mitochondrial surface. But then what happens to hexokinase function and overall neuronal metabolism? So first, when we actually upregulate oligonucleated hexokinase 1 and bring hexokinase to mitochondrial surface, overall activity of hexokinase is high. So we see more glucose 6-phosphate synthesis, which is abolished in the oligonucleation-resistant version. And now when we measure ATP production rate and we use metabolic flux assays for this and overall measure mitochondrial oxygen consumption rate and extracellular acidification to calculate glycolytic ATP production rate, and we calculate how much ATP is being generated in cells. So now this is just the baseline ATP synthesis rate, some glycolytic, some mitochondrial, and then if you increase oligonucleation, you see increase both in glycolytic and ATP and mitochondrial ATP production. So now when we express GFP tag hexokinase, and then do the same experiment either with wild type or oligonucleation resistant, what we see wild type hexokinase, when it gets enriched on the mitochondrial surface, both glycolysis and mitochondrial ATP synthesis rate goes up, whereas this hexokinase glucnac mutant that loses its ability to basically go back and forth between cytoplasm completely becomes insensitive, and then ATP production rate remains the same. So overall, we can say that this hexokinase enrichment and then hexokinase ability to bring other glycolytic enzymes to mitochondrial surface is critical for enhanced ATP synthesis rate. But how is that one post-translational modification can do all of that? So we collaborated with a structural biologist, Abhishek Singhoroy's lab, and what he helped us to do, localize this oligoconnect modification on the N-terminal regulatory domain of hexokinase 1, which is shown here. And this oligoconnect modification sits right where glucose and glucose 6-phosphate binding pocket. And when hexokinase is modified, this groove gets enlarged and it becomes more wider, and that changes its affinity to glucose 6-phosphate, and hexokinase ability to be inhibited by its substrate, glucose 6-phosphate. And at the same time, it causes a little bit of a tilt on hexokinase structure, and then that allows more contiguous electrostatic surface for hexokinase to be attached to mitochondrial outer membrane. So basically, it has more favorable structure for mitochondrial binding, and then it becomes more resistant to dissociation with its product G6P. We decided to also confirm these structural findings using biochemical assays, and what we have done, basically, we purified mitochondria and start treating this mitochondria with different amount of G6P, its product, to see how much is going to be coming off of mitochondria. And you can see as the G6P level goes up, more hexokinase gets dissociated from mitochondria, which we collect this as a soup. And you can see when hexokinase is oligoconnect modified, it's more resistant. So it basically is attached to mitochondria very strongly. But then oligoconnect mutant is completely losing this attachment. So this basically also confirms our phenotypic assays and also morphological assays and the structural evidence. So overall, this is great, but can we phenocopy it? So what we decided to do, we use this chemically induced dimerization system. So what we can do, doesn't matter if hexokinase is oligoconnected or not, or whatever the glucose level, we can always decide where hexokinase is going to go. And we use this FRB and FKRB domains, and then we can add the dimerizer to basically change hexokinase positioning. So this is how it looks like. So this is just a cytoplasmic truncated hexokinase 1. But then now when we add chimerizer in the FRB domain tag, you can see it's becoming really, really enriched on mitochondria here after addition of dimerizer. So now when we do this experiment and measure ATP production rate, so this is the wild type hexokinase 1, you can see that we can boost the ATP production rate the moment we bring hexokinase to mitochondrial outer membrane. But then even the oligoconnect mutant one, if we push that to mitochondrial outer membrane, we can basically get that ATP synthesis rate upregulation. So it really suggests that this localization and regulation through oligoconnectylation is really has this metabolic boosting ability. And when we force even the oligoconnect mutant hexokinase, it gets recruited to mitochondrial surface. So then what is it good for? So in neurons, this is a presynaptic terminal where many ATP demanding processes are happening, especially the synaptic vesicle recycling. And in this presynaptic terminal, there is already evidence that when action potential firing is triggered, glucose transporters can be incorporated. There's evidence of glycolytic metabolome formation. But now what we're bringing here, the coordination with mitochondria. So we decided to see if the process that we discovered can be important for the function of presynaptic nerve terminals in neurons. And this is important again because when neuron goes through this intense activity periods, which is also clearly shown here with genetically encoded sensors. So if you stimulate neuron, calcium goes up, and then that increases glucose consumption, which is shown here with the glucose sensor or another probe that can measure the glucose extracellular level in the earlier studies. And well, we also know that if we stimulate neuron, same stimulation also increases overall oligonucleation level in neurons. And if you think about the synaptoenergetics, it's really nicely summarized by this review in Zhongchuan's lab. This fast energy recovery is really important at the presynaptic terminal, and if there's failure in that, basically it may lead to neuronal dysfunction. And so what we see basically, if you stimulate neurons, the ATP level in a healthy neuron always goes back up, and that because of this immediate ATP synthesis machinery. So we've done the same experiments. We stimulate neuron, measure ATP level using an ATP sensor, and what you can see, there's a little dip, but it immediately goes back up. And we can inhibit glycolysis and oxidative phosphorylation, then ATP continuously decline and there's no recovery. But now when we inhibit oligonucleation using OSMI-4, that recovery is also being perturbed, which is quantified here. So which suggests that oligonucleotide cycling is important for this on-demand ATP synthesis. So now how about vesicle recycling, which is the mechanism that uses this ATP? So what we did the experiments with hexokinase that can sense glucose and they can be oligonucleated or not oligonucleated. And then we express these hexokinase versions in neurons and visualize vesicle recycling using this V-glut pH sensitive presynaptic terminal marker. And the idea is you can mark the presynaptic terminals if you stimulate neuron using these sensor. And here's what it looks like. So if you look at the vesicle recycling, you stimulate neuron, there's exocytosis, and then that if in a healthy terminal, if ATP synthesis rate is accurate, then that's coupled with endocytosis and you see decline of the signal. But then if you have the mutant hexokinase 1 that is resistant to this metabolic sensitivity and ATP synthesis rate is perturbed, what we see is change in synaptic vesicle recycling rate, which is also quantified here. We can also change the acidity of these vesicles and mark the presynaptic terminals, and I hope you can see here, presynaptic terminals are there. It's just the vesicle recycling is not happening the way it happens in neurons expressing the wild-type hexokinase, which suggests that indeed this glycolytic compartmentalization is really important for the function of neurons, especially at these presynaptic terminals where metabolic efficiency is really, really key. So overall, what I've shown you so far, a new mechanism that allows glycolytic enzyme compartmentalization on mitochondrial outer membrane through post-translation modification called oligonucleation. So when glucose level is up, when there's increase in glucose flux, and then when that also is always coupled with high ATP demand in neurons, this hexokinase 1 gets modified and then gets trapped on the mitochondrial outer membrane, and then that brings other glycolytic enzymes to mitochondrial surface, which literally boosts up ATP synthesis rate to fuel neuronal metabolic efficiency. And this is only part of the equation. I've shown the glycolysis part, but we have also mapped the mitochondrial glucanacome in neurons, and we know there are other mitochondrial proteins being modified through this post-translation modification. That also explains why oxidative phosphorylation is also going up. And all of these candidates will have stories of our own, and it's taking us to really interesting directions. So stay tuned for new stories. And overall, what we think is that glucose sensing pathway not only regulates mitochondrial positioning, but also coordinates glycolysis together with mitochondrial localization and mitochondrial oligonucleation to almost kind of synchronize and bring mitochondria and glycolysis into this level of efficiency to really support activity-dependent energy demand in neurons. And we also think that this pathway is involved in nutrient sensing, whether you have glucose as the nutrients or alternative fuels that can be also, this information can be also transferred through this pathway. So within the next 2 minutes, I'm going to focus on the second half of the lab, which is how we investigate neuron type specific regulation of mitochondria. The lab members involved in this project is also highlighted here. Dan Evans, Ahmet, especially Ahmet is currently carrying out this work. He's a PhD student and former lab members. So we know that mitochondria, not all mitochondria are the same. And we can see this across different tissues in their morphology and also in their proteome. And then in neurons, we also evidence that, especially in disease models for dopamine neuron mitochondria, this is again the neuron type that degenerates in Parkinson's disease, even though mutations causing Parkinson's disease are mutant in every single neuron subclass, it causes dysfunction severely in dopamine neurons first. So we ask the question, what makes these neurons more vulnerable? Or you can ask the question, what makes these neurons, other neurons, more resilient? So what we do, we basically genetically label these neurons, investigate their morphological changes, and then try to figure out how they transition from healthy neuron to degenerating dopamine neuron. So I'm going to show you in vivo morphology of the mitochondria in these axons. You can see their axons are packed with mitochondria in the axon terminals. But now if we look at the proximal area, mitochondrial morphology is very different. You see more elongated mitochondria, it's more frequent. And now we're mapping this as the neuron basically start to degenerate, and we're seeing really, really fascinating local adaptations at the morphological level. We're also following this morphological adaptations using cell type specific mitochondrial proteomics, comparing other neuron subclasses to dopamine neuron mitochondria. Here is one of our data set, and you can see we can confirm that we can actually get cell type specific mitochondrial proteome. This is the dopamine neuron specific mitochondrial protein, that's our positive control. So overall, what we're trying to really figure out mechanisms that sustain energy homeostasis in neurons, but our approach has always been looking at these things at the individual pathway level, taking these pathways at the mitochondrial morphology and connecting with the dynamics, and then taking these studies the whole in vivo brain and circuit level to really reveal mechanisms that can actually lead to neurodegenerative diseases, and we're basically continuing on many different directions. With that, I'm going to end my talk by thanking my amazing lab members. So I'm very fortunate to have very talented PhD students, undergraduate researchers, and also postdocs. And also it requires a big team, so we have really good collaborators. I already highlighted Abhishek Simbaro's lab with the structural work, and we collaborate with UCSD's mass spectrometry facility. I haven't had a chance to talk about these other collaborations, but where we have very exciting data in many different avenues, and of course funding. And with that, I'm going to end my talk. And if you have any questions, this is my email address. So I'm going to transition to Turkish here. Özellikle öğrenciler, Türkiye'deki öğrenciler, eğer sorularınız varsa, başvurularınız konusunda yardıma ihtiyacınız varsa, eğer başka bir gözün onları okumasını, değerlendirmesini, size yorum yapmasını istiyorsanız lütfen bana e-mail atın. Elimden geldiğince yardım etmeye çalışacağım. And I would like to thank all the organizers again for giving me an opportunity here to share our work. Thank you, and I'll be able to take questions.
Thank you so much for this incredible presentation, Dr. Pekkurnaz. I am directing our first question. Could targeting mitochondrial metabolism one day be considered in the treatment of certain developmental disorders that may result from brain development that is too fast or too slow. What do you think is the possibility?
I do think that there could be certainly some therapy options. And in fact, for one of the neurodevelopmental disorders, we are starting to get into that field a little bit. I cannot just say it's going to be potentially a full recovery, but at least if we understand what pathways are failing, metabolism is very plastic, it's very flexible. There is always compensatory pathway. If we can really understand how we can rewire things, perhaps we can boost one pathway and then kind of reduce the demand for the another pathway to really enhance the function of a neuron. And I think we need to do more work to really get into that.
Thank you for your response. I am moving on with our next question. Why a human neuron takes years to grow when a mouse neuron grows? Does mitochondria's metabolism affect this situation?
Absolutely. And in fact, there's a really beautiful recent study showing that. And it's because as you increase the neuronal size, the metabolic demand is really, really high. And first you have to build on the anabolic processes, growing proteins and membranes and so on, and then sustaining that. And all this requires really tight metabolic adaptation, and mitochondrial metabolism definitely affects this. So yes.
Thank you for your answer. I'm moving on with our next question. Mitochondrial activity is crucial for the plasticity of central synapses. So, do we know how the firing pattern of pre- and postsynaptic neurons affects the mitochondria?
That's a very good question, and it's always complex to answer. So, one, mitochondrial activity is really crucial. And if you perturb mitochondrial activity, for example, on the presynaptic side, there are also really great studies showing, for example, if it affects the presynaptic consistency, for example. And then another thing, I will give you an example. We have a new work that will be published later, hopefully this year or early next year, that, for example, in fast-spiking neurons, if you modulate mitochondria, the firing pattern of these fast-spiking neurons are changing, and that's affecting the whole brain circuit and even brain oscillation. So that suggests that there is really close collaboration between mitochondria and neuronal activity. And another way that we always wonder, and it's important to think about, if mitochondria is dysfunctional, what's going to happen to neuron? What neuron is going to give up? Is that going to be firing or something else? Because you only can have so much ATP, and if you start losing that, almost like you have so much money in your pocket, and if you don't have enough money, what do you give up first, right? And I think neurons are making that decision constantly as we continue our life. And I think that's really important for neurodegeneration too.
Thank you again for your answer. Moving on to the next question. This one is from YouTube. Can neuronal activity drive an all-glycolyl
All-glycoylation, that's the hardest thing to say.
Yeah.
Translation.
Be a potential way to synchronize multi-organal functions based on fuel availability in neurons?
That's a really, really good question. And short answer, I think yes. We have one work in process at the moment, suggesting indeed that may be the case. And I think if you think about activity brings neurons to a different state at so many levels, and how important these multi-organal functions and interorganal connections, I think it only makes sense to use such a mechanism that can coordinate things, because we're talking about like a meter-long cell. So how do you coordinate everything? And I think all-glycoylation, especially activity-driven regulation of all-glycoylation, could be really a potential way. But we have to do more work, but stay tuned.
Thank you for your answer. Another question from YouTube. Mitochondrial sensor enzyme all-glycinate transferase can regulate to the mitochondrial bioenergy. With this information, is it possible to reducing fuel consumption for highly rate metabolisms used energy?
I think depending on which mitochondrial protein is being oligonucleotide modified, perhaps you can use this as almost like turn on off buttons and then switch fuels. We have one work, again, I actually need new student to follow up on that with ketone body. For example, if you switch from glucose to ketone body metabolism, the enzymes required for the utilization of different fuels are different. And when we think that oligonucleation is actually kind of fine-tuning these enzyme usage based on the fuel availability. And can you kind of tune up and tune down? That would be dream. For example, especially in Alzheimer's disease, it's known that oligonucleation level goes down because brain glucose metabolism goes down. Can we use this trick to almost change the mitochondrial activity level, even though glucose metabolism is low? And that would be a really good way to kind of support the high energy demand. But again, these are really new directions, so we have more work to do to start utilizing the pathway.
Thank you again for your answer. I'm continuing on with another YouTube question. The downstream mechanisms of mitochondrial calcium plus 2 uptake explored in neurons?
Short answer is yes. And in fact, neuronal activity-dependent calcium uptake and then following mitochondrial calcium uptake is one of the first mechanism that's been linked to enhanced ATP synthesis. So we know the calcium channels on the mitochondria membrane. We know partially how calcium can upregulate ATP synthesis rate. And what we don't know, for example, how oligonucleation, this calcium and non-calcium dependent pathways can work together. How do they support each other? But yeah, so short answer is yes. Not every detail, but the main key players are known.
Thank you for your answer. Another YouTube question. Which pathways allow mitochondrial metabolism to be tailored based on nutrient availability?
In terms of nutrient availability, I think oligonucleation and OGT and hexosamine biosynthetic pathway is one of the key pathway. But in addition to that, I think there are other kinases that's been known to sense, for example, AMPK can also sense overall ATP availability, and that's an indirect measure of nutrient availability. But what is so unique about hexosamine biosynthetic pathway, it actually can really sense glucose, amino acid, fatty acid, overall availability through the feedback through this hexosamine biosynthetic pathway. So I do think that it's one of the key pathway that allows mitochondrial metabolism to tailor to nutrient availability.
Thank you again for your response. Continuing on with another YouTube question. We can match mitochondrial dysfunction to neurodegeneration. Using blocking or reactivate methods could be the way to control mitochondrial pathways, and from this way of view, controlling stress response.
That is the dream. So I think one, mitochondrial dysfunction is always correlated with the neurodegenerative diseases, and there are of course a lot of questions about which one comes first. Does mitochondrial dysfunction cause neurodegeneration, or neurodegeneration is causing mitochondrial dysfunction? There's some genetic evidence on both ways. And then can we target mitochondria to create resistance to neurodegeneration or rescue? And there are so many studies, including in my lab. That's the goal. That's the dream.
Thank you for your response. I'm continuing on with another question. It is assumed that most mitochondria are generated in the cell body and then travel to the synapse to exert their functions. So what do we know about the mechanisms promoting mitochondrial biogenesis away from the cell body?
That's a really good question too. So one, Now there's more and more evidence about local translation. And what regulates local translation, we are still trying to figure out. And this is not an active research direction in my lab, but there are really, really great labs actually really addressing that issue. But then another question to also ask, what is regulating? How much is going to be locally synthesized? How much mitochondria is going to be delivered from soma? And how is that going to be coordinated? Like, who is keeping track of like checks and balances? And we still don't know. But this is a great, great question. I think it's a question in the whole field. So excellent questions.
Thank you again for your response. Another YouTube question. How does mutating the organization site of hexokinase 1 affect the functionality of neurons? What problems can arise in this situation?
So we identify that this is one of the key mechanisms to enhance metabolic efficiency in neurons. And so far we know if we affect this pathway, if you perturb, it's basically causing presynaptic dysfunction. And then eventually, if you have dysfunctional neuron, basically you may start losing memories, or its energy deficiency is so severe it can actually start killing neurons. In fact, we see more dead neurons when we have the oligoconnect mutant hexokinase. In disease connection, we know, again, especially in Alzheimer's disease, glucose metabolism and oligoconnect is low. And now we are bringing a new player in this picture. So maybe reduction in hexokinase 1 oligoconnect may actually really cause some of the early events that can lead to neuronal dysfunction. Right? And we're investigating more, though, especially in generation models.
Thank you for your answer. I'm continuing on with another question. Are there gender-based differences in mitochondrial functions in the brain? What are its functional implications in disease and health?
This is an excellent question too, and a question that we haven't investigated as much. That's the reality. And my postdoc actually right now doing some experiments. We're systematically looking at that. And another thing to also consider, hormone-dependent regulation of mitochondria and mitochondrial role in also regulation hormonal balance. Because there's a lot of coordination happening, and it's a great question, and I think we have to do more work on that. And of course, there will be a lot of functional implications.
Thank you for your response. Another YouTube question. Does oligoconnect have any relation with the chronic activation of microglia?
We haven't done any work on microglia or oligoconnect, but I know microglia is also expressing OGT and OGA, and there could be different type of proteins involved in the microglia activation, but there's a lot of work, especially on the immune cells and oligoconnect. So, but again, it's not something that they've done, and I don't know if anyone looked at the chronic activation of microglia and oligoconnect specifically.
Thank you for your response. I'm moving on to another question. Do mitochondrial microRNAs have a major effect on synaptic plasticity? Could they play a role in developing biomarkers, treatments, and/or targets for neurological diseases?
Short answer, I don't know, but it may be. I think we have more to discover in terms of special use of mitochondria-derived microRNAs and how microRNAs also can regulate mitochondria. And again, mitochondria also has some small RNAs being generated from non-coding regions of mitochondria too. So I think there could be really, really tight regulation and could play in different roles, but I don't know. And I know there's some microRNAs involved in the regulation of mitochondrial proteins, but under what conditions, I don't have an answer. And it's not a field that I'm super familiar with. So maybe there is some new work, but I don't know.
Thank you again. Another question. What are the main limits to studying ex vivo synaptic physiology and pathophysiology?
That's a really good question. So I think, in my opinion, one of the major limitations of ex vivo studies, and keep in mind, ex vivo studies, synaptic physiology and pathophysiology has been studied for a very long time. But when it comes to metabolism, we're defining the conditions, right? We're defining what's the media, we're defining how much glucose, how much ATP, and what other metabolites that we need to incorporate in our ex vivo conditions. And I think that's one of the key limitations. So we really need to be very careful about the conditions that we're creating, and then the questions that we're asking based on the conditions that we create. I think now people are more careful now to really define these conditions. But also, I think we also have to be more careful and investigate more what are the ideal ex vivo conditions if we think about especially neuropathologies.
Thank you for your answer. Moving on to the next question. Oxidative stress disrupts neuron signaling. How does this disruption affect interneuronal signaling and cognition? And what consequences does it have?
That's a really good question. Again, the short answer, we don't know. And long answer, there are a lot of studies indicating the importance of oxidative stress and how that actually can affect the cognition. But at the individual neuron level, especially for interneuronal signaling, the studies are very limited with interneurons, especially if we also think about the interneuron subclasses. My lab, like I said, we have one upcoming study specifically looking at the fast spiking parvalbumin positive interneurons. And so we need to do more work. And then again, the consequences, for example, in our work, this is in collaboration with Bianca Bloom and Stefan Lotgub's lab at UCSD, that we see if we perturb some of the mitochondrial function, we see disruptions in brain oscillation. So it can go it's one mitochondrial protein expressed in the spiking interneurons, and then if we change its level, it's causing change in whole brain level oscillation. So I think there's more work to be done, but this is just one example of an interneuron type, and there's so many, right?
Thank you for your answer. Another question. Can a disruption in the processes regulating neuronal plasticity and restructuring of synapses be considered the main factor in schizophrenia?
There's some evidence of that. And there is also some evidence of alteration of mitochondrial proteins in schizophrenia. And so potentially, but the exact mechanisms, we don't have all the details yet. And again, it's not really, we're not doing any work related to schizophrenia, but there's already patient evidence out there. It could be.
Thank you for your answer. As for our last question, what advice would you give to students studying in scientific fields?
So my advice, always remember you're the future. We can have all the ideas and we can talk about them, but I mean, the questions have been so stimulating and amazing. So it already shows you're already ahead, and just stay motivated. And I know sometimes, especially so much stress all around us, you think you cannot do it. You think you don't have the capacity or resources, but don't think that way. Always trust in yourself and your potential, and just look for opportunities. And again, if I can help for applications or looking over your application files, I'm happy to support. Just keep pushing, keep trying, and don't give up, because you're the future and you have an amazing potential. And I think that drive is the most important thing. Don't lose that.
Thank you so much, Dr. Pekkurnaz. On behalf of myself and all of our participants, I would like to express our gratitude to you for spending your valuable time, transferring your knowledge to us, and for this informative presentation.
Ben çok teşekkür ediyorum davetiniz için. Burada olmak çok güzeldi. Dediğim gibi sorular inanılmazdı. Lütfen bu motivasyonunuza devam ettirin ve sizler bizi geleceğe taşıyacaksınız. Çok teşekkürler. Thank you so much. It was a great honor to be here.
Thank you so much. I wish you very happy days.
You too.
At 8:30, Associate Professor Dr. Farhat Ay will be with us to give us a speech on computational methods for studying the 3D organization of the human genome. Thank you all.
Bioinforange platformunun kuruluşunun 2. yıl dönümünde düzenlediğimiz Bioinfocongress II etkinliğimize hepiniz hoş geldiniz.
Merhabalar, hoş geldiniz laboratuvarımıza.
Merhaba hocam, hoş geldiniz.
Merhaba, hoş bulduk. Sağ olun, teşekkürler davetiniz için, organizasyon için.
Davet için tekrar çok teşekkürler. Organizasyon da çok başarılı gözüküyor. Çok teşekkür ederim.
Tekrar teşekkürler davetiniz için. Bugün bilgisayar teknolojisiyle biyomoleküler sistemlerin simülasyonu ve ilaç tasarımı konusunda konuşacağım.
Muhteşem. Çok teşekkür ederim. Evet, hepinize merhabalar. Organize komitesindeki arkadaşlara da çok teşekkür ederim beni davet ettiğiniz için.
Herkese merhabalar. İlk önce davetiniz için çok teşekkür ederim. Buradaki diğer konuşmacılarla beraber sizlerle beraber olmak gerçekten gurur verici.
Bioinfocongress'te sunum yapmaktan beni davet ettiğiniz için çok memnunum ve sanıyorum tamamen insan hücre fonksiyonlarına yönelik olmayan daha basit canlılarla ilgili olan tek konuşma belki. Ama böyle bir değişiklik getirmekten de çok memnunum.
Öncelikle hepinize teşekkür etmek istiyorum. Bioinfocongress oldukça güzel bir organizasyon yaptınız.
Tüm Bioinforange çalışanlarına ya da gönüllülerine teşekkür ediyorum. Beni davet ettiğiniz zamanınızı ayırdınız.
Merhaba, davetiniz için çok teşekkür ederim. Bu aktiviteyi organize ettiğiniz için de teşekkür ederim. Gerçekten çok büyük bir çalışma gerektiriyor.
Thank you, it's a pleasure. Thank you for the invitation.
İyi akşamlar. Başlamadan önce bu güzel organizasyonu düzenleyen arkadaşlara gerçekten teşekkür etmek istiyorum. Bu kadar büyük bir topluluğu bir araya getirmek, böyle bir organizasyon gerçekleştirmek gerçekten büyük bir başarı.
It's a really fantastic program that you've put together over these last few days, so it's been some brilliant science and some brilliant scientists speaking. So thank you very much again for the kind invitation.
Welcome everyone, I'm Neva Çalışkan. I'm a group leader at the Helmholtz Institute for RNA-based infection research. And today my talk will be about how meaning of the genome can be altered by frame-shift RNAs and how this response can be regulated by host and viral proteins.
Teşekkür ederim davetiniz için. Bugün bugün mRNA aşılarından bahsedeceğim sizlere.
Merhaba arkadaşlar. Bugün sizlere katılmaktan çok hoşnutum. İlk önce Bioinforange öğrencilerine bu güzel organizasyonu bir araya geldikleri için teşekkür ediyorum. Gerçekten benden önceki videoları izlemekten çok hoşlandım.
Merhaba herkese. Buraya geldiğiniz için teşekkürler. Bugün ökaryotik genomların organizasyonu hakkında bir konuşma yapacağım.
Merhaba herkese. Katıldığınız için ve dinlediğiniz için teşekkürler.
Merhabalar, hoş bulduk. Başlamadan önce böyle güzel bir organizasyonda emeği geçen arkadaşlara teşekkür ediyorum. Ayrıca davetiniz için de teşekkürler.
It's a tremendous honor and pleasure for me to be here after starting my lab just over 2 months ago.
Gün boyunca 30 saat bir yayın süremiz oldu. 3 eğitim, 17 konuşma, toplamda 20 konuşmadan görüyorsunuz yorgunluğumuzu da arka planda. Öncelikle bizlere konuşmalar, eğitimler veren 20 değerli hocamıza çok teşekkür ederiz. Açıkçası davet ettiğimiz hocalardan çok yüksek oranda kabuller aldığımız için bu kadar çok yoğun bir programımız oldu. O yüzden hocalarımıza çok teşekkür ederiz. Onların bize davetleri bu kadar çabuk bir şekilde, hızlı bir şekilde bizleri kabul etmesi, konuşmalar yapması bizler için çok güzel ve bizleri gerçekten çok motive ediyor. Hocalarımızın haricinde siz şu anda 277 kişi var bu saatte. Çok teşekkür ederiz. Değerli katılımcılarımıza çok teşekkür ediyoruz. Ancak büyük bir teşekkürü de ben buradaki ekip arkadaşlarıma yapmak istiyorum. Bioinforange'ın 2. yıl dönümü, doğum günü. Buradan Bioinforange'a emek veren, gönül veren, Bioinforange'ı seven tüm bilimseverler, katkı sağlayanlar, öğrenciler, insanlar adına iyi ki doğdun Bioinforange diyorum. Bilim paylaştıkça güzel, bilim paylaştıkça ilerler. Bilimle kalın. Bioinforange platformunun bilim yolculuğundan 3.sünü düzenlediğimiz Bioinfocongress IV etkinliğimize hepiniz hoş geldiniz.
Çok teşekkür ediyorum. Mehmet, senin şeyin nezdinde bütün organizasyonu yapan arkadaşları da tebrik ediyorum. Emeğinize sağlık. İyi bakın kendinize, görüşürüz.
Merhaba İvan Hocam, tekrardan hoş geldiniz.
Merhaba, teşekkürler davet için.
For scanning your valuable time, transferring your knowledge to us, and for this informative presentation. Kendim ve tüm katılımcılarımız adına çok teşekkür ediyorum hocam.
Ben teşekkür ederim.
Çok memnun oldum hocam. Sağlıklı günler diliyorum.
Sizlere bir akşam.
Yep, thank you guys so much. I really enjoyed it. Great questions from everyone.
Merhaba Tuğba Hocam, tekrardan hoş geldiniz.
Hoş bulduk. Çok teşekkür ediyorum davetiniz için. Harika bir organizasyondu. Herkese selamlar.
Değerli vaktinizi ayırdığınız, bilgilerinizi bize aktardığınız ve bilgi dolu bu konuşma için kendim ve tüm katılımcılarımız adına çok teşekkür ediyorum.
Ben teşekkür ederim davetiniz için.
Merhaba Nilüfer Hocam, tekrardan hoş geldiniz.
Merhaba, hoş buldum. Beni iyi duyup görebiliyor musunuz?
Evet, görüyorsunuz.
Çok teşekkür ederim davetiniz için. Çok teşekkür ederim. Öğrencilerle böyle bu sanal ortamda da olsa bir araya gelmekten.
Merhabalar. Öncelikle davetiniz için çok teşekkür ederim.
Teşekkür ederim hocam.
Çok güzel bir event ortaya koymuşsunuz. Bu kadar değerli bilim insanı arasında yer almak gurur verici. Teşekkür ederim.
Çok teşekkür ederim. Nazik davetiniz için çok mutlu oldum. Bu arada bu hani YouTube'da olan sayın seyirciler, bu saatte cumartesi akşamı gidip başka bir şey yapmayıp bu konuşmayı dinlemeye geldiğiniz için size de hürmetlerimi sunuyorum.
Çok teşekkür ediyoruz. Hem kendi adıma hem de bütün katılımcılarımız adına.
Rica ederim. Ben çok memnun oldum. Beni onure ettiniz. Çok teşekkür ederim.
Biz onur olduk hocam. Çok sağ olun.
Size çok teşekkür ediyorum. Katılımcılarımız ve kendim adına. Ben çok teşekkür ederim. Benim için de güzel bir deneyim oldu.
Bu harikasınız için katılımcılarımıza bütün ekibimiz adına çok teşekkür ederim hocam.
Ben çok teşekkür ederim beni misafir ettiğiniz için.
Thank you. Thank you so much for this space. Thank you for having me here and for this opportunity.
Bilgi dolu konuşma için kendim ve tüm katılımcılarımız adına çok teşekkür ederim. Ben teşekkür ederim davetiniz için.
Tekrardan çok teşekkür ediyoruz. Katıldığınız için çok mutlu duydum.
Ben çok teşekkür ederim. Çok güzel bir organizasyon gerçekleştirmişsiniz. Beni davet ettiğiniz için tekrar teşekkürler. İyi günler diliyorum.
Biz teşekkür ederiz hocam. Bioinforange platformu olarak düzenlediğimiz Bioinfocongress IV etkinliğimizde çalışmalarına yurt içinde ve yurt dışında devam eden değerli bilim insanları katılımıyla toplamda 15 seminer gerçekleştirmiştik. İzninizle burada organizasyonda emeği geçen arkadaşlarımı davet etmek istiyorum. Şimdi burada ekranda olan arkadaşlarımız haricinde kongremizin düzenlenmesinde organizasyonda emeği geçen tüm ekip arkadaşlarımıza teşekkür ediyoruz. Ve sizler gibi yani burada hepimiz öğrenciyiz biz ekip olarak. Özellikle öğrencilere bir katkımız oluyorsa akademik hayatı ve işte gelecek idealleri konusunda bir fikir verebiliyorsak buradaki konuşmalar, işte bizler bir katkı sunabiliyorsak açıkçası biz bundan çok mutluluk. Tekrardan etkinliklerimize devam edebilmeyi umuyorum. Bizi sosyal medya adreslerimizden takip edebilirsiniz. Sizlerin desteğiyle biz motivasyon buluyoruz. Bizler motive olunca da böyle güzel etkinlikleri sizlere sunmaya çalışıyoruz. O yüzden karşılıklı etkileşim ve destekleşmeyi umuyorum. Tekrardan hepinize çok teşekkür ediyorum. Katıldığınız için de çok teşekkürler tekrardan ve herkese bol başarılı akademik kariyerler, güzel bir hayat, sağlıklı bir hayat diliyorum ve sağlıklı günler diliyorum. Görüşmek dileğiyle tekrardan. Hoşçakalın.
Bioinfocongress IV. Associate Professor Dr. Ferhat Ay. Computational Methods for Studying the 3D Organization of the Human Genome. Associate Professor Dr. Ferhat Ay received his bachelor's degrees in computer engineering and mathematics, both from Middle East Technical University. Turkey in 2007, and his PhD in computer science at the University of Florida in 2011. He subsequently joined the Department of Genome Sciences at the University of Washington as a Computing Innovation Fellow, awarded by Computing Research Association and National Science Foundation. He was a research assistant professor in the Department of Preventive Medicine Health and Biomedical Informatics at Northwestern University in 2015. Before establishing his lab at the La Jolla Institute for Immunology in 2016. He is currently the Institute Leadership Associate Professor of Computational Biology at LGI, an adjunct faculty at the UC San Diego School of Medicine and UCSD Bioinformatics PhD program. His primary research areas are bioinformatics, computational biology, epigenomics, and regulatory genomics. His lab works on understanding the relationship between three-dimensional genome architecture and gene regulation in the context of different immune-mediated diseases. He serves as an associate editor for PLOS Computational Biology and an editorial board member for Genome Biology and the Journal of Computational Biology.
Dear Associate Professor Dr. Ferhat Ay and participants, welcome all of you to our Bioinfocongress IV event. Associate Professor Dr. Ferhat Ay will give us a talk today on computational methods for studying the 3D organization of the human genome. At the end of the presentation, we will have a question-answer section prepared by our team and consisting of questions from our participants. Dear Dr. Ferhat Ay, welcome back.
Thank you very much. Thanks a lot, Nazlı, and thank you to the whole Bioinforange team. What you guys are accomplishing is amazing. I think you guys are showing a good example as what you can do with little bit, only little resources, but maybe the youth energy and the willingness that you guys are showing for organizing such events and teaching a lot more people in our country bioinformatics and related biomedical sciences. I should probably start with my slides, but you think that the aspect ratio, if this is small, then I can just probably turn this one off. I'm sorry, I was trying to do it with 3 screens, but maybe this is just too much.
Oh, it's
So I'll just share another screen. I just wanted to see you guys and the questions all at the same time, but this might be a little bit too much. Hold on 1 second. All right, how about now? Can you guys see the slides? Because I cannot see right now. Can you guys see them?
Yes, hocam. You can see
Okay,
clearly.
perfect.
Yes.
All right. Yeah, sorry that I tried too much, I think. Thanks again. Thank you all. And thank you for staying. I know it's a late night almost in Turkey. And yeah, but I'm on the West Coast, so it's kind of like in the morning for me. I hope that you guys are still able to stay awake and listen to this talk. So as the introduction mentioned, I'm a faculty right now at the La Jolla Institute for Immunology and also at the UC San Diego. And what we study is the 3D organization of eukaryotic genomes in general, but mainly human. So before I really go into any of the science, as you all know, I don't need to, and I think, you know, there's nothing that hasn't been said about what's been happening in Turkey. So I'm not going to belabor, but we've been having one disaster after another. And I know that everybody is still trying to hold on to, you know, what they are working on, their ambitions, their hopes. So I really hope that everything is going to be much better for our country. But in the meantime, my sincere, you know, condolences for everyone who's lost any family, any friends, including you guys. You've lost a team member. So I'm just hoping there's going to be better days ahead. So I wanted to start with a little bit of my journey, and I sometimes show this slide to people here too, just to tell them like what Turkey looks like and what is where, and also like in terms of you guys and in terms of you know, young scientists. It's important that you know, like I don't know, professors don't grow on trees, right? So we also come from real lives and real locations. So I was born in a small town in Manisa in Gördes. It's about 10,000 people. I lived there for a while. Then my family moved to Bursa, and then I joined the university in Ankara, Middle East Technical University. The first time that I flew abroad, it was for my PhD, starting my PhD, and that was University of Florida. So I was there for 4 years. I had a brief internship at MIT in the meantime, then moved for my postdoc to the other side of the country. This is about 3,000 miles away. And then after my postdoc and of course getting married there and starting a family, we moved to Chicago for a year and then to San Diego. So we have covered all the corners of the country. And then together with a lot of the big projects that we have in lab, we also have 2 small projects at home. So this has been my life in the past couple of years, essentially, juggling science and kids and all the disasters and all the things that are happening around the world and in my country and in the USA. So what do we do in our lab? We are a computational biology lab. So we do have interest areas in a lot of different diseases and applications of computational biology to them. So some of them are related to cancer. With some pharmaceutical companies, we are working on autoimmune diseases and also we are working on some infectious diseases. But what ties everything all together for us is the computational methods development. So developing methods that are of high utility and that are accurate and efficient for analyzing the ton of data that's being generated in labs all across the world, right? And then as you can see, the bioinformatics starts with bio, right? I'm a computer scientist. I did computer science and math and all of that, but now that we are doing bioinformatics, we always need to keep in mind that it starts with bio, and the biology aspect is really important. So with that in mind, we are happy with being a computational lab, but I mean, there are some disadvantages of computational work. And then I will only mention one here. This is a quote from my 6-year-old a month ago when I was going for a seminar. It's like, why are you going, Daddy? You're not even a real scientist. You just do stuff on your computer. Jokes aside, but that might be the sentiment sometimes computational biologists might be getting. But this is something that you should, never really worry about. Holding a pipette versus typing things on your keyboard, they are not inherently different. They are all about like discovering something that people have not discovered before, and making humans happier and healthier and more hopeful for the future. So what we do really like in the lab is to try to understand this intricate organization of how our DNA folds and fits into our nuclei in our eukaryotic cells, right? So we have 23 different chromosomes, 2 copies of each, and then they fold and fit into a teeny tiny nucleus, right? So when you add all the DNA on top of each other, kind of stitch them together, it's about 2 meters of DNA per cell, and then it's fitting into a nucleus that's about 1 million times smaller, right? So how does this happen has been a question that the field has been asking for many, many years. But the real revolutions and breakthroughs came when we went from being able to look at only under the microscopes to a few regions at a time to this whole next generation sequencing based assays where you can now look at the many cells all at once, and then all loci, all regions, and then how proximal they are, and so on and so forth. So the working horse in our field for the 3D genome and chromatin organization these days has been this assay called Hi-C, and there are so many variants of it. But essentially, it is you take your cells, you take your sample, you cross-link them. So essentially holding things together in the nucleus for a while. The things that are close to each other will be held together by these cross-links. And then you need to use some sort of scissors, whether it's restriction enzymes or some other versions of it, but you need to cut them so that you kind of like go through some sequencing steps after that. You cut them. When you cut them and don't remove the cross-links, they are still together if they are in the 3D genome, if they are proximal. So after a couple steps, you can religate them, put some markers, pull down with those markers, etc. You need to linearize it so that you can sequence. But once you do that, and once you, of course, spend a lot of sequencing dollars essentially on these libraries, you may get hundreds of millions or billions these days of read pairs, where each read pair will tell you what is the proximity between, let's say, these yellow region and the orange region in the genome, how many times you are seeing a read that puts them together in this one product. So our job starts after that. And then the nice thing about all of these, we can completely forget everything that I said and think about this as a matrix, just a mathematical object, right? And then when you look at this matrix at like different zoom ins and different resolutions, you can see a lot of patterns that explain how our nucleus is organized. So for instance, at the very broad resolution where you see all the chromosomes, of course, you see the chromosomes are tightly folded within each other, not as much interacting within the next chromosome, and so on and so forth. So this we call chromosome territories. So you can take a chromosome, zoom in a little bit, and then you see these checkerboard patterns, flat pattern here. And then this tells you that some regions like to interact with others more than the rest. And then that there is what we call compartments, chromatin compartments, where some of these regions are active and they tend to interact with each other more, and then some of these regions are inactive in their chromatin. These are harsh generalizations. These are multi-megabase regions, of course. There's more fine-grained things happening there. But then again, if you take maybe one of them or two of them and then zoom in, then you start seeing these diagonal blocks. And sometimes you see these dots on those diagonal blocks. So this is what we call, these blocks is what we call domains or topological domains. And then these dots are what we call loops. So this one nicely depicts, the graphic underneath it nicely depicts that these loops can be between the two endpoints of such domains, or they can be within the domain and connecting regulatory regions such as genes, their promoters and enhancers, and so on and so forth. So in our lab, we have been very focused on the loop calling, the identification from these matrices, identification of patterns that are suggestive of loops, whether they are at the boundaries like this or whether they are connecting regulatory elements. So our lab has developed a series of tools in this for different data types, and as the resolution and the field has evolved. More recently, we have also gone back to this, the broader architecture, which is the chromatin compartments. Calling them has been not much of an issue, but comparative analysis is, and then now we have a ton of different data sets that you need really solid statistical methods to do this. We have just developed one recently. And then one other aspect of this we have worked on, and I'm going to talk to you about this also, is the genomic rearrangement. So you can think of them mainly in the context of cancer, when a linear DNA is kind of like shuffled around, when something is deleted, when something is amplified. What happens? And what happens is that some unexpected patterns are seen in these high CMAPs, and we can opportunistically use them to discover such rearrangements. And this is very helpful in the context of cancer. So this is a classical example. I've seen this slide and I presented this slide, I mean, maybe hundreds of times right now, but it never gets old because it is amazing. So it is a gene called sonic hedgehog that is very important in development and in different stages in development. And it has different enhancers in different places. In one part of the development, in the limb development, in either mouse or human, there is an enhancer that controls this gene, and it is 1 million base pairs away. So that blows your mind when you think about what people are looking. When we generally look at what regulates the gene, you look at the gene and then a little bit around it, maybe 5-10 kb at most. This has been the status quo for many, many years, but now, of course, it's changing. There are very important genes such as sonic hedgehog that are being impacted by, multi-megabase loops such as, I'm just checking my email just in case everybody is here, but let me know if I disconnect. I'll have my email. What's happening here is that changes or deletion of this enhancer totally determines how your limb development is going to work out. And in humans, the same enhancer is conserved and also still quite far away. Even a single base pair change in that enhancer is changing the phenotype. You can see all these diseases where you have like a polydactyly and more digits than what is assumed normal. So what this is telling you is that maybe we have been overseeing certain regions such as these enhancers for many years because we haven't been looking at the right places. And the only way for someone who's rational to be even thinking about this enhancer as something to do with this is some sort of physical evidence that they are coming together in the three-dimensional structure, right? Otherwise, you cannot exert anything over such distance. So a little bit of background in the looping field and kind of where we are right now. So the loops and TADs, these domains, how are they formed? So in 2014, with the increasing resolution of these high-C data sets, Erez Lieberman's group was able to come up with a hypothesis that said, when we look at these strongest loops, what we see is the binding of CTCF, which is an insulatory protein, on the anchors of these strongest loops. They are highly enriched. And then what they showed was that not only they're enriched, but it's not palindromic, so it can bind to one strand or the other. So the orientation at which this CTCF binds it matters. And then only at the divergent orientation, the loops kind of like are the strongest here. So how is that happening? What is how do these two ends find each other? Right? And then came the idea of this loop extrusion, which is there must be something that's extruding the DNA, such as this slide that you're seeing here. And then maybe CTCF, these divergent CTCF sites are where this extrusion stops. And then you have this domain formed in the number 4 here, as you can see. And then they have tested this idea by deleting CTCF sites and adding CTCF sites, and then the predictions and the experiment in at least a limited number of cases have kind of really agreed, supporting the idea of this loop extrusion. So what is this loop extruder though, right? I mean, CTCF is where it stops, but what's this motor kind of thing that extrudes the DNA? So we think that it's cohesin, but before going there, people have looked at what happens when you degrade the CTCF. If you cannot 100% degrade it, but if you can, get it to a minimal level, what happens is all of these loops and domains disappear, but nothing changes in terms of these compartments, these A and B compartments. Nothing changes in terms of replication timing. We have shown nothing changes. So when you go into cohesin, which is what extrudes these loops, and it's a huge complex, so you really need to look at its individual components and knock down some of its subunits. What you're seeing is, again, it does eliminate loops. Compartments are not touched as much. In both cases, though, what has been puzzling to all of us in the field is that if this is so important, if all of these loops and domains are so important for gene regulation, then when you fiddle with the CTCF or cohesin, then you expect a lot of changes in the transcription too. But that's not the case. The changes to transcription has been very minimal. And there are reasons for that I will not go into today, but happy to discuss with you guys if anybody is curious. Then people have gone into depleting what loads and unloads cohesin. And then these have been very interesting, that if you deplete what releases cohesin from the chromatin, then you have loops going on and on and on, stronger and longer loops. But compartmentalization is kind of like lost a little bit. That plat pattern is lost a little bit. Vice versa, if you deplete what loads cohesin onto the chromatin, then you really cannot establish these longer range loops that you're seeing here. They are lost, but it kind of gets compartmentalized a little bit. So then came the idea that maybe these are two different forces, that the looping versus compartmentalization is two kinds of different forces, that one keeps the other one in control. What was really cool for me reading when this paper came by Xiaohui Zhang's group at Harvard was that all of these data I've been talking about is, of course, high-C data and in bulk cells. And then they have done this beautiful chromatin tracing experiments, which then they can look at a single cell at a time. And then they have shown that even if you get rid of cohesin, these blocks at these diagonal patterns or triangles when you rotate them, but they are squares when you don't, they are still there in each cell. But what's very interesting is that the places at which their boundaries are occurring, there's not much of a preference. When you take away chromatin, cohesin, then the boundaries can open anywhere. And then when you combine 10,000 single cells and plot this, the boundaries can be anywhere. Whereas if you have cohesin, then you have these regions that are going to be more likely to be boundaries, and then you see these in the bulk data. So this is kind of an introduction, and of course a lot has been happening in the past 5 years or so in this field, but just wanted to let you guys know about what looping is about and what people are thinking right now. So we have even before some of these papers, we have when the first IC datasets started coming, we started thinking about, okay, how can we best analyze these datasets? Because with every genomics data, there is confounding factors and biases and other things that you need to worry about. And the initial methods generally, do not going to there, and then later these things are developed. So we developed an algorithm back in 2014 with the very simple idea that essentially, if we looked at the genome and where there's sequencing depth back then was the strongest, can we develop a way to look at the contact patterns and to identify these really specific and cell type specific interactions and loops between genes and other regions? So this is what we employed back then, and it has been actually highly utilized and cited because sometimes these simpler methods get better traction. In this case, it is because it's a method that is very flexible. So this background, we are learning from the data itself. We are not assuming a specific distribution. We are not doing anything fancy there. We are just saying the data, if you look at the data, it will tell you what the background should look like. And there are some statistical things that we are doing to make sure the null part of the data is separated from the real loops. But once you do that, then you can just simply use this, and then you can incorporate the biases and other things very easily into this formulation as well, which then for each pixel will assign a p-value statistical significance, which then you can multitest correct because this is billions of tests literally, and then identify regions that are significantly coming together. This we did in human and mouse and malaria at the time, but it has been applied to essentially any genome that you can think of. Again, all because it's very flexible and it's an empirical null model. So more recently, though, with the increasing resolution of these high-C data sets and then the introduction of micro-C, which does improve the resolution also conceptually because it uses a different digestion system, we revisited this problem. So FitIC is a global background model. We look at globally what should be the background, and for each pixel we ask whether this is about that background. Whereas this one is a little bit different. So this came in when a PhD student at UC Riverside joined my group for a postdoc, and then he comes from a signal processing and image analysis background. So then when we look at these contact maps, I always think of them as a matrix. And then he started saying, okay, let's look at it as an image. And then there are a lot of tools in the image processing field that can be adapted for this type of problem. So one thing that he mentioned that is missing in the field is that think of these sunflowers, right? This is beautiful. And I've grown in such farms back in Turkey when I was growing up. So when you look at it, if you wanted to write an image processing algorithm that detects sunflowers accurately, what's going to need to happen is that it's going to have to deal with the scale issue, right? Something all the way in the back is a sunflower and something all the way in the front is a sunflower, but they're of very different scales. So the theory in computer vision that deals with this is called scale space theory. So essentially, scale space theory is, I will show you in the contact maps, but it does take an original map, original image, and then it does smooth it with a different Gaussian parameter. So it creates you a set of images that are smoothed to different extents. So then what it does is that you can take a difference between each of these different smoothed versions and get this difference of Gaussians. These are very easy to, manipulate. I mean, the Gaussian assumption and then a bunch of other statistical properties make sure that we are not introducing some random dots and points that are not there in the image. So it does limit the false positives. But when you get this difference of Gaussians, then you can do a computation of local maximum both within a given scale and across the adjacent scales. And then what this tells you is that not only that there is likely a loop here, but also that loop is most pronounced in the specific scale. So it allows us really to not only detect the loops, but to detect the scale of the loops, where if a loop is narrow and involving two pointy regions like this or more broader between the two regions, we can capture them. So this has been extremely useful and extremely powerful because compared to the state of the art, the algorithm that everyone uses, which is a little bit data hungry, it doesn't work with the lowly sequenced genomes. We were able to essentially find pretty much 80%, 90% of what this algorithm is able to detect in terms of loops, but much more, right? When you do this, of course, then you really need to show the ones that you are now finding are de novo, and then we are doing that. I have some data for that. But another thing that was nice is that not only that, but we also compared to this algorithm, we also improved the reproducibility between replicates to 80%, right? This was more like 60 to 70%, which becomes really critical for downstream analyses. And when we looked at the one question that we asked, there is this high-C data that we have all analyzed and high-C data that we are familiar with. And there was this micro-C that was the new guy on the block, essentially. The fundamental difference between the two is that high-C, you use restriction enzymes to cut the DNA, whereas in micro-C, you use MNAs, which does cut whenever it finds nucleosome-free regions, if you think about it. So this does give you much higher resolution. Of course, you need to sequence this deeper too. But when we looked at the same cell type and the high-C data versus micro-C data, what we have observed is that micro-C really does capture 90% of what high-C captures, but also much more. So this has been then validated in the field, and recent work points to this as well, that a lot of these loops that are micro-C captured, they are between regulatory elements, and they are very important for gene regulation. So when we look at this is an aggregate plot of these peaks. So think about 36,000 such small matrices centered on each loop that we call, and then we take their average. As you can see, micro-C is like the signal to noise ratio is much better. It enriches that. So you have these more point loops here. And then when we looked at the genomes, when we forced high-C and micro-C to call the same number of loops, you can see that the high-C will go towards these bigger and broader loops, whereas micro-C is going to go for these smaller and darker loops. And then when you let the micro-C identify however many loops you can identify, as you can see, it captures everything that high-C does and more. That's very interesting and important for us. So one thing that allowed, and this is a very efficient algorithm, Abbas was able to implement it in a way that this runs in a couple minutes per chromosome for these extremely large data sets. So, and by extremely large, I mean like this is 1 kilobase. So if you think about the human genome as 3 billion base pairs, then you might be talking about a 3 million by 3 million matrix, and of course the chromosomes and so on and so forth. But it is a very big matrix that you are dealing with, extremely sparse. So what happened was that this enabled us working with resolutions that other tools haven't been able to scale, such as 1 kb resolution, and that enabled us to zoom into places that other tools and other papers have completely ignored, such as within 20 kb, because there's so much noise there due to physical proximity or linear proximity of the DNA that other people have essentially, including us in the past, we have refrained from really going and looking into that, thinking that we might not be able to identify specific loops there. So more recent paper from a group at Berkeley, Robert Tijan, so they have utilized our tool with their micro-C data and have studied the role of these specific proteins, that some of which I mentioned are important in the, creating the DNA loops and domains. So what they have done is they have found 75,000 dots and loops, and they have done it in a way that they were able to, using this extremely highly sequenced micro-C data, they were able to go down to a couple hundred base pair resolution. And as you can see, our algorithm was able to really find these patterns of, enrichments that are corresponding to loops. So another again, like the field is going towards more and more high resolution assays, generally genome-wide, but this was from Anders Hansen's lab at MIT. There again, they have come up with this region capture version of the microseed. So instead of sequencing the whole genome, they are capturing a region of their interest, and they sequenced it in a way that, for these regions, in a way that if you, had a genome-wide, it would be 200 billion reads, right? So for, I don't know if I should put a price on this, but this would be, let's say, almost $1 million if you did it genome-wide for one cell type, one condition. So then what you see here is that, again, Mustache was able to identify these loops, these patterns that are only appearing at this ultra high resolution. So we believe that we kind of have a good way into this new set of experiments that are coming up that are pushing the envelope in terms of resolution at which we can find loops. And we think our tool is essentially going to be very critical in that. So this was work by Abbas, and then Tuvan was a student who came from Sabancı one summer, and then he really extremely helped us as well, publishing this paper. And Stefano Lonardi was also involved towards Abbas's PhD advisor. So then we went on to, then the data sets or the assays have evolved, and then so did our tools. So we have developed a tool called FitHighChip for analyzing some data that comes from an assay that's called HighChip. So HighChip is essentially like Hi-C again, so you're trying to understand the 3D genome organization, but you want your sequencing money to be spent on regions that you care the most about and not on other places. So essentially then you have an antibody pull-down step where, whether you're interested in active histone modifications or a specific transcription factor, places it binds will be highly overrepresented. So this was discovered back in the 2016s. And then, unlike the Hi-C maps that I have shown you, this one has this pattern that it kind of has these enriched regions and then the background regions. So the same methods that we developed for Hi-C had the assumption that the background is going to be more or less uniform and was utilizing that. So we had to develop another version of it, which essentially can take such contact maps and then turn them into these loops here. Here you are seeing a promoter and enhancers that were validated to impact the expression of this promoter. So if you are given this kind of a contact map, what you want to really come up with is these loops that are linking the promoter to these functional enhancers and nothing else, and nothing less and nothing more. So a very talented postdoc, Surya Bhattacharya, he has developed this method here, which I'm not going to go into details, but kind of relies on the same idea, but then has a different way of dealing with the biases through a regression method. And then also deals with these indirect loops and kind of like finds where is the likely, anchor of these loops rather than reporting all of these. So this has allowed us to hit a balance between specificity and sensitivity of loop detection, because again, state-of-the-art previous methods, if you have given this region that I mentioned to you, that there are multiple validated enhancers, one of these would have found only the strongest one here. Another one that was in Nature Methods back then, it would essentially link everything to everything, whereas we think we have hit the right balance. And this is just one example, and we have, of course, like all quantification of this in the paper, that we believe that this is a good way to identify such loops. So this is, of course, online, and Surya and with some collaborators at LGI, we have applied this to immune cells, and then this was in the context of just characterizing different immune cells and the disease-associated variants. And then later on, the same resource has been very helpful for us to look at when the COVID data came, COVID GWAS data came, identifying risk variants for severe COVID. We were able to then use these looping maps and identify the gene targets of such genetic variants, which gave us some ideas as to what are the actual genes rather than the regions that are important in immune cells. for differences in COVID severity. So that's I guess like we can call it part one of my presentation. Now moving on to the part two, we have done, again, as I mentioned, went back to this chromatin compartments. And then this happened again with a talented postdoc and a high school student back then when he joined, that they started this project. How can we compare compartment calls across different conditions now that we are having more and more high-C data sets every day, and it goes beyond this pairwise comparisons. So again, these compartments are those two big chunks of regions, these flat pattern regions that like interacting with each other but not across. And then generally, as conventionally, we call the A compartment the open chromatin regions and the B compartment the closed chromatin regions. And they fall out from this matrix. You take this matrix, you normalize, you correlation transform it, and then you do essentially principal component analysis or eigenvector decomposition on this. And then the first eigenvector does give you the positive and negative signs, these A and B compartments. So their identification hasn't been much of an issue, but when you look at it, then across different conditions, those values may not be scaled. The sign is completely arbitrary. Sometimes you put it in one method, and this is plus and this is minus, and another package, and it's completely arbitrary, which makes sense. Then you need to really flip it. Distinguishing between technical and biological variations, so essentially using the replicate information, hasn't been properly done, and then the comparisons were mainly limited to pairwise, and then now we have many data sets and time series and so on and so forth. So with that motivation, we have developed a method that's called DC High-C, differential compartment analysis of High-C data. So it starts from these contact matrices or these compartment calls, and then after some normalizations, it uses this distance called Mahalanobis distance, right? So this is a generalization of a 2D Euclidean distance to multidimensional space. And then by using this distance, we are able to both look at within replicate and across condition distances and come up with statistical significance, again, of these changes in the compartments across different conditions. So we have of course shown it in pairwise comparisons that this is finding you the right kind of changes and consistent with what's out there. But then we applied it to a three-way comparison. Then we went to 13 different cell types in mouse, blood cell development, white blood cell development, if you think about it that way, or immune cell development, and then also a recent High-C data, single cell High-C data that came from studying developing mouse brain. So what we have learned is that this is loaded, and I'm not going to, of course, go through this, but hematopoietic stem cells going all the way to different lineages, common lymphoid progenitors, granulocytes, megakaryocytes. So you see a lot of changes in the compartments. And then what our method actually is able to do, I'll first show you this, that it's of course able to find cell type specific compartmental changes, such as this one where this region and this gene is active in the granulocytes. And then as you can see, when you look at the compartments, it's only here, and then you can see that we assign it a statistically significant, very high or low, I guess, p-value here that determines it's significantly different. This is another gene where the traditional methods would have failed to find because it's in the A compartment across all of these different conditions. It's an important transcription factor, and it's expressed throughout hematopoiesis, but to different extents in different progenitors and different cell types. So it's kind of like highly fine-tuned. And then as you can see, our method captures this as a statistical difference between these conditions, highlighting that not only things that flip like this one and obvious, but fluctuations is also important, which you can capture from this multi-dimensional data analysis. So maybe I will not go into this too much, but what you are seeing here is again doing it in the mouse brain development for cortex and hippocampus, and 6 time points each. When we looked at it as early, middle, late in a given tissue, we found these compartmentalization patterns and clusters of them that change concurrently. And again, they correspond to regions such as this one, where the compartment goes from active to inactive, or also this one, where the compartment goes in accordance with the role of this gene, and this region in the early and late development compartment gets more and more active, or different changes significantly. That highlights an important feature of this gene. So again, this was developed by Abhijit, who's still in the lab, and Jeffrey, now undergrad at Harvard. And then they have developed not only a really nice package, but also super easy to browse IGV browser visualizations that immediately fall out of the application of the method. And this was published last year. So going back, I kind of I could have talked about some other published work and all of that, but I really want to talk to you about something that is new and that I'm excited, and that I think is holding a huge potential in terms of what we can do with all of these computational tools in a context of disease. In this case, it's pediatric cancer. How we can use it to find interesting and novel targets and potentially inform new therapies. So as I mentioned, we can go back to these Hi-C maps or contact maps, and then go after patterns that are unexpected or abrupt, or that are not explained by the regular 3D folding. So when you look at this, you may realize that some regions are more faint and some regions are more stressed. And then when you look at the coverage, you will see a difference there. Overall coverage, essentially, the marginalized counts per row or column here. And then on this side, this is a cancer cell line, breast cancer cell line. And then on this side, what you can see is translocate, well, I shouldn't give it away, but interchromosomal patterns that are not supposed to be there, like between two chromosomes, strong interactions that are as strong as within chromosomes that are not supposed to be there. So back when I first joined, this was our first paper with Abhijit, where we have said, okay, let's utilize these patterns and then identify genomic rearrangements. And then we utilize these patterns to identify copy number variations. And in this case, this is a translocation where arms of two chromosomes are exchanged, essentially. And then that leads to these patterns. So we developed algorithms back then for this. But while we were doing that, we were thinking, okay, this is like, these may be easy cases. And we know that there are more like complex rearrangements. What can we do? Or what would be a good model to study whether our methods are useful for solving something that's more complex? And then with some collaborators, we ran into this data where there is a mouse model, mouse embryonic stem cells, that to study Down syndrome, which is a trisomy of chromosome 21, scientists have tried to insert human chromosome 21 into mouse cells. And of course, the chromosome is big and you cannot stick it into there immediately. So how they achieved that was irradiate human chromosome 21 and then transfer those irradiated pieces, which goes into mouse cells. And then when it goes into mouse cells, somehow it finds its pieces back together. But you can think of it like it's finding its pieces back together is done in a hurry, that it's kind of like putting everything in a bag, and it's not putting them in the right order. So they are coming into an order that is completely rearranged and reshuffled. So when you then have Hi-C data from these mouse cells, when you look at the human chromosome, which you can distinguish because the reads will map to human genome and not to mouse genome, when you look at the Hi-C data for this chromosome, you can see all of these patterns. That wouldn't be there if that was a normal chromosome. On the left, you're seeing a normal chromosome and how Hi-C map should look like. So then this was our challenge. We are like, okay, let's see whether we can go into this and then develop a computational method that first detects all of these breakpoints and all of these small fragments, and then essentially, just like Rubik's Cube, shuffles them back into place, into the right order, and then gives us, in the newly created genome sequence, gives us something that, is the right sequence. So Abhijit has developed an algorithm there, and this was not a very difficult one. We used genetic algorithms here to find these correct rearrangements. And then we started thinking, okay, this is very important, the complex rearrangements, and we can solve them. But this is a mouse model where, solving this may or may not be as valuable. Can we look at something where these kind of rearrangements happen in reality and in relevant human diseases? So that rearrangement that I just mentioned, when it happens on its own, it's called chromathripsis. Essentially, it's a catastrophic event that hits a chromosome. And then a chromosome like this one, when this happens, which we believe happens immediately, not like one by one, when this happens, it does get shattered into pieces, sometimes tens or hundreds of fragments. And then, as I mentioned, then some of those pieces get lost, some of those pieces get amplified, and some get put back together, but not in the correct order. And then this is progression towards cancer after that. So where does this happen? So the interesting thing is that when we first started studying this, the common knowledge was that, well, yes, chromathripsis is an important event, but it's only happening in like 2 to 3% of cancer genomes. So it's kind of like a rare event. And then the Pan Cancer Atlas came with a paper that said, no, it was just the way that we were looking at these things with the old technologies. Now that we can look at it with whole genome sequencing and newer technologies, actually chromathripsis happens in 30 to 40% of cancers. So all of a sudden, the profile of what we are studying has gone up by tenfold, and we already, by then, we have identified the one kind of disease that we wanted to study, which is the pediatric B-cell leukemia, and a specific high-risk subset of it, which there's chromathripsis. So that subset is called IM21, and then if you look at IM21, it is the one with the worst survival and the highest relapse risk. Luckily, this is from quite some time ago. After realizing this, people and doctors have started treating these kids more aggressively, and an aggressive treatment actually makes a survival chance much, much higher, although it is still one of the worst ones, and relapse rate still remains high. It's a very rare category within all BALLs, pediatric BALLs. Only 2% of the kids will have this, and those are the ones who are not going to fare very well comparatively. And it's marked by that chromathripsis that's again on the chromosome 21. So what did we do? We collaborated with clinicians and other scientists, and we have gathered the pilot set of samples. These are difficult to get, like one hospital or one center is not going to be able to find you. Even for 5 of these cases, you really need to look for bigger collections of them. So we were able to get a pilot 9 patient samples, and then 10, again, these are control, but these are leukemia, but non-IAMP, not the one that we study. So we have done RNA-seq, whole genome sequencing, the Hi-C, the experiments that I talked about, the single-cell RNA-seq, and more recently, long reads with the PacBio platform on all of these samples. So what we have first confirmed is that really these kids with the IM21, their chromosomes are rearranged, and there are large amplifications that are happening in the chromosome 21. And a few of the control cases actually seems to have that. When you go into the high C maps we have generated, you see this kind of pattern. When you zoom in a little bit, it's clear that you have these shufflings and you have these small fragments, and they're all over the place. something for us to resolve. When we looked at our small set of samples with a huge cohort study of 700 pediatric samples, we have seen that our cases, most of them are co-located with these high hyperdiploid cases and pH-like subsets that were identified before, but a couple of them corresponded to these ETV6, a specific gene, and the mutations of it. And then when we look at it, we see deletions for those two samples. We see deletions of ETV6. And one of our samples corresponded to PAX5 alterations group. And then when we look at this sample, we saw that it does have a PAX5 deletion also in our case. So we are trying to just put our findings in the context of what's been known and then also what's novel. So large amplifications in chromosome X is what we found, and then chromosome X deletions have been found before, but I think this is the first time we are showing that there are large amplifications of this chromosome. And more interestingly, we have found a region where we see this same pattern, same rearrangement pattern across two different samples. And this is recurrent, and in this case, it doesn't come with any other interchromosomal translocation or rearrangement, whereas in this sample, it's very interesting that it does have a rearrangement also interchromosomally. And in another patient sample, there is no rearrangement in this region. So this may have important implications in terms of stratifying patients into disease groups or their therapies. And then another rearrangement we found is a translocation that's unbalanced translocation. So essentially, a piece of chromosome X gets amplified and sticks into the end of the chromosome 12 and creates this pattern. So it's amplified. It's not deleted in its native location, but it's there. But it's also goes and translocates. It's this little piece on chromosome 12. So when we look at these rearrangements in the single-cell RNA-seq data and whether they are clonal or subclonal, what we have identified was that some of them are subclonal. So there are two different kinds. You look at the same patient samples, and then none of them are happening in control, fetal bone marrow B cells here. When you look at these when you look at this one patient and all of its single-cell data, you can see that the chromosome X amplification happens only in a subset of cells and not in the others. So this does tell you that you really need to look at, in cancer in general, you really need to look at clones and subclones and then try to understand what comes first and what comes later. So then we looked at the single cell data of these different patients. And as you can see, IM patients group in one place and all the other control patients and control cells grouped separately. We have then went on to looking at the gene expression, and then we have identified differential genes that demarcate this IM21 and separate it from the other cases of leukemia. And obviously, chromosome 21 had certain genes that where this is happening, but we have found, very interestingly, genes that are not on chromosome 21, but are also markers of this specific disease subset. So some of these targets are very interesting, and we are now working on them. Actually, two of them, we have some follow-up experiments and follow-up work. So I'm very hopeful that we will be able to come up with new leads for kids that are of this subset, and then maybe potentially targets that are going to be useful for them in the therapy. So this project is essentially all spearheaded by Abhijit in the lab. So when we needed, Adam joined. We had a small wet lab, and we still have it. Adam has generated some of these data. Vijay's lab has generated a lot of the single cell data and also RNA-seq and whole genome sequencing. Now that we have a lot of data, Abhijit needed help. So Laura, an undergrad student from Mexico, she joined the lab and now helping Abhijit. And we were able to get some funding from foundations, a Spark grant and a Conrad Prebys Foundation, and the samples we gathered from children's oncology groups. So we are thankful to all of them. Yeah, but with that, I'm going to end, and hopefully we will have time for questions. That I really thank all my lab members and my collaborators. We are a very diverse lab, as you can see. We have people from all over the world. I really value this and appreciate that I'm able to talk to people with such different perspectives, and our funding, and of course our GitHub, where there's a whole bunch of very useful tools if you are in this field. And also thankful to our collaborators who helped us apply all of the methods that I'm talking to you about today into different kinds of domains and made the difference in such domains. So with that, I'm hoping everyone is still here and not asleep, and we have some questions.
Thank you so much, Dr. Ferhatay. We appreciate for your very informative presentation. I'm moving on to the question and answer section.
All right.
I'm continuing with the first question. Can 3D modeling studies use the data to characterize promoter-enhancer interactions in single cells or to validate and interrogate predictions about genome folding?
That's really nice. So yes, the short answer is yes. The longer answer is that the single cell data doesn't have the resolution to really go after such fine scale enhancer promoter-like interactions at this moment. But as I have shown, imaging has gone a long way in imaging in a high throughput DNA. So it is now showing us certain enhancer promoter interactions at the single cell level. And people have started like going from single cell data to such loops, but it is very difficult and challenging at this point.
Thank you so much. I'm moving on to the next question, coming from the YouTube participants. Profiles of chromatin interactions captured by the Hi-C are formed by the joint action of different mechanisms. So does this profile to result in extreme complexity of Hi-C libraries?
Yeah, good question. It does, yes, exactly. So, and then that extreme complexity is why we need more sequencing than, couple, maybe at least an order of magnitude more sequencing than other assays such as RNA-seq, ChIP-seq, etc., which you can do with tens of millions of reads. Whereas here, the complexity of different types of loops is said mediated by different proteins and different mechanisms, and also the number of such things. And of course, also the dimension, right? I mean, when you do ChIP-seq, you have an array that you are trying to fill with numbers. When you do Hi-C, you have like a 2D n by n rather that you're trying to fill with numbers. So extremely complex libraries, and then even more complex with the micro-C and other new techniques, and they need to be sequenced, and their analysis also becomes a little bit tricky if you don't know what you're doing. So that's where we are hoping that we can help.
Thank you so much. The next question is it possible to predict chromatin interactions for different cell types lacking experimental Hi-C data?
Oh, great question. That's a bioinformatics student, I would think, who's asking this question. So yes, to a certain extent, it is possible. And there have been papers that are using tensor decomposition and other deep learning models where they use correlations of other data sets such as ChIP-seq and so on and so forth, or chromatin accessibility to determine cell type specific Hi-C interactions when you don't have the Hi-C data, or when you have it and you can cross compare. So it is to a certain extent, but my approach to a lot of these can we predict X from Y kind of problems is that it does teach you a lot, and they're good projects for students to learn how things work. But it is very difficult to convince, let's say, a biologist or immunologist to say, oh, like you don't have Hi-C data in that. Let me go through this whole process of doing X, Y, Z and all these super complicated methods and predict it. So they would rather generate it, right? It's kind of, for me, it is difficult in that sense to justify just using these predicted, loops or predicted contact maps when it is possible to actually generate the data. But there are cases when this would be very useful, when there are not enough cells, or you're not able to access them for this experiment, whereas you can perform the others and then use the other experiments to infer that. There are such cases, but they are not as common. Great question, though.
Thank you for your answer. I'm continuing with the other question. Too many methods source could be the reason of chaotic information source. How can we be sure about reliability of genome data?
I have no idea what this question is trying to ask, but reliability of genome data. Can I skip this one? Because I mean, maybe it's the wording.
Of course. I'm continuing with another question. If we fully reached out the genome mapping for gene modeling compounds within protein bindings and epigenetic factors, it can be scaled up chromosomes modeling to biologic regulation viewing.
Yes, I guess if you reach out genome mapping or gene modeling, so we are doing genome mapping and gene modeling and all the protein binding epigenetic factors. So if the question is whether we can use those to do the 3D modeling of the chromosomes, then the answer is pretty much the same as what I mentioned, that yes, you can, and yes, there are algorithms, again, in tensor decomposition, etc., using that one-dimensional signals, epigenetic factors, etc., to predict, to a certain extent, the cell-specific regulation or regulation weaving, I guess. Yeah.
Thank you so much. I'm moving on to the next question. It is crucial to improve the computational performance of 3D modeling methods in understanding the genome structure-function relationship. Could we achieve this goal by developing highly parallel code that scales well across several CPUs or GPUs, or by creating our lattice models that allow for much faster stimulations and parameter scanning?
Yeah, so yes, the answer is yes to this. And again, we want to, first of all, it's like software engineer, hardware engineer kind of thing, right? So we want to, first of all, come up with the right kind of algorithms and methods that they are in their own, like scalable and as efficient as possible. But when that fails for maybe like structural modeling and so on and so forth, then we really need to go into these. And then some of the algorithms we have developed are efficient enough that and that they are these shamefully parallelizable, that you can run it across different chromosomes separately, and then that's your parallelization. But some more complicated models and maybe molecular dynamics simulations and so on and so forth, they need these type of things. And more recently, I haven't talked about it, but we have some data, some method development in single cell RNA-seq analyses where we really needed to go into GPUs and parallelization and learning parameters and tracing the parameter space much faster with the GPUs. So all over the place, this is useful.
Thank you so much. How can we get a depth of sequencing to enable robust and sensitive detection of individual chromosomal contacts?
By spending more money. So you just need to sequence more, and it does cost a lot of money. So there is a recent paper that's suggesting for proper differential analysis of contacts that you need to have at least a couple replicates, of course, but also like 6 billion reads per condition. 6 billion reads is like almost the whole NovaSeq S4, and that is like 30,000 plus. So it's just money, essentially, more sequence.
Thank you so much. We will appreciate for your answer. Another question from YouTube participants is, fit high C2 make any assumptions about the size or the structure of the genome?
It doesn't actually, yeah. That's a good question. I mean, neither FitIC nor FitIC2. It doesn't make any assumption. It can be any genome, any, as I said, people have used it for yeast, for cotton, for, I don't know, mouse, human, and all of that. It does infer the background itself from whatever data you give, and that's one nice feature.
Thank you so much. I'm continuing with another question. Which methods the best for genome-wide and promoter-wide detection of potential gene regulatory interactions at high resolution using low cell numbers?
Yeah, that's a good question. So there are the major methods, especially when you think about promoter or like regulatory element centric, there have been, other than HiC, of course, there have been high chip, and also promoter capture or region capture or enhancer capture. You can talk about capture-based chromosome conformation assays, and all of them have improved to a very large extent in the number of cells that they need. So the capture experiment started from 100 million cells. Now I think they are down to about 1 million. High chip has started from a couple million. It's down to a couple 100,000. So my personal favorite is high chip, because we have used a lot of high chip data, and we believe that the dynamic range of chromatin contacts that you can capture from high chip using low cell numbers is really sufficient to detect interesting biological differences. But yeah, depending on your applications, it may change.
Thank you so much. We would appreciate. Another question is Fit high chip be able to call loops between peak and non-peak regions?
Yes, that we allow, because there are very interesting and important interactions we think are happening, and they have different chromatin marks on the two sides. And one anchor may have an activating mark, but the other may be missing it, but it may still be important. So if we only require that the peak-to-peak interactions are discovered, then we would be missing such, and then that's why we develop. But for the people who are really interested in maybe like these structural loops between two CTCF sites, etc., we have a peak-to-peak version too, so it's just a user choice.
Thank you so much for your response. Can DC high C also be applied to the comparative analysis of other coarse-grained genome-wide signals, such as replication timing and lamine association?
Yeah, someone read the paper, it sounds like. Okay. Yes, so we mentioned this in the paper, and that signals from anything else that covers the whole genome, such as the high C compartments and replication timing, lamine association, and other assays that you can think of. Yeah, it's essentially a multi-way comparison tool for genomic signals, and it does, and we don't have it in the paper, but we have applied to replication timing data and got pretty good results in working on that.
Thank you so much. I'm continuing with another question. In high C and other methods, is the presence of each loop associated with higher gene expression? How can we differentiate this?
Yeah, nice question. Something that the field is also always thinking and asking about. The answer is that not always, right? Generally, when you do an aggregate analysis, generally the presence of a loop, especially if it's from an activating chromatin kind of H3K27 acetylation, etc., kind of histone modifications and their high chip data, then you do have this correlation of increasing interactions correlating with increasing gene expression. But again, there are other loops, there are silencing loops, there are repressors in the genome, there are insulating regions, and looping to them may actually decrease chances of gene expression. So how you differentiate this is through integrating it with the gene expression data and the looping data. But how you can model this is very difficult. Like a genome-wide model like certain assumptions will not work for every single loci. So
Thank you so much. As a last question, is the any methods for distance-dependent analysis of high-C interactions within and between chromatin compartments?
I see. Yeah, I'm not sure exactly. This person can reach out later, but so some of the methods that we are talking about, regardless of the compartments, they do allow for the distance dependence and its correction, but nothing to the effect, if it's what I'm understanding, nothing to the effect that the distance between compartments and then those maybe aggregate plots and other things that you are doing with compartment to compartment interactions being accounted for their distance. Not that I know of. Of course, we do regular normalizations and all of that, but nothing that's properly done.
Thank you so much. And I would like to ask something about what advice would you give to students studying in scientific fields?
Yeah, I mean, in scientific fields is broad. I think I can talk about maybe bioinformatics and students who are in the same kind of track and mindset that I have been in. And the one thing that I'm learning as we progress through our careers is that bioinformatics is a very exciting discipline. But it is not a discipline where you can stick to one hammer that you have and then expect nails coming and then you hitting them, right? So it's not a place where you can just rest easy because you have developed something and then it will work forever. So it is extremely dynamic, and the right kind of bioinformatics student will have the mindset that they will come and they will work on the interesting problems of the day and maybe develop some very important tools, but tomorrow or next year they might be working on something quite different. And they should be excited by it rather than, being afraid of it. So if you have that kind of mindset, I think then you might be the right type of person in bioinformatics, that you should be open for change and dynamism and collaborations and being scooped and your tools being kind of obsolete within a couple of years and so on and so forth. If those are okay with you, then bioinformatics is the right place, but it's not a place where you can come and rest easy and works at a very fast pace compared to wet lab and other things. So you may have an idea, but there's a lot of other smart people. The moment some data comes, you may have a good idea, but a lot of other people will have that good idea too, right? There's lots of smart people. So you really need to act fast and then execute. So you really need to have those skills, in our field at least.
Thank you so much. On behalf of myself and all our participants, I would like to thank you for spending your valuable time, transferring your knowledge to us, and for this informative presentation. Thank you
Thanks
so
a lot,
much.
Nazlı. And then thank you to the whole group. And this has been I wasn't able to watch it with the time difference and all of that, but I scanned through the YouTube and it seems like an amazing lineup, such a diverse set of people. And I'm glad that you're kind of like connecting all our scientific researchers working in other countries back to our country, and then helping our students, motivating them. Hopefully, if they didn't understand anything from this, the one thing that they can take away is that if they're motivated, I'm sure they are smart. If they're motivated and if they're up for the challenge, there's no limit. So good luck to everyone.
I wish you healthy days.
Yes, you guys too. Take care.
Değerli katılımcılarımız, Bioinforange platformu olarak düzenlediğimiz Bioinfocongress IV etkinliğimizde çalışmalarına yurt içinde ve yurt dışında devam eden değerli bilim insanlarının katılımıyla toplamda 12 seminer gerçekleştirilmiştir. Bu kapsamda 12 etkinliğimizin 6 tanesine katılım sağlayan izleyicilerimize İngilizce katılım belgesi verilecektir. İçeriklerimizi ve duyurularımızı sosyal medya adreslerimizden takip edebilirsiniz. Burada sözü Bioinforange platformu kurucusu Mehmet Çalıseki'ye bırakıyorum. Hepinize katılımınız için çok teşekkür ederiz.
Merhabalar tekrardan. Bilim dolu 2 günün sonuna geldik. Öncelikle davetimizi kabul edip bizlere bilgilerini paylaşan, değerli önerilerini sunan 12 bilim insanına çok teşekkür ederiz. Sonrasında ise tüm katılımcılarımıza teşekkür ederiz. Ama en büyük teşekkürü de bu organizasyonda emeği geçen ekip arkadaşlarıma etmek istiyorum. Bir ekip işi yapıyoruz ve 12 konuşmada da 2 gün boyunca 5. kongremiz oldu bu bizim. En sorunsuz kongremizi geçirdik. Bu konuda çok mutluluk duyuyoruz. Bu kongrede özellikle İngilizce olmasını istedik. Hocalarımız da İngilizce sunumlar yaptı. Çünkü bilim dilini yani İngilizce olarak kabul ediyoruz. Araştırmalarımızda, çalışmalarımızda her yerde İngilizce karşımıza çıkıyor. Bu zamana kadar da hem webinarlarımızda hem de kongrelerimizde 5 tane kongreden daha öncesinde de Türkçe etkinlikler yaptık. Türkçe konuşmalar oldu. Bu kongremizde de İngilizce, tamamen İngilizce olmasını istedik. Böyle anlamayan, biraz İngilizcesinden dolayı zorlanan katılımcılarımız olmuştur ama bu da bilim yolunda bir zorluk, bir challenge denilebilir. İngilizce öğrenmek mutlaka gerekiyor. Burada anlamadığınız noktalarda YouTube kayıtları YouTube adresimizde, bu canlı yayınlar YouTube adresimizde bulunacak. Anlamadığınız yerlerde tekrardan geri sarıp dinleyebilirsiniz hocalarımıza. Ben öncelikle burada bulunan, arka planda bulunan arkadaşlarımı da ekranda davet etmek istiyorum. Herkes kameralarını açıp katılabilirse. Katılım belgelerini arkadaşımızın da belirttiği gibi 12 konuşmadan 6'sına katılan katılımcılarımıza 2 hafta içerisinde katılım belgelerini Certify üzerinden göndermeye çalışacağız. Bunun haricinde katılım koşulları sağlayan katılımcılarımız arasından da bir çekiliş yapacağız. 3 adet mikropipet anahtarlık armağanımız var. Onun haricinde de 2 adet Pokemon figürlü tüp tutacağı hediyemiz var. Bunlar size kongre hatırası olarak kalmasını istedik kazanan arkadaşlarımıza. Ben arkadaşlarımı şu an davet ediyorum. Hepsini. Betül kamerasını açmamış ama kamera açmak isteyenler açabilirler. Süper. Biz burada doktora, yüksek lisans seviyesinden birçok öğrenci hiçbir kar amacı gütmeden tamamıyla gönüllü olarak ülkemizde bilimin gelişmesi amacıyla, bilimin paylaşılması amacıyla elimizi taşının altına koyup sorumluluk alıp bu konuda faaliyetlerimizi devam ediyoruz. Yarın itibariyle 20 Mart tarihi itibariyle Bioinforange 3. yaşına gelmiş olacak. Biz bu kongreyle birlikte bu doğum gününü, Bioinforange'ın doğum gününü kutlamış oluyoruz. Ayrıca web sitemizde şu an bilimsel dergimiz olan Bioinfo Journal'ın da 6. sayısı bulunmakta. Biz o dergimizi de çok böyle arkadaşlarımız bütün görsel çizimlerine kadar derleme yazılarına her şekilde kendi emekleriyle hiçbir yerden almadan hazırlıyorlar. Bu çok büyük bir emek. İlgi göstermenizi, mutlaka incelemenizi diliyoruz. Bundan dolayı yani destek verirseniz bu konuda çok mutlu olacağız. Ayrıca biz mesela 6. sayısı için bu zamana kadar dergi sayılarımızın da basılı sürümünü maalesef elde edemedik. Belki aramızda dinleyenler vardır, destek olabilecek insanları yönlendirecek insanlar vardır, katılımcılar vardır. Bu konuda da destek olabilirsiniz. Tekrardan son böyle uzattım. Var mı arkadaşlarımız arasından bir konuşma, söz almak isteyen? O zaman kapanış cümleleri olarak tekrardan Bioinfocongress IV etkinliğimize katılan, davetlerimizi kabul edilerek konuşmalar gerçekleştirilen 12 bilim insanına çok teşekkür ediyoruz. Siz değerli katılımcılarımıza çok teşekkür ediyoruz. Kongrenin organizasyonunda emeği geçen tüm arkadaşlarımıza teşekkür ediyorum. Sizlere maalesef depremden dolayı buruk bir bilim şöleni gerçekleştirdik. Ekip arkadaşlarımızdan Zeynep Nurçıra'yı da kaybetmenin hüznünü yaşıyoruz. Onu anıyoruz ve unutmayacağız. Diğer depremden vefat eden tüm insanlarımız için de başsağlığı diliyoruz. Tekrardan bilimin ışığında, bilimin önemini hepimiz farkındayız. Ülkemizde de bilimin ışığında mutlu günler görmeyi diliyoruz. Herkese tekrardan çok teşekkür ediyorum. Bizim hep sözümüz var. Bilim paylaştıkça güzel, bilim paylaştıkça ilerler. Bilimle kalalım. Bay bay.